terus berlayar dan berlabuh untuk ilmu pengetahuan. Penjelajahan kolaborasi ini diharapkan dapat bermanfaat bagi perkembangan ilmu kelautan di Indonesia dengan mengenal lebih dalam kekayaan lautnya.
Halo semua, kenalkan kami kelompok riset dinamika lingkungan laut dalam atau KRIS Dalam dari Pusat Riset Laut Dalam, Badan Riset dan Inovasi Nasional. Dalam video ini, kalian akan menyaksikan informasi singkat terkait KRIS Dalam. Selamat menyaksikan dan kita akan berjumpa lagi di akhir video ini. Dilihat dari berbagai aspek kajian, Indonesia memiliki sumber daya kelautan yang sangat melimpah, baik di kolom air atau water column, di dasar lautan atau seabed, dan di bawah dasar lautan atau subseabed. Adapun yang harus dilakukan saat ini adalah memahami kondisi lingkungan di mana sumber daya kelautan ini berada. Kami adalah kelompok riset yang dirintis sejak tahun 2019 untuk melakukan kajian kondisi lingkungan, mengungkap dinamika yang terjadi, dan mengidentifikasi ancaman yang mengintai, baik di dasar lautan secara umum dan khususnya di kolom air dengan kedalaman lebih dari 200 meter. Potensi area kajian laut dalam sangat luas. Indonesia adalah negara dengan luas lautan 6,4 juta kilometer persegi, di mana 32% adalah lautan dengan kedalaman kurang dari 200 meter, 25% memiliki kedalaman antara 200 sampai 2000 meter, dan 43% memiliki kedalaman lebih dari 2000 meter. Riset-riset yang sudah dan sedang dilakukan adalah riset karakteristik dan dinamika fisis laut dalam, riset nutrien dan karbon laut, dan riset pencemaran mikroplastik. Sedangkan topik riset ke depannya adalah riset biogeokimia laut dalam, riset pencemaran di laut dalam, dan riset hubungan lingkungan laut dengan kondisi makhluk hidup di laut dalam. Melakukan eksplorasi laut dalam tidak diragukan lagi sebagai salah satu kegiatan yang menantang dan frontier di Indonesia. Hal ini juga mendukung salah satu visi kemaritiman 2045, di mana menjadikan Indonesia sebagai poros maritim dunia. Melalui kelompok riset ini, diharapkan dapat memberi kontribusi dengan mengungkap teka-teki terkait karakteristik dan dinamika laut dalam, serta bagaimana kondisi tersebut mendukung kehidupan makhluk hidup yang belum pernah terungkap sebelumnya. Selain itu, peran laut dalam mengatur iklim global juga menjadi fokus di dalam KRIS Dalam. Menuju lebih dalam adalah semangat kami, dan kami telah mencoba sejak tahun 2018 untuk mengembangkan diri, menggeser batas-batas, dan mencari kebaharuan melalui kegiatan ekspedisi ilmiah di perairan Indonesia. Banyak permasalahan dan topik penelitian laut dalam yang perlu dikerjakan saat ini. Ratusan fitur dasar laut yang perlu diungkap, jutaan persegi luas lautan yang perlu dijelajahi, dan tak terhitung jumlah tantangan yang perlu diselesaikan. Kelompok riset kami saat ini terdiri dari 4 orang peneliti dan 7 orang teknisi. Dan jumlah ini masih jauh dari kondisi ideal untuk mewujudkan ide dan mimpi kami untuk riset laut dalam di Indonesia. Kami adalah jiwa-jiwa muda yang memiliki ide dan tujuan besar dalam riset laut dalam di Indonesia. Namun kami belum memiliki Anda, yang merupakan talenta-talenta berpengalaman dalam bidang ini. Mari bergabung di KRIS Dalam dan kita wujudkan ide dan mimpi kita untuk riset laut dalam di Indonesia.
Good morning. Welcome and selamat datang to the first ISEIME, the International Symposium on Eastern Indonesian Marine Ecosystems. This conference is organized by the Research Center for Deep Sea of BRIN here at the National Research and Innovation Agencies. We are one of 10 research centers under the Research Organization for Earth Sciences and Maritime here at BRIN. My name is Intan Sucinarhati. I'm the head of the Research Center for Deep Sea, and I will be your host for this morning's plenary sessions. Ladies and gentlemen, why are we doing this conference? Why here we are gathering together as researchers of the deep sea? BRIN, as a new institution, has set 3 priority research areas, which are green economies, blue economies, and digital economy. Therefore, BRIN recognized the importance of advancing, of pushing further into the research of the deep sea in Indonesia as part of the international community of the global science. As we all know, the deep sea is our last frontier on Earth. The sections of the oceans below 200 meters, often we refer to as the deep sea, covers about 65% or more than the majority of the Earth's surface. Yet, we only know a bit of it. We only have mapped just a small portion of it. Therefore, we need this push for focusing on the deep sea. We know the extreme environments of the deep sea brings many scientific discovery, the newest of the newest discovery of today. The deep sea holds organisms that adapt to the darkness, the wide-ranging temperatures, the extreme pressures. Therefore, it prompts the need for sustainably using them by first we need to map them, understand them, and therefore we can protect them for the use of humankind. Now, why a gathering, let's say that this as a symposium on the Eastern Indonesia? Because once we remove the top 200 meters in Indonesia, which is a very important area in the Indo-Maritime climate variability, then we are pretty much left with the eastern and the outer parts of Indonesia. So therefore here, thank you again for joining us to get together to further our understanding. And we also understand that not all of us have dived into the deep sea, but we can chip in our expertise, how we can push further and go deeper into the science. Therefore, with the aim of BRIN to support the blue economy research, here at the Research Center for Deep Sea, we aim to contribute to BRIN's blue economy research through the sustainable use of deep sea ecosystems. Here at Research Center for Deep Sea, we have been focusing on 3 aspects. First, we need to understand our environments with all of its dynamics. So we do have the Deep Sea Environmental Dynamics Research Group. This is a group that studies an integrated study of the physical dynamics, the biogeochemical cycles, the anthropogenic signatures that we know have reached the deep ocean. And we also have the Deep Sea Extremophile Bioecology Research Group Our scientists explore and use novel utilizations of deep-sea macro and microorganisms that have been adapted to extreme high pressure, low temperature, and low light environments. As much as we would like to push and keep inventing in the deep sea areas, we cannot close our eyes on the interactions between the coastal and deep sea ecosystem. Therefore, the connectivity of the coastal and deep ecosystem services is also something that we keep in our heart. There are critical interactions of the upper oceans and the deep sea ecosystems affecting ocean sustainability, affecting humans all over us and our society. With that, again, I would like to thank you for your participation, for your contributions. We are a relatively small research group, but we are very active, full of young scientists and very supportive senior scientists in here. For the next session, I would like to give the floor to Dr. Sam Likumahua. Dr. Sam Likumahua is the lead organizer for this event, bringing us together, working here, bits and everywhere. So I would like to call him to the stage. The floor is yours, Bung Sam.
Thank you very much, Dr. Intan Suci Nurhati. Thank you very much for all of the friends that already are present in this meeting. So good morning, ladies and gentlemen, fellow speakers, and good morning to our lovely presenters and audience for joining us online. Firstly, may I thank each of the ISEIME committee for your hard work in bringing up this symposium to happen today. I personally really appreciate all contribution from many aspects you have brought. Please allow me to say a line from a Lego movie song. Everything is awesome. Everything is cool when you're part of the team. I really like this line from this movie song, especially when I work in a team. Secondly, I would like to thank our speakers for taking part in this event. The quality and caliber of speakers in today's meeting is outstanding. I also want to thank all of you for connecting this morning, and I hope you are looking forward to the discussion as much as I am. This year, Indonesian National Research and Innovation Agency, which is known by the name BRIN, hosts a number of international conferences in order to not only for disseminating research findings, but also to open an international network and collaborations among researchers. One of the conference is the first International Symposium on Eastern Indonesia Marine Ecosystem, which is called as ISEIME. ISEIME is a result from an evaluation of the first symposium on Bandasi ecosystems held by the Center for Deep Sea Research, formerly under the Indonesian Institute of Science, also known as LIPI, in 2017. The evaluation indicated that the need to expand the geographic coverage area of the symposium, hence leads to the initiative of the symposium's organization. As we know, marine ecosystems have been recognized as an important ecological service that contributes to massive benefits for human beings, such as economics, health, science, and technologies. The eastern part of Indonesia in particular has been known as an exotic region for marine research, as it is located in the Coral Triangle Initiative area, which is known as a mega biodiversity region. For decades, many studies have shown interactions between marine organisms and their psychochemical as well as the geological environmental. However, rapid population growth extensively threats marine ecosystems, resulting in environmental degradation and biodiversity loss. Hence, a meeting of marine researchers is one of the important steps in dealing with those problems. During today's meeting, we will disseminate our results of the results of our research and further discuss about marine ecosystems in Indonesia in some topic areas such as oceanography and climate change, marine biodiversity and ecology, marine management and monitoring, marine geosciences, and marine pollution. About 32 presenters will disseminate their research and findings to enrich our knowledge and add more data on marine observation. There is a big hope that this meeting will contribute not only for knowledge of marine research in Indonesia, but also can produce science-based recommendations for Indonesian government, both in local and in the center, in managing marine ecosystems nationwide. Ladies and gentlemen, let me once again thank you for our fantastic speakers for giving their time and insight today, imparting their knowledge in enriching our understanding on marine ecosystem research. And I hope all of you watching enjoy the conference. Thank you very much.
Thank you, Dr. Likumahua. Thank you for you and your team who has been working hard for your commitment in making this symposiums today. Okay, ladies and gentlemen, for the next session, we will be listening to the national anthem of Indonesia, Indonesia Raya. So I would like to invite our Indonesian colleagues to sing along and to our international colleagues to enjoy the anthem. Organizer, please play the song. Thank you. Ladies and gentlemen, the next session, the next item is our photo documentations. We know that we all are scattered in many places, not just Indonesia, but also we have our international colleagues. And we have a saying here in Indonesia, jauh di mata, dekat di hati. Although we are far in sight, we're close at heart. So for this photo documentation, we will have it set up in the classroom setup. So we feel like we're all in the same room, although everyone's virtual. So organizer, please start the photo documentation. Thank you. All right, everybody put your biggest smile and say, ISEIME. And of course, turn on your video first. Okay, organizer, you can start giving the cue.
Okay, wait. Once more. 1, 2, 3. Okay, I will move to the next. Wait a moment. Okay, once again. 1, 2, 3. Give you a big smile.
Wow.
Okay. Wait a moment, please. Again. Okay, finish. Thank you, Bu Intan.
Thank you, Mas Fadli. Thank you. Okay, we are ahead of our schedule, but we can continue, expecting that we'll have a lively discussion from all of you. For the next sessions, we come to the keynote speakers. We are very lucky that we could have Prof. Oki Kanarajasa and Prof. Dwi Listio Rahayu here at ISEIME. Both of them are truly our seniors in deep sea science and pioneers in deep sea research in Indonesia. Now, for the first keynote speakers, we have Professor Oki Kanarajasa. He is the chairman of the research organizations for Earth Sciences and Maritime at BRIN, and he's also our proud colleagues here at the Research Center for Deep Sea. Prof. Oki, as we often call him, is a deep sea microbiologist, focusing on microbial diversity, microbial natural products, and coral disease. Prof. Oki received his PhD at the University of Tokyo, his master's degree at McMaster University in Canada, and his bachelor degree in Sudirman University here in Indonesia. He is a prolific writer, even though with his super busy schedules as a scientist and also as our boss. And we are so proud of him that he just recently won the very prestigious Habibie Award 2022 just this month. And of course, he has been awarded so many awards before. So Pak Oki, thank you again for joining us. We will be giving the floor to you. Please, Pak Oki.
Thank you, Dr. Intan. Just allow me to share my. Do you see my slide?
Yes, I can see it, and you just have to put it in the presentation mode.
How to do that? It's not here.
And we can also help as a backup.
Yes, please.
Okay. Organizer.
Assalamualaikum warahmatullahi wabarakatuh. Very good morning. First, I would like to congratulate the organizing committee for having such a reputable symposium on Eastern Indonesian marine ecosystem. And I'm very happy to be here to share my talk entitled The Quo-Vadis Deep Sea Research in Indonesia. Honorable speaker, distinguished scientists, ladies and gentlemen. Can I have the slide, please?
Oops, sorry. Yes, I have just opened it and I'll be sharing it. Apologize for this technical issues.
Okay.
But it's still also loading on my file. Wait a minute. Seems that the file is quite okay. It seems it's still loading on my screen, or perhaps this other committee organizer can also start.
Yeah, it's
Thank you.
yeah.
Thank you, guys. Sorry about that.
Again, the title of my talk is the Kuvadis Deep Sea Research in Indonesia. Next slide, please. The deep sea is broadly defined as the ocean depth of 200 meters, or a depth where light begins to fade at an approximate depth, or the point of transition from continental shelf to continental slope. This condition of the deep sea are combination of, in general, low temperature, darkness, less nutrient, and high pressure make them the very extreme environment. For every 10 meters depth, there is an increase of 1 atmosphere. So we can expect that the deepest part of ocean is 11,000 meters. So we experience the pressure up to 1,100 atmosphere. The deepest part.
Is everything okay, Pauki?
Oh, okay.
Okay, take your time.
I cannot see the slide yet. Okay. Okay. The deepest part is Mariana Trench. Of the shallow deep is the deepest part of the ocean at 11,000 meters. Next slide, please. Indonesia is the largest archipelagic country with 17,500 islands. 70% of its area are waters. It has 108,000 kilometers shoreline, and it is one of the mega diversity country and a global center for marine biodiversity known as the Coral Triangle. Next slide. If we look at the bathymetric of Indonesian waters, there are indeed deep sea area indicated by blue areas. Most of them can be found in eastern areas of Indonesia, and the deepest part is the Weber Point of Bandasi at a depth of 7,440 meters. Anyway, this is a very challenging area to be studied. Next slide. This slide shows how the physical pressure on the impact of the physical pressure on the noodle cup. When I was in Japan to do my doctoral degree, I went to several research cruises. As for example, in the Middle East, the noodle that I brought to the Mariana Trench at a depth of 10,000 meters. So you can see the curry cup, normally similar to the sedap noodle cup, but now it's become very small after experiencing the pressure, high pressure effect. So we can imagine that there are micro and macroorganisms that could stand and survive at the depth of 10,000 meters. Next slide. Of course, when we talk about the deep sea extreme ecosystem, there is also submarine volcanoes, seamount, and hydrothermal vent that support the growth of macro and microorganisms that offer new potential. Deep sea is very wide ecosystem that need to be a multidisciplinary team to do this ecosystem. For example, we are talking about the diversity of micro and macro in the molecular scale. We talk also about the paraphilic biodiversity that can survive in the extreme environment. Also, this offer economic mineral deposit that need to be explored. So when we're talking about the deep sea, we need to focus in several options that need to be explored. But in this regard, I will focus my talk on the microorganisms that are living in the deep sea environment. Next slide. To be able to explore the deep sea environment, we need to have access. For that, we need some investment and very expensive equipment. For example, we can use the Niskin butterfly, high pressure serial sampler. We can use pressure retaining sampler, remotely operated vehicle, man submersible, for example, Shinkai 6,500 meters, Shinkai that means deep sea. And then we can use also the unmanned kaiko operated by chopstick. Kaiko means trains. So we can put several investment in order to make our dream come true by exploring the deep sea environment. Next slide. The development of study in deep sea has created a new branch of marine microbiology known as a deep sea microbiology that caters study of high pressure adapted microbe known as a badophile, low temperature adapted bacteria known as a psychrophile, and the high temperature microbe known as the thermophile. All these microorganisms need special techniques in order to be able to explore their potential in the future. Please. This is an example of the extreme barophile from Mariana Trench. It was collected by Chopstick Group. They collected sediment and at a depth of 11,000 meters, and they study of two isolates that been collected from Mariana Trench. The trench are isolated from sample of the world's deepest sediment, which was obtained by unmanned deep sea submersible Kaiko in Mariana Trench. As you can see here, the two isolates are similar to Sewanilla bendica, which has optimum growth pressure at 80 mega atmospheric, and also they have also another one is Moritella yenosi at 70 mega pressure. And they are also indicated that these two isolates capable of growth as indicated by the presence of ORF1-2 primers and ORF3 primers. As you can see here, both of them are very obligated barophile, only able to grow at least 500 atmospheric pressure. Next slide. This is the global data of deep sea microorganism, and we can see here that there is no report regarding the true deep sea microorganism from Indonesian area. The microbial ecology of marine environment of the Indonesia Sea potentially to be developed mostly at the depth of this slide. At the depth, mostly at the depth less than 200 meters. And there is a neglected study regarding the deep sea area from a depth of 200 meters to the bottom of the sea, especially to explore genetically encoded deep sea secondary metabolite that has been rarely exploited due to the difficulty in our equipment. Next slide. This is our research on the bioprospecting of marine bacteria from the Java Trench. We conducted this study as part of the Triumph expedition in 2019 by using research vessel Barunajaya 8 from November 18 to December 2. We collected the sample from a depth between starting from 1,000 to 4,000 meters with average average temperature at 5.6 degrees Celsius, salinity about 34, and dissolved oxygen concentration 2.2 milligram per liter. So this is representing the truly deep sea area in the Java Trench. Next slide. Here's the genome characteristic of our isolate from the Java Sea, Pristia flexa. We can see various attributes that have been characterized in this genome. We can see the genome of Pristia flexa is about 4 billion pair with a high DNA coding region up to 83%. The DNA coding region are made up of a coding sequence with about 5,000 coding sequence. About 5,000 coding sequence were detected, but only 3,000 coding sequence were successfully annotated into two database. This indicates that there are still around 2,000 coding sequence that has not been annotated. Coding sequence with unknown function have potential that will encode novel byproduct that might be utilized in the industry. Next slide. We use the molecular approach to study the map of a circular genome of Pristia flexa from the Java Trench. We can see various attributes that have been characterized for this genome. We can see that the genome size is also about 4 million base pair with high DNA coding region up to 83%. The DNA coding region are made up of a coding sequence with about 5,000 coding sequence. This is a circular genome that map every coding sequence annotated function into various categories. For example, cellular processes, signaling, information storage, and processing and metabolism. This genome data is very powerful because it can map detail the blueprint of an organism. From this map, we can see that there are still coding sequences that are poorly characterized, whose functions are not yet known and have potential to become novel bioproducts. So this map showing us that our bacteria from Java Trench has a potential to produce deep sea bioproduct. Next slide. This table shows the list of biosynthetic gene clusters contained in genome of Prista flexa. This data was obtained by genome mining method using ISEIME software, and the gene cluster that encoded various proteins that play a role in the formation of particular byproduct. The biosynthetic gene cluster can predict the type of the end product. The potential novelty of a biosynthetic gene product can be seen from the low similarity and no hit in the KC blast results. As you can see here, there are two potential products from the deep sea from Java Trend, which is the NRPS like similar to bacitracin 22%, and the product of lasopeptide about 80%, but they have a very unique sequence that's very different from the reported sequence so far. Next slide. The following figure is a visualization of the structure of the gene cluster of NRPS 1 and 2. It can be seen that BCG or biosynthetic gene cluster, there are many genes that gather to form a cluster. Each gene will produce with various enzymes with various domain of function, and this enzyme will work together to form a final product on the right. The product structure shown here is still a prediction. Further study are needed in vitro to determine the actual structure. This is another challenge for us in the near future. Next slide. This is a biosynthetic gene cluster of lycopene according to ISEIME's software. Toxin biosynthetic gene and additional biosynthetic gene are genes that encode same enzyme than the lycopene production pathway. For your information, lycopene is very high economic value, and the only country that export lycopene is Brasilia. So if we can realize our product from the deep sea to produce lycopene, then basically we can provide new lycopene from deep sea water, from Indonesian water for our industry and pharmaceutical industry. Next slide. To conclude my talk, I will introduce you the current status of approach with new approaches by combining metagenomic and synthetic biology as general strategies for mining biopark. The field of synthetic biology is both practically and philosophically reliant on the idea of biopark, a concrete DNA sequence meant to represent discrete functionality. In the first approach, the whole genome shotgun sequencing is performed on environmental pool DNA sample for gene from deep sea and species discovery. The second approach, to explore environmental DNA in a culture-independent manner by isolating DNA from environmental sample and transforming into large insect clone. For example, in this experiment, it is aimed to obtain gene promoter as biopart, which express desired product. The chopped DNA fragment are cloned into plasmid, which has green and red fluorescent protein, then transformed into E. coli. Screening of E. coli is performed using cell sorter on colonies. Positive colonies then further analyzed to know the strength of our promoter. The desired product are picked up and to be sequenced and compared with the whole genome sequencing result at first approach. Other example can be performed to explore coding region of gene of interest as biopart. The chopped DNA fragment are cloned into plasmid which has promoter then transformed into E. coli. Colonies can be chosen then sequenced to know the sequence of the inserted gene. This technology will help us, especially to solve the difficulty of our condition for collecting the isolate from this environment. By using this strategy, we hope that soon we will be able to collect sample and get this mining biopath from the Weber Point, the deepest part of Bandasi in Indonesia. Next slide. Again, thank you so much for your attention. Thank you.
Thank you, Prof. Oki Kanarajasa, for your presentations. Ladies and gentlemen, so we will be having the keynote speakers back to back. But if you have any questions for Prof. Oki, you can put it in the chat box, whether you are in the Zoom for the speakers or for those who are listening this sessions from the YouTube, you can also put your questions on the YouTube box, and we will be compiling those, and we will be asking it at the Q&A sessions of the plenary sessions. Okay, so please keep your questions, put it in the box, and we'll be reading through those questions. Thank you, Prof. Oki Kanarajasa. And for the next speakers, we're also proud that we could have Professor Dwi Listio Rahayu, or we always call her Buyoyo. She is a research professor at the Research Center for Marine and Terrestrial Bioindustry of BRIN. Prof. Rahayu is a marine biologist focusing on the taxonomy of crustaceans, such as erbid crabs and crabs. She has found 2 genera and 71 new species of hermit crab, and 6 genera and 76 new species of crabs. She was also the co-PI of the South Java Sea, the South Java Deep Sea Expeditions, or SJADES, in 2018. Professor Dr. Rahayu received her PhD from the University Pierre and Marie Curie and has received her master's degree also from the same institutions. And before that, she received her bachelor from Bogor Agricultural University, or IPB University. Professor Rahayu has published many publications along with many of the new species she has found. I'm just hoping that one of her species could be named after us, hopefully. And she also has been awarded many awards, such as the Lipi Sarwono Award 2021. Buyoyo, good morning. How are you?
Fine, thank you.
Thank you again for joining us. We are so happy to have you today, and the floor and time is yours. Silakan, Buyoyo.
Thank you, Bu Intan. Good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. I'm very honored to be in this webinar of ISEIME of Eastern Indonesia. So this today I will give the presentation about Eastern Indonesia as a mega biodiversity spot. I will share screen here.
Okay. You could share screen, or we also have backup ready.
Yeah, I will try.
Perfect.
Okay, you can see my slide.
Perfect.
Yeah. So Eastern Indonesia as a mega biodiversity spot. Lesson learned from 4 decades of taxonomy research. So I work already in taxonomy since 1986. So it's almost 4 decades working on taxonomy. So, well, here I started with the very classic about Indonesia, that Indonesia is lies between Indian Ocean and Pacific Ocean, and comprising over 17,000 islands. And most the territory is covered by water, with shallow seas in the western part and some area in the eastern part, deep sea in the east, north, and southern part. With this very long Indonesian territory and this, Indonesia probably has the most diverse marine habitat and considered to have high marine biodiversity. Here, Indonesia, we are in the center of a coral triangle, where coral triangle is recognized as a global center for marine biodiversity and a global priority for conservation. So its biological resources make it a global hotspot of marine biodiversity, and we are has to be very proud that Indonesia is in the center of this coral triangle. A huge volume of water connecting life from everywhere, and over 70% of the surface of Earth is water, and 60, like Pak Oki is saying that 62 till 65 of it is more than 200 meters deep. It is known that Indonesia has a reputation as a center of species richness of marine organisms or center of marine biodiversity, so that many marine biologists are attracted to explore marine biodiversity in Indonesia. Several worldwide marine exploration expeditions made sure that Indonesia was included in the route of their voyage around the world in order to investigate the marine biodiversity in Indonesia. So among others, expedition here is started in the 19th century. Is a physician, and then Koki, astrolabe, and also here Snellius. But all this expedition is mostly is in the eastern of Indonesia, except for this Anambas and Esjadis, that is in the Anambas is in the Anambas waters, and Esjadis is in the south of Java. But besides this all international expedition, LIPI, that it's now is BRIN, has also conducted expedition of Vidya Nusantara or EMIN since 2007. But the question is still, how many marine species are currently described? How many marine species are recorded from Indonesian water? And how many marine species are recorded from eastern Indonesian water? I have made a list here. This is the global marine species in Indonesian, and this is eastern Indonesia. We see here crustacean, mollusks, and fishes is the most recorded Indonesia species in marine. But when we see what is Indonesian species, it is too small. And again, when we see eastern Indonesia, the number is too small to be true. So more exploration on the remote area and more scientists to study biodiversity will increase this number of marine taxa record. So Indonesia is surrounded by shallow water and deep sea with very diverse habitat. New species, rich marine community being discovered at an astonishing rate from all different habitats. Here, shallow water. This is all is in the eastern Indonesia. I went to Halmahera last August, and here is in Halmahera. mangrove just grow on the coral reef. And here is all the boulders and sand and mud, sand and seagrasses, and also how clear water in the coral reef area with the fish around it. And deep sea water, offshore water, the deep sea biodiversity is challenging. To explore the deep sea biodiversity is very expensive. We need a good research vessel with good equipment and have to take into account the timing of the exploration. When I say the timing here is about season. We cannot go to the place where the season is not right. For example, Eastern Indonesia, we can explore only in the month of between September until November, I think, when the water is very calm. Then we can explore the marine biodiversity with the ship and exploring the deep sea very calmly without being disturbed by the very rough water. Of course, if it's intertidal survey, we also need to look at the tide table as well, because when we come to a place when the tide table is not correct, I mean when the tide is very high, then we cannot collect the intertidal organism. So to find a new species rich community, we need to go and explore places that was not well explored. So a place when we do the deep sea, we need a ship and this calm water, or the place where it's very difficult to explore, like a mud in the like this in between mangrove, or the very dense mangrove forest. Here, if we go to littoral, intertidal, and subtidal area, we have to do hand picking or just here the breaking coral, breaking hard coral to find some organism inside, or doing the transect, or using the yabi pump and collecting here in the supralittoral water. Or here, if we go to the subtidal area, and then we need a trawl, the small trawl like this, and in the coral area, sucking or pumping to collect specimen. And for expedition to collect organisms from the deep sea, it is expensive and tough. We have to prepare equipment we will use, the rough sea we will face, and the hard work to separate the species from the mud. We see here, we go, this is in the South Java deep sea expedition. We go to the until 957 meter deep, and then here we collect the specimen with the dredge, and here is with the grab and the mud from the trawl like this. And sometimes the mud is just very clean because we cannot catch anything, because the trawl is just go above the bottom, so it's not really touching the bottom. And then here we collect with the multi-core. Sometimes we do the multi-core until to the 2,000 meter deeps, and to put the equipment in the water and then to get it up again from the water, we need about 2 hours until 4 hours. So we do it until late night. You can see here, it's a late night that we come up with the multi-core. And some new species discovered during 10 years program of the Census of Marine Life. Here we recorded the diversity, distribution, and abundance of life in the ocean. Scientists found and formally described more than 1,200 new marine species, with 1,000 more awaiting formal description. Some new species were from Indonesia. We can see here the fish, hermit, and also this Hemicillium halmahera from Halmahera. And here is some new species that we found during the South Java Deep Sea expedition. I'm sure that as in the eastern Indonesia water, we will also find a lot of new species. In the expedition of Karubar in the year of 1991, is it in the southeast of Maluku? I know that we find about 18 genera of hermit crab and 30 something of new species of hermit crab. So this is all come only a place like K Island and Tanimbar until close to the border with Australia, but all is come from the deep sea. And here we can see all the deep sea creature from the south of Java. We even have this very big sponges, Semperella, that we call it Semperella aesjadis. And we have bivalve that named after Pak Dirham, the former director of oceanography. And we find also his hermit crab that I call it Diogenes berduri, because all the calliped is full with spines. And this all species is a new species from eastern Indonesia. Elamenopsis gracilipes here is from Papua, and this even we call it Ambonensis. This is from the Bay of Ambon, and this is also from Ambon. The type is from Eri, the Ambon Bay, and the three species under this is from Papua. Yeah, this is too many species, too little time, and too few experts. We still a lot of things to discover in the marine habitat. So with all the specimens and the species we found in the marine environment, we cannot work alone. Here is also all the marine species that we find. Here again, the beautiful color of the deep sea creature. And also when I say that is the scary fish from the deep sea with the very sharp teeth. Voila. So science is global. No country have all experts to work with their biodiversity. To know and understand your species, you need to know also what is elsewhere. Cooperation is needed to be able to discover the biodiversity, and good networking is a must. Here, I would like to tell you the story about land hermit crab. This Cenobita lila that we described it in 2014. For 50 years or more than 50 years, this species was a synonym of another species of Cenobita cavipes. And this species also for years is already in trade, in aquarium trade. And people collected and keep them as pet. However, my colleague and I somehow know that this is not the species that we thought it is. But we hesitate to describe as a new species since it is very common in the beaches of Singapore. And how come that nobody described this species if it's so common and but so different? So the last taxonomy publication of this genus Cenebuta is in 1988. Then we start to look at this and check the DNA, and then voila, the difference is so significant. But we have to check other species and other specimens from different locality. So it is why we need cooperation and networking. At the end, we have specimen from Madagascar, Mayotte, Thailand, Singapore here, and also from Philippines and Taiwan and Philippines, and also from Papua. And then after that, we very sure and very confident to describe it as a new species. And it is only published in 2014. And this is a second story. Let us start with a small specimen from Lombok. The small decapod that have 8 legs only, because the last leg is very reduced, so we almost disappear. And from family Hexapodidae. When I check and compare with the specimen in the Museum Natural History in Singapore, I found several bottles with the species of this family. And it turned out the specimen from Lombok is from a genus that has been put into synonyms for more than 50 years. So I start to look at this and then start to collect the specimen from any other museum and asking around with the colleague from the museum. And finally, we have a specimen from here from Singapore, Lombok, Sulawesi, and also here from Japan, China, and also Vietnam, I think. And then because of this, then we start to look at all the genera of this family. And finally, this Hexapodidae, we have 3 new genera with 18 new species from Africa to Australia. This is published then in 2014 as well. And this only possible if we have good networking with all of our museum natural history in the world. Then that's why the good networking we need, and the good relation with all the scientists in the world, that we need to be able to discover more new species, more new genera, and all the distribution of the species. So this is not only deep sea research, but a deep sea research strategy that we can do for the Eastern Indonesia is a cooperation with other countries, networking of various experts, because I thought earlier that science is a global, and no country have all expert in to study the biodiversity, and capacity building for young scientists is really needed. Reference collection supported by molecular DNA, because now the biodiversity, the taxonomic work is not only morphology. We also have to look at the molecular DNA to be able to be sure that it's really the different species. And campaign on the importance of deep sea biodiversity is a very important tool. Thank you. This is I think it's a Krakatau from the Jakarta Bay. Give back to you, Bu Intan.
Thank you, Prof. Yoyo. It's always amazing to hear your stories with all the pictures. Okay.
Okay.
Thank you, Prof. Oki and Prof. Yoyo, for your presentations. I think it's they put a very amazing and very I think we always know both of you are ahead of your time in Indonesian deep sea research. So it's always motivating to hear your stories, not just the amazing science, but your stories of perseverance. In Indonesian word, we will call it, this is like super istiqomah. Yeah. All this work that you do involve a lot of patience and perseverance. And there was a highlight that Prof. Yoyo mentioned. There's few experts. We still need more experts. And we also know that both of you are very active in terms of capacity building, in terms of sharing your knowledge to the younger scientists. So it's really up to us, the younger scientists, to capture those moments, to really recognize which directions of new science that we need to develop in deep sea research, because this is not just ordinary science. This is not easy science. What we need to push is really the novel science for the deep sea research. So thank you for those reminders. So we are going to start into the questions. I would like to ask the committee, the organizer, to show some of the questions that have been compiled from the YouTube and the Zoom. Please, you can share your slides. Thank you. Okay, so we have our first questions for Prof. Oki first. Well, a string of questions from Prof. Oki. This is from Mbak Dewi Zilda, Bu Dewi Zilda. Bu Dewi says, I work with the thermophile bacteria. Do you consider, with all of the facilities BRIN has, is it possible to collect water or sediment samples from hydrothermal vent at the depth of more than 3,000? So that's the first question. Prof. Oki, you could enlighten us. What do we have and what do we still not have to do this line of research? Silakan, Prof. Oki. You may answer the question.
Thank you, Bu Intan. In the current condition, with the facilities at BRIN, even though we have a fleet management unit that cover 5 research vessels, but they all have limitation. Actually, most of them only able to collect sample up to 4,000 meters. We are now constructing a new ship, 80-meter ships research vessel that will be available in 2024 with the capability to collect sample up to 11,000 meters. So this is a good news. And of course, we are trying to buy and to meet the standard for conducting deep sea research by buying some equipment, including the roof with arm and other facilities. We are currently working with a professor from Virginia Tech to develop the equipment to collect sample from surrounding hydrothermal vent in the future. Thank you.
Thank you, Prof. Oki. Yes, I think even with the consolidations of research vessels, our newest research vessels was built in 1998, if I wasn't mistaken. So we need to have that research vessel, the newer one first, before we have all this other facility. And this is also the key for collaborations, right? Sometimes we have the research vessels and we need all these other equipments, and we could actually loan that from our colleagues overseas. Thank you, Prof. Oki, for reminding that information. That will be important also for our international colleagues who would like to have collaboration in Indonesia using research vessels in the near future. So the second question, Prof. Oki, from Bu Istri Yulianti. Thank you, Prof. Oki, for the nice presentations. I have one question. Is the potency of Indonesian deep sea waters as a source of marine bioprospecting derived solely from microorganisms or bacteria? Is there anything else? Okay, Prof. Oki, you may answer the question, please.
Thank you, Quinta. For buoys, of course, marine microorganism is not the only source. We can use macroorganism as well. With the molecular biology technique, we can now collect the sample directly and using prokaryote primers and eukaryote primers to collect the DNA sample from the deep sea. So basically, it's open not only for micro, but also for the macro organism as well. Thank you.
Thank you, Prof. Oki. Okay, the next question is from our lead organizer, Dr. Samli Kumahua. He asks, I have one question regarding the lycopene that, as you mentioned, that Brazil is the main producers. I believe that Indonesia also will be a producers in the future. Do you agree with this statement? Are there any homework that need to be done in the near future for lycopene research in Indonesia? Please, Pauki.
Thank you. Of course, I believe so that there will be a producer in the near future. And based on my work on Java trains, I collaborate with the ISEIME of ITB. We now try to get the bio part that encode the lycopene to be, we will make a gene circuit to make several products based on our sequence from lycopene. So of course, there is other sources from shallow water as well as from the terrestrial for lycopene. But we need to do, especially to do the bioprospecting, because at the moment, most of Indonesian industry and pharmaceutical company, they all import 100% of the lycopene for their product. Thank you.
Wow. Okay. Thank you, Prof. Oki. Okay. So the next questions from Professor Riz Hananto. Prof. Oki, suppose areas of Indonesian deep seas have prospect of mineral resources that needs to be extracted. He hoped not all areas will be conserved, as this policy would permit mineral extraction that we need. Thank you for the kind response. So what's the balance between exploring, understanding, protecting, and using the most of it?
Thank you, Maintan. Thank you, Professor Riz Hananto. Of course, we are currently in the process of identifying the potential of mineral resources, and we have the blue economy concept that not only use the resources but also to conserve as well as to restore our ecosystem or marine mineral resources as well. So not only the coastal or deep sea organism, but we also considering the blue economies as our concept to make use our resources in marine environment. Thank you so much.
Thank you. This is a very important aspects indeed, and also reminder the ecology and economy has to be on the same page, but we still have to keep our ocean sustainable for the blue economy. That's a good reminder. Okay, I think we still have more questions, and we have more questions being compiled for Prof. Yoyo. Pak Oki, the next question is from Pak Fismatman Rusli. I have a question. What's the biggest challenge to conduct deep sea research in Indonesia, particularly in the field of biodiversity and biotechnology? The challenge and solutions, perhaps, Pak Oki. Please, Pak.
Thank you. There are two items I would like to mention. Number one is capacity building. I am at the moment also visiting professor at the ITB ISEIME. So starting 2023, I will conduct several workshops and training for students, postdoc, and young scientists to do, for example, how to isolate the true deep sea barophile that are adapted to high pressure environment. Luckily, I was in Tokyo to learn this technique, so I'm very happy now. There is some new generation very interested to study on deep sea. So that's number one. Number two is we need to have a very well-equipped research vessel. So hopefully by 2024, we will have our new research vessel with equipment that will support us to collect sample from deep sea environment. For example, we can use the pressure retaining sampler that will maintain the in situ pressure of the macroorganism collected from deep sea. For example, Prof. Yoyo may want to collect crab from near hydrothermal vent. Then if we have that equipment, then we will be able to collect and maintain the pressure, in situ pressure of the macroorganism.
Thank you, Prof. Oki. Okay, I think the next question was from Esther, but I can read it. So we can still see Pa Oki and Bu Yoyo's face. This is related to the first questions, similar to Mbak Dewi. Prof. Oki, as a deep sea microbiology researcher who needs complete equipment for exploration, especially in extreme deep sea environments, what do you think about the readiness of Indonesian research vessels to explore barophile organisms? So this is for barophile organism, macro and microorganism. Perhaps there's anything more you would like to add, Pa Oki, from your first answers, please.
Thank you. Of course, we can have a very simple equipment. We can have the pressure vessel that will be connected to the high pressure pump after we collect the sample and put the sample in our vessel, pressure vessel that will maintain their pressure, in situ pressure. And secondly, I have talked with one of the the head of the research center for, I forgot the name, related to technique from the from other research organization. They told me that they will be able to collect sample from DC and they will modify and provide us with a sampler from deep sea as well as the pressure vessel in the on board of the research vessel, as well as in our laboratory in the cold room. So I'm looking forward to have a discussion with them very soon, so that BRIN at least can produce local basic equipment for the deep sea research. Thank you.
Thank you, Pa Oki. That would be great. So the science and technology both advancing, and we could actually expand into the research of engineering involving those with the expertise to have a low cost but effective sampling equipments made by Indonesian scientists. Because here in BRIN, we're very much encouraging collaborations between research centers. So thank you. I would like to read one of the comments from one of our invited speakers to Prof. Yoyo. This is from Prof. Wei Dong Yu. He said, very impressive to see so colorful biodiversity, amazing. Hope to better understand their habitat and ocean environment and climate impacts. So therefore, the collaborations between biology and physical oceanographers. So we have a question for, from to Prof. Yoyo, Prof. Rahayu from Zen. Is there difference in the characteristics of species in Eastern and Western Indonesia? Let's say hermit crab or another species. Is there any striking difference, and perhaps what do you think cause it, if there is? Please, Prof. Rahayu.
Thank you. I think no, not really different. What we have in Eastern Indonesia, sometimes we also find it in the Western Indonesia. But I think that is depend on what the habitat, where we collect, or is it I mean, is it a different ecosystem? Is it in the mangrove or in the coral or seagrass? That is not really different. What is the different is if it's shallow water and deep water. That the deep waters, usually the species crab and hermit crab have a long leg, and then we see, oh, this is a usually from a deep water. That is not really different. Okay.
Because you even mentioned that some of your breakthrough was actually after collecting from even different countries. So there is this connectivity, but the relation to deep sea actually the one that produced more striking difference, right?
Yeah. Yes. True.
Excellent. Okay, so we have another question for Prof. Yoyo from Budewi. Where's the most interesting site to collect crustacean samples with high possibility to find a new species?
Go diving. Yes, the diving depth. It's rarely collect crab or hermit crab here, because when we go diving, usually we concentrate on coral and fish, but not in the invertebrate. And if you collect more here in Indonesia, for instance, for the hermit crab, there's some hermit who live in the coral. I mean, there's a hole in the coral that the hermit live inside, and there is no report of hermit crab live in the coral from Indonesia. While we know that this Indonesia is many places with a center of coral diversity, so it's impossible. There is no hermit crab live in the whole of coral in Indonesia. So go diving, then surely you will find a new species. I have one species new that Bu Intan collected for me from the coral reef that I still didn't work with it yet now. So if you go diving, you can collect a hermit crab or crab, then it is a possibility to find a new species. Or digging in the mud. Not many people like to dig in the mud because it's dirty, and then it's really very difficult to walk in the mud. And then when you dig there and then you sieve with the water, then surely you will find many species. That it's strange, and sometimes it's also new record or even new species. Yeah.
Yes. Let's go deep, right? That's amazing how most of the works have been in the intertidal that's already produced a lot of new species. And if you just go slightly deeper, and the case with Prof. Yoyo, the one that I didn't, I was doing something else, waiting for coral to be drilled, and there's this crab passing by. I just took them, and amazingly, Prof. Yoyo said it's a new species. That's just the potential of new species we just don't know yet, and that therefore, when you found your team found 27 species from the SJDIS, it sounds a lot, but it really that's the reality of new species to be found. It's just that just amazing new areas. Okay, I think there's a question previously in the previous slide, Bung Zen. Okay, from Prof. Wei Dong. He's one of the invited speakers who will be giving us his talk. So I will read his questions. My general question to the two professors. I understand that deep sea exploration is very challenging and needs strong regional international cooperations. In the UN Ocean Decade, can we imagine some frontier project to be planned as one way of following up action this important ISEIME? Oh, this is a great question. After ISEIME, what's next. Let's get together, and perhaps there's something we can design within the mission of UN Ocean Decades. I will give this question to both Prof. Oki and Prof. Rahayu. So I will give ladies first, perhaps Prof. Rahayu.
Yeah, that is maybe Pak Oki is more to answer this question because it's a more political or what?
Okay, we give it to the boss.
Thank you, Prof. Yoyo and Bu Intan. Thank you, Professor Wedong. I think before we merge into BRIN, I think, for example, LIPI and BPPT has several regional and international cooperation, including with China. So I believe this will be more and more in the future, especially BRIN. We have a global engagement program. We want to share the facility, the infrastructure, and share the funding for research, including research in the deep sea area. So we are very welcome you to continue our collaboration in the future. And regarding the Ocean Decade up to 2030, of course, we are in the research organization has responsibility to do the research on marine sciences to support the decision making by the stakeholder, especially to make use our resources in the ocean wisely. Thank you so much.
Okay, thank you, Prof. Oki. Are there any questions? I think there is one more questions in the slide, if I'm not mistaken. Okay, from Pak Karsono Wagyo to Prof. Oki, how prospect maso product active substance such as antimicrobe this difficult must use hyperbaric conditions to compare product active substance with no use of hyperbaric chamber. I'm a bit not following, but maybe Pak Oki can get the question.
Thank you. Of course, this is one of our challenge when we work with the true barophile from the deep sea that only grow at the elevated pressure. But again, there is also not non true barophile that are capable to grow with or without pressure as well. For example, the isolate I collected from Java Trends is temperature tolerance and also pressure tolerance. So they are capable to grow and adapt to our atmospheric pressure. But this is also another challenge for us, especially if we really have the true barophile. So we have to have a quite a complex fermentor with special pressure. But I'm not sure at the moment if we can isolate the gene cluster for producing substance without pressure. Our sample, our isolate now is being carried out to produce our identified product, and we don't use any pressure condition at the moment. So, Pak Karsono, we are still having our homework to see if we will be able to make use our true barophile that needs some hyperbaric condition that is less effective compared to the non substance or product without use of any hyperbaric chamber. So that's my answer at the moment. I don't have a very deep knowledge on obtaining the bioactive compound from the hyperbaric condition. Thank you.
Thank you, Prof.
Thank you, Prof. Oki, Prof. Rahayu. We do still have time for the Q&A session, but we can also open for direct questions if you like. Especially, we can do that for the people in the Zoom room. So if you have questions, you can just raise your hands, and I can let you ask question. We have two of our seniors here. So yes, if there's any question, just raise your hand. And also there's note.
Hello.
Hello. Oh yeah, Stephen.
I have a question to Professor Oki, because you mentioned this new research vessel, and I'm interested to know if it is also capable for gravity coring and multi-coring.
Again, capable of what?
For gravity coring and multi-coring, right? Using a multi-corer and gravity corers, to sample sediments.
Yeah, I think so. That because this is quite modern research vessel. After 20 years, we don't have any research vessel during 20 years. So, but I'm not sure the specific condition of the research vessel and the facilities, but I'm quite sure it comparable to the research vessel owned by China and Japan. So I hope they will meet the standard as you requested. Thank you.
Okay, thank you.
Yes, thank you, Prof. Steinke. I'm also a paleoclimatologist on the coral side, but yes, we do use gravity cores in our cruises here, even with the existing research vessels. Gravity cores, we do that frequently, just normally. I think we have multi-cores, but as like the one that Professor Rahayu used also, the upper multi-cores. I heard that we don't have is the piston core. We don't have that, but yeah, we have also. Well, the question is, once you drill, where do you put the sediments, right? That's already another question. But the another ministry, the Ministry SDM, actually has facility of like a big core room facility there, but also BRIN is building that facility in Karang Sambung. So Karang Sambung is an area that we develop for geologic samples, including having a large cold room. But we also have a smaller cold room facility in Ancol. So I mean, this kind of research we definitely can develop and think about the process afterwards.
Yeah, because at the moment you are talking about this research vessel Barunajaya, right?
Yes.
Okay, good. Thank you.
Yes. So all the Indonesian research vessels actually call for research is called Barunajaya. So Barunajaya 8, Barunajaya 3.
Okay, yeah. Okay, I got it. Yeah.
But the youngest one, I think Barunajaya 8 is already was built in 1998. So we need a new research vessels, as Prof. Oki mentioned. Yes, so in the chat room, yes, Ministry of SDM has cold storage in Cirebon. And I know many of our colleagues in LIPI also store their samples there.
I think I visited that once, but
you did? Oh, there you go.
Yeah, I visited the core repository in Indonesia, but I do not remember the name of the place.
Okay. Yes.
Long time ago. Yeah.
Yes, I think what we also need is the core scanner that can have more elements. There is, I think SDM has one, but I heard, typically we would need more elements to be measured before before subsampling the cores.
Yeah.
A lot to do, many things to do still. Okay, Pak Cahyo, please, Pak Cahyo, he's a geologist.
Many thanks, Bu Intan, as your opportunity. I would like to give edit information with Mr. Stephen question is about the sediment center or sediment core. Uh, I have joined it's not more about the 4 months ago, but my former institution is Marine Geology Institute. Yeah, we have Indonesian facility. We have like you mentioned, like course. Sediment coring, gravity core, piston core, and then multi-core. But the problem when we have when we research more than 30,000, 3,000 meters. I mean, there's a problem with the length of the the cable or the fan. So maybe in the future, I hope Mr. Professor Oki as chief research organization can propose these vessels, this equipment, because there's more important way to give to put the sample from the safe floor. Thank you, Ms. Intan.
Thank you, Pak Cahyo. Pak Cahyo is also very interested in the hydrothermal research, right? So he has a geology background, but we also have microbiologists who really also wants to go to hydrothermal, so therefore we can cooperate. But we also need chemists to understand the chemical property of the seawater around hydrothermal vent. Okay, are there any questions on new questions on YouTube, or are there any questions on the Zoom? We can we have time before we can go into break session. Silakan teman-teman. Okay, Arianti, Mbak Arianti, please you can.
Hello, Mbak Intan.
Hello,
Thank you for the opportunity. I'm sorry, I'm off cam because I'm still at home. Sorry. Thank you. I have a question for Professor Oki regarding the natural compounds produced by marine bacteria from deep sea. The way I understand it is that I'm doing marine natural products research as well. And I work mostly on organisms living in the tropical, in Indonesia or Australia, in the tropical weather, which means they live in a warm seawater. And because the competition is very high, as we know that in the tropical marine area they have a very diverse marine organisms, as we saw earlier in Professor Yoyo presentation. There's a space of competition of space and competition of food, and that's why they're producing these natural compounds as a way to survive, as a way to defend themselves from these predators. So deep sea is a new area that I am not familiar with. So I'm just curious, in the deep sea area, do you think there are many predators just like in the surface area? If there are these bacteria producing natural compounds, do you think these compounds used to defend themselves from the predators, or do they have other ecological surfaces, perhaps for maybe for mating, or maybe finding food, or maybe the way for them to communicate with their with other bacteria or other organisms? So I'm really curious what Professor Oki thinks about this. I think that's all my question. Thank you so much, Mbak Intan.
Thank you, Mbak Ari. Please, Pak Oki. That's a
Thank
very
you,
interesting
Mbak Ari.
question.
Of course, deep sea is very extreme, and competition for nutrient source, for example, is very fierce, and the source is very limited. For example, mostly come from the marine snow from the upper ocean into the down to the deeper parts. But some of the micro and macroorganisms in the deep sea also gaining their nutrient by eating others. So this is another mechanism. So on the other hand, some true barophile, for example, they produce lipid in their cell wall that 70% of its contain of DHA. Yes, the very good fatty acid that are needed for different purposes. For example, to maintain their flexibility, and also there are some potential enzyme that are produced by this micro and macroorganism in the deep sea environment. So, for example, Jamstack has successfully obtained new product of surfactant from the deep sea environment, and they found that their product is very powerful. So I think with the our research finding that at least our bacteria produce 8 different gene clusters similar to the terrestrial or shallow water organism. We are very optimistic that we will be able to collect more gene cluster from the deep sea that are very much different compared to the existing sequence in the NCBI. So that will be a good opportunity, because when we have the, for example, in my isolate, we have an RPS, non-ribosomal peptide synthase that also produced by many shallow water organisms such as sponges and micro from the low shallow water that are capable of producing antibiotic, antioxidant, anti-cancer, etc. So I'm quite sure that if we will able to collect the gene cluster, we can test them for different purposes. So I cannot really answer your question regarding how they will survive, but based on our result, there are 8 different gene clusters that are also found in the shallow water, but they have a unique sequences that will produce different product. Thank you.
Thank you, Prof. Oki. I hope that answers Mbak Ari. There's these things to discover, it seems like. Okay, we can have one last question if there is any questions before we go to the break. Are there any questions for our keynote speakers? Because after this, after the break, we'll go into the invited speaker session. Going once, going twice. Okay, and there are no new questions in the YouTube, correct? All right. If there are no more questions, again, we would like to thank Prof. Oki Karnarajasa and Prof. Dwi Listio Rahayu for coming here, for giving your insights, your presentations, and motivating us and opening new possibilities, not just in Indonesia with the young scientists, which is a huge reminder for all of us, but also the possibilities with our international colleagues here. Thank you again. Please give a big applause to Prof. Oki and Prof. Yoyo. Thank you so much. And ladies and gentlemen, we will be having a 15 minutes break and we'll be reconvening at 10 Western Indonesian time or 12 Eastern Indonesian time. So it's 15 minutes break. So we hope to see you again for our great 3 invited speakers afterwards. Thank you again.
Thank you.
See you soon. Bye-bye.
Bye-bye.
Thank you, Prof. Thank you.
Thank you.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Mengawali awal tahun 2021, Lembaga Ilmu Pengetahuan Indonesia melakukan 2 penelitian dengan kapal riset Barunajaya bersama Institute of Oceanology Chinese Academy of Sciences dan First Institution of Oceanography China. Berlayar mengarungi Laut Indonesia, penelitian ini dibagi menjadi 4 perjalanan. Barunajaya melaut dari Jakarta menuju Ambon, lalu dari Ambon menuju Bitung. Kapal riset LIPI ini kemudian melaut kembali menuju Banyuwangi dan kembali menuju Jakarta untuk melego jangkar. Penelitian pertama yang dilakukan dengan kapal riset ini adalah Circulation and Ecosystem Study in the Indonesian Seas under the influence of the Western Pacific Friability. Kerjasama ini dilakukan dengan Institute of Oceanology Chinese Academy of Sciences sejak tahun 2014. Tahun ini dilakukan perawatan mooring yang telah dipasang pada 2018, unduh data, dan pemasangan mooring baru di Selat Livamatola, Laut Halmahera, Maluku, Talaut, dan Sawu. Sedangkan penelitian kedua adalah Transport of Indonesian Seas, Upwelling and Mixing Physics atau TRIUMPH. Kerjasama ini dilakukan oleh Pusat Penelitian Laut Dalam berkolaborasi dengan First Institution of Oceanography China dan University of Maryland USA sejak 2018. Pada tahun ini dilakukan pengukuran sifat fisika air laut secara terus-menerus menggunakan berbagai peralatan yang ditempatkan di dasar laut maupun di kolom air. Data ini berguna untuk mengetahui kondisi air laut secara deret waktu dan dampaknya terhadap kondisi atmosfer. Selain itu, dilakukan pengambilan sampel air laut, plankton, dan mikrobiota lainnya pada kedalaman hingga lebih dari 3.000 meter. Para peneliti LIPI tidak akan lelah untuk terus berlayar dan berlabuh untuk ilmu pengetahuan. Penjelajahan kolaborasi ini diharapkan dapat bermanfaat bagi perkembangan ilmu kelautan di Indonesia dengan mengenal lebih dalam kekayaan lautnya.
Halo semua, kenalkan kami kelompok riset dinamika lingkungan laut dalam atau KRIS Dalam dari Pusat Riset Laut Dalam, Badan Riset dan Inovasi Nasional. Dalam video ini, kalian akan menyaksikan informasi singkat terkait keris dalam. Selamat menyaksikan dan kita akan berjumpa lagi di akhir video ini. Dilihat dari berbagai aspek kajian, Indonesia memiliki sumber daya kelautan yang sangat melimpah, baik di kolom air atau water column, di dasar lautan atau seabed, dan di bawah dasar lautan atau subseabed. Adapun yang harus dilakukan saat ini adalah memahami kondisi lingkungan di mana sumber daya kelautan ini berada. Kami adalah kelompok riset yang dirintis sejak tahun 2019 untuk melakukan kajian kondisi lingkungan, mengungkap dinamika yang terjadi, dan mengidentifikasi ancaman yang mengintai, baik di dasar lautan secara umum dan khususnya di kolom air dengan kedalaman lebih dari 200 meter. Potensi area kajian laut dalam sangat luas. Indonesia adalah negara dengan luas lautan 6,4 juta kilometer persegi, di mana 32% adalah lautan dengan kedalaman kurang dari 200 meter, 25% memiliki kedalaman antara 200 sampai 2000 meter, dan 43% memiliki kedalaman lebih dari 2000 meter. Riset-riset yang sudah dan sedang dilakukan adalah riset karakteristik dan dinamika fisis laut dalam, riset nutrien dan karbon laut, dan riset pencemaran mikroplastik. Sedangkan topik riset ke depannya adalah riset biogeokimia laut dalam, riset pencemaran di laut dalam, dan riset hubungan lingkungan laut dengan kondisi makhluk hidup di laut dalam. Melakukan eksplorasi laut dalam tidak diragukan lagi sebagai salah satu kegiatan yang menantang dan frontier di Indonesia. Hal ini juga mendukung salah satu visi kemaritiman 2045 di mana menjadikan Indonesia sebagai poros maritim dunia. Melalui kelompok riset ini, diharapkan dapat memberi kontribusi dengan mengungkap teka-teki terkait karakteristik dan dinamika laut dalam, serta bagaimana kondisi tersebut mendukung kehidupan makhluk hidup yang belum pernah terungkap sebelumnya. Selain itu, peran laut dalam mengatur iklim global juga menjadi fokus di dalam keris dalam. Menuju lebih dalam adalah semangat kami, dan kami telah mencoba sejak tahun 2018 untuk mengembangkan diri, menggeser batas-batas, dan mencari kebaharuan melalui kegiatan ekspedisi ilmiah di perairan Indonesia. Banyak permasalahan dan topik penelitian laut dalam yang perlu dikerjakan saat ini. Ratusan fitur dasar laut yang perlu diungkap, jutaan persegi luas lautan yang perlu dijelajahi, dan tak terhitung jumlah tantangan yang perlu diselesaikan. Kelompok riset kami saat ini terdiri dari 4 orang peneliti dan 7 orang teknisi. Dan jumlah ini masih jauh dari kondisi ideal untuk mewujudkan ide dan mimpi kami untuk riset laut dalam di Indonesia. Kami adalah jiwa-jiwa muda yang memiliki ide dan tujuan besar dalam riset laut dalam di Indonesia. Namun kami belum memiliki Anda, yang merupakan talenta-talenta berpengalaman dalam bidang ini. Mari bergabung di keris dalam dan kita wujudkan ide dan mimpi kita untuk riset laut dalam di Indonesia.
Welcome back. Selamat datang to ISEIME 2022. Thank you for joining us. And in the morning session, we just had our keynote speakers. And now we have more speakers that will be sharing with us their knowledge and wisdom. We have 3 invited speakers. The first one is Prof. Stefan Steinke from Xiamen University, followed by Professor Wei Dong Yu from Sun Yat-sen University, and followed by Professor Madya Dr. Tuan Nurul Sabika Tuan Anwar from University of Malaysia, Terengganu. So keep tuning into the ISEIME and hear about their great talks. So for the first invited speakers, I would like to invite Professor Stefan Steinke. He is a professor at the Departments of Geological Oceanography. Professor Steinke is a deep paleoceanographer. He used trace metals, trace elements, and isotopic compositions of planktonic foraminifera. His research interests include tropical paleoceanography, paleoclimatology, the reconstructions of water mass variations in the Indo-Pacific region, and understanding climate and oceanic related processes controlling sediment supply across ocean margins. He received his diploma as well as his PhD from Kiel University, and he has written so many publications that are also very highly cited. So we are so happy to have him with us. Hello, Professor Steinke.
Yeah, hello. Yeah, thank you very much for this nice introduction. Yeah, it's a great pleasure and honor to get invited to present our climate reconstructions, right, in the maritime continent. Yeah, so what to do now? I have to share my screen, right?
Yes, you may share your screen. And like the plenary sessions, like the keynote speakers, if you have questions, if all the listeners have questions, you can put it in the chat room and we can compile it afterwards. Thank you. The time is yours, Professor Steinke.
Okay, can you see my?
Yes, I can see your slides, but it's need to be in the presentation mode.
Yes.
Perfect. Thank you.
Are we ready?
Yes, we are.
Yeah. Thank you very much for the invitation and for the nice introduction. Yeah, as you just said, so I'm also would call me a micropaleontologist because, as you mentioned, right, and as you will see in the following 30 minutes, right. So we are using foraminifera as our signal carriers for the environmental history of the ocean. And yeah, my main interest is about the monsoon. So we worked on the Australian-Indonesian monsoon, and at the moment I'm working on the South Asian or Indian monsoon during the Miocene. What I'm doing today here is a kind of synthesis what we have done over the let's say 15. I would not say 20 years, but over the last 15 years, right? And this is about the late Quaternary environmental history of Western and Southern Indonesia. It's not about Eastern Indonesia because we don't have sedimentary archives for that area, for these things, So this is, all what I'm presenting here. Are contribution by many people. You will see their names later, but I in particular would like to mention here Dr. Maya Mortadi from Bremen University, who was chief scientist of many cruises to Indonesian waters, and he was also proponents of many proposals, supervised many students, right? And that's the reason I would like to mention him here, as one of the, let's say, one of the most active scientists from Bremen University, right, doing paleoclimate research in Indonesian waters here. Why we are interested in the climate. Let's talk about the maritime continent. We have seen that already. If you're seeing already on this slide here, right, all the different influences on the climate. Sorry. Here. Yeah, on the influences on the climate on the maritime continent, right? That is coming from seasonal changes, right? As you can nicely see here, the monsoon, right? So interannual variability, right? The influence of the Indian Ocean dipole, right? The El Niño Southern oscillation, yeah. And so here, El Niño, La Niña conditions, and of course, Indo-Pacific warm pool dynamics, right? That are really important for deep atmospheric convection, by strong precipitation, and this in general, right? We are talking here mainly about precipitation changes, but of course also changes in the structure of the upper water column. For example, upwelling, right? As you can see. And on the other hand, why is this area so important? It's because of the Indonesian through flow, yeah. Very complex current system, and honestly, I do not really understand that system, right? So it's so complex, and yeah. But what is important is, right, it's connecting the Western Pacific with the Indian Ocean, yeah. And part of this water is supposed to go also in the Atlantic Ocean. So it's part of the thermohaline circulation here, right? And as I said in the beginning, right, that makes this area so interesting, but also very complex to understand, yeah, because this interaction between seasonal and interannual variations, yeah. So it's a great challenge to do that. So what is as a paleoceanographer, as a paleoclimatologist, yeah, what is our general interest, right? It's to understand Earth's past climate system, yeah, and its natural driving factors, yeah. As you know, right now climate is strongly influenced by us humans, right? And therefore it's definitely important what is natural variability, yeah, and what is the human influence, right? And that is definitely needs for future projections here, right? That is actually what is driving us, yeah. You see here, this is the map showing different sites here, the dots, the start of various research cruises to that area, right? There was a cruise in 1999 with the German research vessel Sonne. Yeah, we also had a cruise in 2005. I was able to participate in this cruise, and in 2006 here. Yeah, and as you see. Therefore, my question about sediment sampling, yeah, all is based here on sediment cores, right? So based on gravity coring, piston coring, but also multi-coring here, right? And you can see already how many people contributed to the research that I will present in the next minute, right? From Bremen University, but also from the US, yeah. And also we had also Risa, he was a PhD student at Bremen University, yeah. All these people contributed to this study here. As I mentioned already in the beginning, what really matters is rainfall, yeah. So when we are mainly talking about the monsoon, IOD, and so we are talking about variability in rainfall. that is which really matters and which is really important here. You know. Oops. So therefore, one of this reconstruction for past variations in rainfall are changes in the seawater data 18.0 records. Yeah, so that is a tool to infer past rainfall changes in that area here. Yeah, so what we are doing here, we are measuring stable oxygen isotopes together with magnesium, calcium temperatures to get an estimate of the seawater data 18.0 that is a proxy for changes in salinity and also in runoff or precipitation over the ocean here. As you can see here, we are covering here the last 50,000 years, yeah, from the different basins here, right? Simulo Basin, Mentawai Basin, also close to the Sunda Strait, Sabu Basin here, right? And one of our most important findings is that data of seawater, and later I will also show you element ratio, titanium, calcium of bulk sediments offshore Sumatra and Java and Sumba, that we do not see any glacial interglacial variability. Yeah, so we have here no significant change in the rainfall related runoff on this time scales, as you can see here, right? When you look on the Holocene, right, and the signal periods here during the last glaciation or Mentawai Basin, much higher resolution also from this basin here, right? But what we found is that there is strong evidence, right, that during the melting of large ice sheets and the slowdown of the ocean thermohaline circulation in the North Atlantic, so this so-called Heinrich events that you can see, right, that resulted in warmer and drier conditions, yeah, as you can see here, drier conditions during this period, yeah. And we interpreted that, right, that a weaker or disrupted thermohaline circulation results in a reduced northward heat transport by the surface ocean in the northern hemisphere. So the atmosphere compensates for the reduced ocean heat transport by transferring warm air from the warmer southern hemisphere to the cooler northern hemisphere during this period, during the Heinrich stadials here. Yeah. So, and this is also accompanied by a southward displacement of the intertropical convergence zone here. Yeah. What was the result of that, right? Of the more southerly position of the intertropical convergence zone, the ITCZ, general drying of the northern hemisphere, you know, and the equatorial ocean, including the maritime continent and wetter conditions in the southern hemisphere over Australia here, right? You see the references, yeah, and people who contributed to this study here, right? Oops. Yeah. What we also used, right, to monitor changes in past rainfall is the ratio between titanium, yeah, as a terrestrial end member, and calcium as a marine end member in bulk sediment deposits, indicating qualitative changes in the rainfall-related runoff, right? So the idea is if we have higher precipitation over land, we have a higher runoff of terrestrial material here, you know. And on the other hand, what was also used here are compound-specific isotope signatures of hydrogen and carbon in leaf baxes of higher plants. Same thing, right? So that this leaf baxes, right, this organic material, yeah, of this higher plants is transported by the rain, rivers to the oceans here, right? So how we can use that, right? Also, this carbon isotopes here is that this in the tropical regions, the C4 photosynthetic pathway, of course, in warm season grasses and sedges, while the C3 pathway is utilized by trees and shrubs. You know, so what simply means, yeah, that higher carbon data set in sea values can be used to detect periods with a higher contribution of C4 plants, reflecting open canopy vegetation, while periods of lower values indicate more C3 plants and a close canopy vegetation, which tells us much about the rainfall over land, right? When we're looking on this leaf waxes from Nias Basin and the Java basins, right, show more heterogeneous pattern across Indonesia than the sediment element composition and the data element of seawater. As you can see here, this is this titanium calcium ratios, right? In this direction, right? This would indicate more wetter conditions here. Yeah. Again, what we do see here, right, on this leaf waxes delta values are again no interglacial shifts, right? That confirms our data of seawater records from the Simule Basin and the northern Mentawai Basin, right? So what this together, right, our stable delta ANO seawater reconstruction, titanium calcium and leaf waxes, those data suggest that the glacial climate of West Sumatra at and north of the equator was as wet as, if not wetter, than today. What was kind of surprisingly, right? Only during this so-called Heinrich events, right, we had drier conditions here. Yeah. Offshore Java, the delta D values, yeah, here in green, are lower and imply better conditions in the montane region during the last glacial period compared with the present interglacial, right? And those records, right, are in contrast, right, for stalagmite records, right? Cave records, right? From those stalagmites from Borneo, Sumatra, Florence, who clearly show a glacial, interglacial variability, right? That might be related that they are recording here not only the amount of precipitation, but also changes in the end member or the source of our moisture, you know, where it was coming from, you know. When we're looking also on our sea surface temperature, that is also another very important target parameter for us, as sea surface temperatures provides gives us information about changes in latent heat release to the atmosphere and strongly influences also the Walker cell and the Australian Indonesian monsoon, right? And so changes in temperature, right, can strongly affect Walker and the monsoon system here. Yeah. So here we are using two tools here: planktonic formula for magnesium calcium pilot temperature thermometry and alkanone pilot temperature. Yeah, we can also use, right? That is extracted by this long chain ketones are extracted by cocolytic fluids, yeah, in algae. Yeah. What we different to our rainfall reconstruction, we clearly see changes on glacial, interglacial timescales. And interestingly, right, they vary akin to Antarctic temperatures here. Different what we have seen, what we have seen when looking on the rainfall reconstructions here. You know. So we interpret that changes in the mean annual SST in the eastern tropical Indian Ocean, yeah, to changing global ice volume and atmospheric carbon dioxide on glacial interglacial tension as a reason for those changes here, right? I would like to give you another example on high frequency on Holocene high frequency climate variability. We were looking on a high resolution sediment record here north of Sumba Island here. What we have done again here, we used mixed layer and thermocline dwelling planktic foraminifera, yeah, which combined can be used to get an estimate of the upper ocean structure, so depths of the thermocline, if deep or shallow, which gives us information about the upwelling here. And what we definitely found is that during the Little Ice Age here, we have an increase in upwelling, which we relate to changes to ENSO, right? So that during the Little Ice Age, the Western Pacific, right, was more dominated by El Nino here. That is, confirmed here. That is compared here to records from Conroy and Kopp et al. Right, and that is also what we nicely see when looking on modern chlorophyll maps. Right, here during the southeast monsoon season, January, August, September. Right, we have this high productivity here, because of this upwelling of Java and Sumba here. During an El Nino event, it is even stronger, the upwelling here. And when looking on La Nina events here, you see almost no upwelling during La Nina events here. So what we found is that the prevalence of an El Nino-like mean state during the Little Ice Age and the La Nina-like mean state during this medieval warm period, and the Roman warm period. That is what we found as an example for Holocene high frequency climate variations. I would also talk a little bit now about what we know, but what are the knowledge gaps and direction for potential future research here. What we are definitely lacking is continuous sampling on land and in the oceans. So satellites, meteorological station, and buoys have delivered important data during the past decades, but to extend the instrumental data back in time and improve the climate memory for future predictions, in such a complex setting and diverse setting as presented before, it is important to understand how the environment is being recorded by the biosphere and geosphere. To my knowledge, there are presently no efforts in sampling proxy carriers for temperature and productivity, such as marine plankton, foraminifer algae, cucullitophorids and benthos, corals, bivalves, and measuring their chemical composition on weekly to annual basis. I might be wrong, right? There might be systematic efforts, but to my knowledge, there is no systematic effort in existing monitoring water isotopes of rainfall in terrestrial plants, stalagmites, and riverine discharge to understand hydrologic cycle, and its in a fingerprint on proxy carriers. That is also one topic, right, that we would like to improve our proxies, right, to get a more detailed and better understanding. And this modern, let's call it ground truthing of those proxies is critical, to monitor changes related to seasonal, monsoon, ITZ, or interannual ENSO and IOD in the past. What we also definitely lacking are long records that cover several interglacial cycles. What is available now from the maritime continent, right? There's only one stalagmite record from Borneo and 2 marine records offshore the Sunda Strait. But as we have seen when looking on our proxy records, it's not sufficient to look only on one side, because it's the variability of climate is very complex and different from the different places in Indonesia. That is seen. And what is ideally needed would be a record, at least back to marine stage 11 or the Pliocene, to compare paleo data and future projections. even nicer if we can even go back to the Miocene, 15 million years before present. But it seems if we continue to warm up our Earth, right? So our planet, right? So the modern analog would be the middle Miocene, you know, climate optimum, you know, so 50 million years ago before present, right? Yeah, I would like to thank also in the name of Maya Mortadi and all other researchers who contributed to our studies, right? To the LIPI and the MIG in Bandung, the BPT, Agency for Development of Technology, and the Indian Ministry of Research and Technology for the great support in preparing and conducting the research expedition. I also would like to thank the Federal Ministry of Education and Research, BMBF, and the German Science Foundation for funding our research. To end my presentation, I would like to talk a little bit about the research and education program SPICE, yeah, Science for the Protection of Indonesian Coastal Ecosystems, yeah, in short, SPICE, because all these studies have been done under the umbrella of this, in my opinion, very successful cooperation program, yeah, that started in 2003 and ended in 2015, right? When going back in time, right, so that the cooperation between Germany and Indonesia dates back to 1979, yeah. So during this period until the late 90s, we had their research cooperation that had been mostly in the field of marine geosciences, conducted on the cruises of the German research vessel Sonne in Indonesian seas, right? And we had this individual contact and research opportunities, yeah, through the German exchange service, right? And the SPICE program has its origin in the new contact established between Indonesian and German scientists and science activists in the late 1990s, right? This is a kind of synthesis of this research and education program. I would like to emphasize that this was not only on research, education was always a very important part of this program, right? So that was just published, this book here, edited by Tim Jenajan, Tim Brixen, Irianto, and Samiari. And not to forget, right, one of this important scientists behind this is Professor Ittekott, Hamburg, later from the ZNT in Bremen. Here in this book, you can find a lot of information about coral ecology, etc., etc. Yeah. With that, I would like to end my presentation. And again, thank you for listening. And again, thank you for the invitation to give you an idea what we have done over the last years in using sedimentary archive from Indonesian waters. Thank you.
Thank you, Danke, Professor Steinke. Thank you so much for your talks. We have geologists also here, paleoclimatologists. So there's been some question that we will keep for the question and answer session. So please keep those questions coming, guys. We'll be reading those questions. Thank you, Professor Steinke, for your presentation on what have been done in the Indonesian maritime region, not only for science, but also for research cooperations that with capacity building aspect. And I just want to inform you that now recently we have been investing more on research infrastructures. So now we have a new IRMS, isotope ratio mass spec. It's not the kill device, but it's the one that we purchase it so that we can do carbonate as well as organic measurements, as well as water samples. But as you know, this is not an easy equipment. It takes a lot of
I know.
experience.
Yeah.
I was told that don't get an IRMS before you get your tenure. Someone said that. So I think there's, even with this new equipment in Indonesia, there will be a lot more capacity building that we can collaborate to make the most use of this equipment, and also collaborations with Xiamen University. Just something.
Yeah, that would be really great. That, actually, again, right, coming back again about the coring, right, and the coring facilities and things like that, right? Because I was mainly talking about Western and Central Indonesia, right? But Eastern Indonesia, right? That would be of great interest, right? So in particular for us, right? In particular, when talking about the ITF, right? In particular, the inflow passages and things like that, right? Unfortunately,
Yes. We hope you'll be staying until the Q&A session, right? We'll be
of course.
Okay. Thank you so much. Yeah. There's
thank you.
New ideas. Thank you again, Professor Steinke. So next, we will be inviting Professor Wei Dong Yu. He's from Sun Yat-sen University. Prof. Wei Dong will be giving a presentations on monsoon and connecting the Western Indonesia and the North-South Hemisphere. Let me share his slide.
How can I, sorry, I'm not so familiar with Zoom. How can I stop?
Fine. Oh, you can stop maybe once I share.
Stop. Yeah, okay. Yeah. Did you stop?
Yes, I stopped, then it just
thank you. Good. I just found the button. Yeah, nice.
Oh, sorry. Let me introduce Professor Wei Dong first. Hold on. Professor Wei Dong Yu is a professor at the School of Atmospheric Sciences at Sun Yat-sen University. Professor Yu works, he's a physical oceanographer focusing on ocean-atmosphere interactions at various temporal spatial scales. He received his PhD at Chinese Academy of Sciences, his master's degree from the First Institute of Oceanography, and his bachelor from Ocean University of Qingdao. He has written so many publications, many of his publications related to the Indo-Pacific region. And I know he's been also very active in international working groups, such as in the IOC WESPAC and the Cliver Goes Indian Ocean panel. So with that, we will be giving the floor to Prof. Wei Dong Yu. Hello, Professor Yu. Hello, can you hear us? Professor Yu, are you having a technical? Oh, you're still mute. And your video is also still off. Okay, now you're unmute. But we cannot see you yet. Okay, we can see your slide. But can we test your voice? We can't hear you yet. If you are having trouble, Professor Yu, you can also write on the chat room. Can we hear you? That's my first question. We can't hear you. Professor Yu, you can hear me, but okay, but we cannot hear you. We can see your presentation. You can, yes, we cannot hear you. or at least I cannot hear you, or maybe other people here. So perhaps there's an issue with the speaker setting of your Zoom. Okay. So is there any suggestion from the technical team? Yes, we cannot hear you. Oh, there's something. No, not yet. I hear something, but I don't know who just talked. Can you, we still cannot hear from your side. Let's see. What can we do?
Oh.
No. Test. I think so. The issue is not his presentation. I mean, this is Professor Yu sharing from your computer, right? But we cannot. Okay, I retreat and come back. Okay, take your time. Okay, let's just be patient and wait for him to settle things first. In the meantime, perhaps if we have Professor Sabika, we can also maybe test that things are also okay on your end.
Hello, everyone. Do you hear me?
Oh, okay, we can hear you well. Hello. Okay, so we won't be.
So you guys want me to start first ahead of Professor Wu?
Let Professor Yu. Let's wait for him to. He said he want to leave and come back. If he's still having issue with that, that would be a really nice options. Maybe in the background we can tackle that with the technical teams on another Zoom room.
Okay. Sure.
Professor Yu, are you here? Can you talk to us on the chat room? Okay, maybe his connection. All right, let's just wait about 2-3 minutes before we decide. Okay, I don't see him coming back yet. Okay. Let's see. Let me check on him. Okay, he's coming back. Hello, Professor Yu. You're back. Can we hear you now? Can you keep saying something? But we still can't hear anything from you. I can't hear you. I can see your mic is already unmute, but I can't hear anything. Okay, let's see. Okay, maybe while Professor Yu is settling, so we also don't want to rush him. I know it could be very panicky if things not working on the technical side. Professor Yu, we will give you time to settle with the technical issue from the organizing teams, Pak Fadli or Pak Ferdi. Please coordinate with him, making sure that his Zoom is okay. But we will switch the order for now. We will welcome Professor Madya, Dr. Tuan Nurul Sabika Tuan Anwar. Thank you for joining us.
Thank you.
How are you?
Not so bad. How's everything in Indonesia?
Good. I think this is the first time we actually meet, but I hear you might be coming to Indonesia, so we can meet in person.
Again for this Saturday, I'm going to UI.
Ah, yes. Okay. Great.
So I'm sharing my screen right now.
Let me introduce you first properly.
No problem.
So Professor Madya, Dr. Tuan Nurul Sabika Tuan Anwar, she is Associate Professor at the Faculty of Science and Marine Environment at the University of Malaysia, Terengganu. Professor Sabika Tuan Anwar is a chemist specializing in the field of oleochemistry, analytical environmental chemistry, physicals, chemical methods, and also microplastic. So she is currently an advisor to one of our colleagues at Research Center for Deep Sea. She received her PhD from the University of Alberta in Canada, a master's degree from UKM, and bachelor degree from University of Science and Technology Malaysia. She has published many publications and she is very active, has many active research activity currently with 8 active programs going on. So we thank you again for joining us and enlighten us on the aspects of marine pollutions that perhaps we could also develop and more into the deep sea area of Indonesia. With that, I'll give the floor to you, please.
Okay, thank you for Miss Intan for a very good introduction of me. And okay, I just sharing my screen. Okay. Okay.
Hello, I just came back.
Oh, okay. We have Professor Yu here.
Oh.
Okay, I waited for a while. It's go ahead. I
Okay.
will talk after you.
Okay, thank you so much. But we're happy you're back. We can hear your voice.
Okay.
Thank
Yeah,
you.
sorry for the problem.
No worries.
Okay, I'm sharing my screen.
Yes, I can see your screen and I just need to be, oh yeah, perfect. Thank you.
Okay. So good morning, everyone. Selamat siang, selamat pagi. And my topic today will be on the chemical characterization of microplastic from a few environmental matrices, how this technique actually, this kind of techniques is important aspect in monitoring and risk assessment, especially in marine pollution monitoring. So this is our campus. Just to let everyone know, we are located very close to the sea. You can see the South China Sea at our background. So very, 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 very nice campus. You can hear the waves here, and everybody is welcome to UMT. And our niche area is actually aquatic and marine research. So we have a few research stations at Bidong Islands. Is one island that dedicated for the research in is given by the state of the state government of Terengganu to our university. So we can do all the marine research base here. And we also have a research station at Kenyir Lake, one of the biggest lake in Southeast Asia, I think. It's a man-made lake that being used for dam. And also we have a research station located at Stew Wetlands. This wetland has 11 interconnected ecosystem from the peat swamp, from the estuaries, from the ocean, and so on. We also have a very dedicated research station dedicated for turtle sanctuary. It is located at the east side of Redang Island. We call it a Chaga Hutang Research Station, UMT. At UMT, we also have a small research vessel, not as big as LIPI ones, okay? But we try to do our best to make to utilize it. Okay, we have a RV Discovery UMT that currently being used for monitoring offshore waters around Peninsula of Malaysia. And so because of this, I welcome everyone to have a collaboration with us at our research group, our faculty, or any research group in University of Malaysia, Terengganu. Okay, so moving forward, we are back to our topic of interest, microplastic. So as we know that we are living in a plastic age or plastic era. Okay, everything that we are using now consists of plastic. If not much, we have some parts of things that we use nowadays is coming from plastics. Even in our consumer products, we have a microbeads that made from microresins that's currently made from polyethylene or polypropylene. The synthetic one is a plastics. So we also have a bigger plastic that being used in automotive as our things that we use in house, toys, and so on. So actually, I'm not trying to say that plastic is not good, because it's a good thing. It's handy. It's a very cheap. Mostly very readily available, versatile, durable, lightweight, and in some area we cannot eliminate of using plastics, especially, for example, in medicine. So everything has been to be in plastic because we need to sterilize and so on. So that's why I'm not saying that plastic is not good, but the thing is how we manage the byproduct of the plastic, or how we manage the waste of the plastic, how we dispose the plastic properly. And that's come to the term that everybody needs to have some sort of integrity in managing this plastic. Okay, because after that it can become the most harmful material to our planets, causing a very huge global pollution. And this is one of the, I think I'm very interested in this front page of National Geographic magazine. So this is the thing with the plastic situation nowadays. So how dire is the situation? We see at the surface is not there, but it's actually there. Because once these plastics go into the environment, it becomes smaller and smaller because it's fragmented into a smaller size, become a microplastics, become a nanoplastic, and so on. And the thing is, with the Southeast Asia region, is become a very hotspot region for the plastic pollution in the world. Maybe because of the current or the sea surface current, or maybe because of we are among the most abundant consumer that using the plastics, or maybe because of this region is produced as a plastic producer, is one of the region that produce most plastic as a plastic producer. So in a recent, I think it's quite recent data from Professor Major, they published that more than there's a lot of our river is being in rank in the top polluted rivers in global. Okay, and this is a data from Professor Jambek, like 10 years ago, but it's not really changed. The trend is the same. This region is still is a hotspot for microplastic and plastic production. So this is a few years ago when they found a very lots of plastics in a very big whales that end up at the shores of Philippines. So almost 40 kilos of plastics has been found in this whale's stomach. We don't know that if this whale is dying died because of the plastic or not, but we found that. So from plastic into microplastic, when it's degrade, okay, the smaller size of microplastic or the smaller size the plastics became, the bigger is the problem. So this is one of the examples of plastics that we found in our water samples that we are isolating right now in our microplastic research institute lab, MRIG lab. So you can see from 1 liter of water, we can found up until 10,000 of microplastics. So it's a very loss, it's a loss, okay? So from the microplastic, so we know that the smaller the size of any substance, the bigger the surface area of that. So from the macro level to the micro or nano level, we increase the numbers, of course, we increase the bioavailability, we of course increase the target organism, toxicity, detection difficulty, and cleanup difficulty because of the size is smaller. And that's why I say that small microplastic, smaller size is bigger the problem. So as introduction, the microplastic can be divided into 2 categories. One is a primary and one is secondary. Most of you guys maybe know about this already, but I just tell you guys again. Primary microplastic is a plastic that has been produced in a micro size during their production or manufacturing. For example, all the microbeads or resins that we using in our toothpaste, in our shower gels, and so on. So they are being made into a very small micro size of beads or resin. Meanwhile, the secondary microplastic is coming from the degradation of a larger plastic. So when we throw away our waste or plastic waste or the plastic bags, the bottles, the drinking bottles, when when these kind of plastics being in the environment, there is a few process, a chemical or mechanical process that can degrade this plastic. For example, photooxidation, and also mechanical abrasion. Because and when this plastic enter our river system and going into the marine area, the degradation become more faster because of you have a wave, you have a surface current, and so on that speed up all the degradation and fermentation. So this microplastic, the secondary microplastic, this degrade ununiformly because the primary microplastic, when they manufacture it, is in it the size and the shape is in uniform. Okay, they are the similar size, similar shape, and so on. It's easier to be tackled in the environment compared to the secondary one, because the secondary microplastic, when they degrade, is degrade in different shapes, different size, and different reaction rates. And this secondary microplastic actually made up more up to 80% compared to primary microplastic in the environment. So this secondary microplastic is actually more harmful compared to primary microplastic. That's my personal take. Because plastic, actually, when they are making the plastic, the bigger plastic at the production, plastic supposed, or polymer supposed to be inert. It means that it has, it's not supposed to react with everything. It's supposed to be inert. But when it comes to into the environment, it degrades and it has been photooxidized. All the functional group becomes open, and there's some radical process that speed up the reaction and so on. So this plastic can leach out or absorb other pollutants. It's become a vector for other co-pollutants, such as heavy metal, or it actually can release all the material or metals that we made, that been used to make the plastic into the environment. So it's become absorbed and also leaching. So all these, okay, for example, co-pollutants that can be absorbed into the plastic is or persistent organic pollutants, such as DDT or organocides, okay, and also heavy metal as well. All these can bioaccumulate in our environmental system. So this is a data from WHO. They say that an average, WWF, an average person potentially consume as much as more than 1,000 plastic particles every week from just a water source, not from the seafood or not from the food that we take, from the water. So imagine that we actually consume more plastic than other things. So the toxic effect of microplastic has been documented in many research, in phytoplanktons, in zooplanktons, especially in worms, mussels, oyster, all the bivalves, plus the toxic effect of chemical associated with microplastic. This is a very important thing when we talk about microplastic. So on polymer, we actually want to know how harmful these pollutants to us and to the ecosystem. So in one of our preliminary data, like a few years back, and we're going to publish one or two more in this years forward. This is our first data that we detect the microplastic in living human colectomy specimens. So we found that this is a specimen. We found that there's a lot of microplastic can be retrieved or extract from human guts actually. So this microplastic, when we compare with the environmental microplastic that we found in our lab, it has a similar trend. So we assume that it can be coming from the similar source, meaning that our food. So, but there's a lot of gaps. There's a lot of question that when we're dealing with the microplastic research, first we need to actually know the occurrence, the distribution, the abundance of microplastic. So the consistent monitoring program is needed. So that after that we can relate it with the ecotoxicology or toxicokinetics of the polymers, and we can calculate the risk assessment from the whatever data that we have before we can actually made it into a guideline or into what we call it index. Okay, just like a water quality index. So we don't know yet, because a microplastic is actually a physical contaminant. Is quietly different to tackle microplastic issue compared to organic contaminants such as EDC compounds. So when physical contaminants is a heterogeneous contaminants, meaning that is the amount in estuary might not the same as the amount in marine water or open space. So it's kind of like more research need to be done, more monitoring need to be done to establish a very good baseline data before we can come out with a finding that related to the rules and regulation of microplastic in water system, for example. So that's why I say that the real challenge in microplastic analysis is the method for the standardization method and harmonization. I was a part, is currently is a part of IOC WESPEC, a working group that working for microplastic and marine debris analysis in the Southeast Asia region. Up until now, we still struggling, or we still trying to have a one method, one standard method that can have been harmonized for being used for every part of the world, but it's very hard because of the different capabilities, different capacities, and different different demography or geography area. So this is I just go through very fast of this. This is a few way of we doing surface water collection. I know there is few more. We can use a ROSET or CTD for open sea, but this is for surface water that we've been using now. So this is what we are using by towing. We try to standardize our method by towing in referring to UNEP and IOC WESPEC guidelines, but at some place that cannot be towing. For example, this is our research in Antarctica, that we have to do a pail bucket collection compared to the towing, because in Antarctica our limitation is we don't have a very big boat. We just using we just walk around the island, the Antarctica islands. And we collect the sample using the mesh that custom for the microplastic. It's the same mesh that we custom, just a different way. But we try to correlate the amount of water sample bucket that we collect, that will be equal to whatever volume that we collect with a towing net. So this is a sediment. I think everyone that working with microplastic in sediment know how to do this. Corey maybe know. We try to differentiate a sediment based on the water age, middle age, strand line, or bed shore. So because of we try to see if there's a different trends of microplastics coming from inland or coming from the sea. And then this is how we collect the microplastic from the biota. Is a self collection. It's either in the oysters or in the green mussel, Penaviridis, or in the butt clam. So it's different way, but all these samples need to be treated or before it can be extracted and undergo a very extensive QC and QA blank contamination control. So after you get a sample, microplastics, for the analysis of microplastic, we divide the analysis into two parts. First is physical parts, and another one is a chemical parts. I strongly encourage everyone not to skip the physical analysis of microplastic, because later on we still need this kind of physical data to rationalize all the chemical data that we obtain. So the physical analysis, such as a visual techniques using microscopic compound dissecting or Azio camera, and also scanning electron microscope is very important to see the morphology, to see the colors, to see the shapes, and to see the size of microplastic itself. So and after that, we after all the sample we subject to the chemical and analysis, is either by spectroscopy or chromatography, depends on whatever that we have in the lab. So of course, physical analysis is very easy to be done or to be carried out, especially when you have a large microplastics. Okay, so the microplastic that usually we divided microplastic into three. 1 to 5 mm, and then below than 1 mm into 500 micrometers, and below than 500 micrometers. So, or depends on whatever ratio that you've been using. So this is how we use our analysis in our lab. So we use a AzioChem to measure each of microplastic particles, the size of each microplastic particles, and spot the color, the shape, and so on. And after that, all the microplastic we re-assign again, assess again under scanning electron microscope to see the morphology, whether it's cracking, has a hole, has a pit, and so on, or maybe a biofilm attached to the microplastic and so on. Because this SEM is quite important because it tells you the story of aging of the microplastic. So it will tell you actually this microplastic is actually already in the environment for quite some times. And after that, for chemical analysis, of course, we are using either either a benchtop FTIR, especially for a very large microplastic. And if you have a small microplastic that cannot be picked up or cannot be analyzed with a FTIR, you can use a micro FTIR or micro Raman that can give you all the data about the polymer composition or polymer types. And or you can use a pyrolysis GCMS if you want to see a polymer concentration or any additive that related to the polymer or microplastic that we are looking for. So this is a few examples from our lab, how we determine the color, the shapes, the size of the microplastic. You have a very range of colors and a very range of shapes. Maybe it's a pellet, fragment, fibers, foam, and so on, and a variety of shapes of variety of size of microplastic that is being detailed in our measurement. Especially if you're working with microplastic in biota or microplastic in human, the size of microplastic is very important to see if it actually can penetrate our guts and all the vital guts as well. So this is an example of microplastic pictures or a photo from microscope. We have a few different types of microplastics from sediment and water, and also this is from biota. You have a few types of microplastic as well. From biota, I think this is from fish, Lattice calcarifer, the sea base, and also this is from human. Sample from human. We have most of the sample that found in human guts in fiber form. So it's when there is a few study come out after our study, come out after we publish our study, and the other study also in line with our findings before that they also found most microplastics in the shape of fiber in human guts, and as also they found it in human blood as well right now. So this is actually the microplastic that hasn't been sorted. Okay, you see this is from water sample. So it's a lot. It's thousands of microplastic that need to be sorted and so on. So it gives you story, and after that, you can determine whether this plastic is actually a polymer or not. So this is a SEM EDX. Okay, SEM scanning electron microscope, when it's alone, it's standalone, it's actually a physical analysis. But when we couple it with the EDS, okay, it give you information about the carbon or the element in the microplastic, in the polymers. So for example, this shows that the carbon elements, meaning that this actually a plastic or polymers. And sometimes you can spot a few elements, other elements in the microplastic. It can be metals and so on. So this shows that a very cracking with some sort of biofilm attached to the microplastic. And it shows you the morphologies, as either it's crack, aging, peak hole, biofouling. Okay, you see the biofouling on the microplastic, meaning that this plastic can be it has been in the water or in the environment for quite some time. That is become a house for other microbes to attach to the bioplastic. So that's why some of the plastic, even though it's a light density of plastic, such as polyethylene or polypropylene, that's supposed to be floating on the water surface, but sometimes we still found them on the sediment bed because of this kind of co-pollutant that attach to the microplastic that make it more heavier or denser than the water density. So it sink into the water column and onto the sediment bed. So that's why SEM EDX is one of the important technique as well in characterizing microplastic. If you want to know the whole story about the microplastic. And later on, we go to spectroscopy. It's either Raman spectroscopy or IR spectroscopy, but the most widely used nowadays is IR spectroscopy, infrared spectroscopy. It's either the ATR or the microspectroscopy. So as to introduce the spectroscopy is a process that we get information by interaction of the light with our substance, because of each of the molecule has when the light penetrate into the molecules, it can vibrate. When the vibration happens, it's either the molecule is stretched or it's bending or rocking or twisting or scissoring. So meaning that it's stretched and it has to be deformed again. So this information has been given into the spectrum, and then for chemists or analytical chemists, we are very when we analyze the microplastic, of course we're using the polymer library, but we not solely depend on the polymer library to rationalize the findings. We also see each of the bands and spectrum to see whether this is logical or not. So if for example, this is a stretching for CH stretching, and it must come with the bending of CH and the rocking of the CH as well. So this is the stretching of the carbonate that can be found in aging microplastic, because, for example, this is a polypropylene. Okay, you have a CH stretching here a lot and CH bending here a lot. Okay, and then the carbonyl here not supposed to be in a polypropylene structure, but it formed in environment because of oxidation happened to the polypropylene. So it gives you story that it tells you that this polymer or this microplastic has been aging or has been oxidized in the environment, so that there's a one new peak come out, presents. Okay, so it's a carbonate peak, C double bond O at 1700. So that's why using a spectroscopy method, you can actually use usually use for a qualitative analysis, meaning that you compare with the library search. But actually, if you want to use spectroscopy technique for quantitative analysis, yes, you can. Especially if you have a chemist in your team, you can use simple Beer-Lambert law analysis by converting all the peaks into concentration amount, and you have a calibration, and you will get a concentration of each of the peak, and we can convert into each polymer peak. So using a simplified Beer-Lambert's law. Okay, so if you are using FTIR, we can either choose either transmission or ATR. Okay, transmission, of course, it has a good sensitivity. Okay, but you need to have a sample size amount that at least 30% of your KBR mix, so that it can be detected. Okay, and then that sample usually is gone because you press a sample in the KBR mixture. And then, but nowadays for microplastic, we usually use ATR mode, attenuated total reflector modes, using a diamond. We just place the microplastic here on top of the diamond, and we can search for the library for the microplastic. So, but there's a tricky part of you using the diamond, because you need to have a bigger, large microplastic to be able to be put on top of the diamond. Otherwise, it can be blown off from the diamond stage, and you cannot see it. Okay, so to overcome that problem, we can actually attach the FTIR with a microscope. There's a few IR that coming standalone. There's a micro FTIR was standalone, such as a Bruker Lumos or Nicolette Thermo Fisher and so on. But some FTIR as well can couple or can be attached to a microscope. Not the standalone mode. Okay, this is an example of micro FTIR results that you can point and shoot your microplastic and it give you the spectrum and you analyze it. So for example, this is a polycarbonate, polypropylene, and polyethylene microplastics. So, but there's a comparison between micro FTIR and FTIR is the sample size. You need to have a large size of microplastic if you want to use or if you want to analyze with ATR FTIR. But if you have a smaller size of microplastic, please use micro FTIR, or you can use Raman. So this is example of micro FTIR analysis method that being published. So it's easy. It's either this is a standalone micro FTIR. This is also standalone micro FTIR that we have. So it's actually is easier, but the time is a little bit consuming compared to when we are using chromatography GCMS pyrolysis. Because when you point and shoot, you need to have a very good background resolution and so on, because the smaller the microplastic, the harder you want, the harder you can point and shoot the particles. And then this is a Raman microscope. So you have a Raman spectroscopy that attached to the microscope. So it's also a vibration spectroscopy technique, but the range of wavelength that they are looking for is actually different with the IR. So actually, the IR, the FTIR spectroscopy, they are looking for the vibration of the functional groups, meaning that anything molecule that attach to the backbones. So they are looking for all the stretching and bending of the functional group. But for the Raman microscopy or Raman spectroscopy, they are looking for the vibration of the backbone of the polymers. So each polymers is the bigger the polymer have a different vibration capabilities. So it's given you a different peak style and so on. Okay, so the good thing about the Raman microscopy, it can detect a very small size of microplastic. Less than 20 micron of microplastics can be detected with Raman microscopy. But we don't have this in our university. So we have to collaborate to do this. Okay, and later on, the scientists or all the chemicals, all the chemists, they come out with analytical chemists, they come out with a different technique to analyze the microplastic. One of it is nowadays is pyrolysis GCMS. So the technique is similar to other GC, gas chromatography, mass spectrometry methods, but because of our analyte of interest is a polymer. So polymer is hardly to be broken down with at a normal temperature. So it has to be pyrolyzed at a very high temperature. For example, 500 Celsius or to 600 Celsius. Before it will be volatilized and it will be carried out in the GC and detected in the mass spectrometry. So actually, this is one of the examples that you can see from a few labs. For example, this is from LDP lab that been extensively used the pyrolysis for polymers and rubbers. So when the sample is introduced into the pyrolysis cup, so the sample has been volatilized, okay, fragmentized and separated based on their molecular weight or polarities, and been detected by the GCMS, been detected by the MS and come out with this kind of data. So each of these peaks actually tell you about the chemical compounds that broken down based on that polymers. Okay, So this actually, that's why you need a GCMS here. So that you know what's actually of the polymer. But this kind of technique, usually the pyrolysis GCMS is a very expensive technique nowadays. It's still expensive. And then that's why it's not really routine. I think FTIRs is more routine for the lab that working with microplastic. And there's some sample preparation needed still. Usually we need a clean sample that easier, so that we can overrule all the other contamination from environmental contamination. And but still the sample preparation is still minimal compared to thermal desorption or headspace GCMS. The sample will be disrupted, okay, meaning that you won't get the sample again back, okay, and the cost is expensive, and the size usually you only need 100 microgram of microplastic for each of analysis of microplastic. So this is how we do it in here in our research group. We have a polymer standards that we run to the GCMS pyrolysis, and we validate and optimize the technique and before we try with the real sample analysis. During the validation, there's a few parameters that we taken into consideration, such as the temperature of pyrolysis time, oven temperature, or other or GC parameter for the analysis. So this is the method that we published before. Usually the sample preparation that needed for the GCMS, the pyrolysis GCMS, we need to clean up the sample. So for this, we clean up with a TMAH that been diluted 25% in methanol, so that we can speed up the thermokinetic reaction or thermochemolysis reaction of the pyrolysis, okay, before it's been introduced into the pyrolyzer, meaning that we speed up the fragmentation of the polymer. So this is a few of our data. Okay, so using this technique, we actually can know the concentration of the microplastic in that sample. Meaning, for example, using a GCMS pyrolysis, okay, we actually looking for the iron interest, meaning that fragment iron, because this type of polymer, when it's broken down, it's broken into a specific fragmentation iron. So this specific fragmentation iron that we are looking for in the GCMS using this method. So actually we know the concentration of the plastics in our samples. So this is a few things that we need to consider when you are working with microplastics. Okay, we actually, you're supposed to work with microplastic. It's a very cross-contamination lab. Okay, so to avoid airborne microplastic, or if you don't have this kind of lab, you can actually prepare a negative blank or positive blank contamination for the QC and QA analysis. So everything that been detected in your blank, you should exclude it from your sample during the analysis. So this is a few airborne microplastic that's available in our airborne. So if you are not working with a cross-contamination lab, this kind of microplastic might contaminate your sample that we not realize it. So when we talk about microplastic, of course, we would like to see how this data can relate to the risk assessment, how this data is important. So we try to do a very extensive statistical and data correlation using a hot map or multivariate analysis. So using this, this is, for example, that we are trying to correlate the microplastic in a water sample with physical parameter that we collect in situ during the sampling time. So we found that, for example, here, there's a very positive correlation quite positive correlation in salinity with the microplastic, for example, but it's a negative correlation of pH for microplastic in sediment. So and so on. So using this, we can correlate the importance of our data. And for the risk assessment, we try to use a biplot using a PCA analysis. The risk assessment can be predicted or can be calculated based on predicted environmental concentration, where we have an amount of particles in our samples, and in a ratio with the RCR ratio, that our RCR or risk characterization ratio is more than 1 will indicate the environmental concentration that exceeding the safe concentration, while the RCR less than 1 will stipulate that there will be no environmental concern for that kind of polymer. Okay, so this kind of assessment is very important, especially when we know that nowadays there is also a plastic pandemic, okay, because of the pollution, plastic pollution because of the pandemic, sorry. So we know that there's a lots of polypropylene mass has been thrown up in our ecosystem, so it can be dangerous to our ecosystem. So I think that's all. I covered everything. Microplastic, at the end of the day, we know that microplastic is everywhere. So a very good monitoring program is needed, especially I know that in Asian, we have we just have Asian regional action plans for combating this marine debris. And in Malaysia also, we just started a national marine litter policy that just been launched early this year, I think, but still working on that. And there's a lot of standards and protocols that can be found in many in the UN, UNEP, IOC WESPAT, and so on. And that's what and that's all, I think.
Thank you, Professor Sabika Tonanwar. That was an excellent, comprehensive presentations on the methods with respect to instrumentations, as well as the statistical analysis down the road. I think we learned a lot from that in the efforts to have a regional SOP in a way so that we can have more compatibility in terms of monitoring in different areas, coastal areas, deep sea, wherever you are, particularly in the Southeast Asia, that seems to be one of the biggest culprit of this issue globally. Thank you so much. That was super comprehensive. I think many people, especially people who are starting microplastic research, could learn a lot from your presentations. Thank you. Thank you, Dr. Sabika. Next, I would like to introduce our last invited speakers. Last but not least, I have introduced him previously. Professor Wei Dong Yu, I guess what I want to highlight also has been collaborating for many years with Indonesian scientists before at LIPI and now at BRIN. So without further ado, finally, time is yours, Professor Wei Dong Yu.
Yes, thank you, Chair. It's yeah, I share my screen. So you see my PPT file. It's
Yes, I can see your PPT files.
okay. So my voice is okay now.
Yes, perfect. Everything perfect.
Okay, thank you very much for your patience. It's very sorry for the trouble. Yeah, today it is my great pleasure to join the meeting and share with you some information I have, particularly with the relevance of collaboration with Indonesia. So I will talk about the monsoon climate connecting the eastern and western Indonesia. And
Professor Yu, I lost your voice. And I think my colleagues also have the same issue. Professor, can you hear us? So again, we can see your slides, but your voice just left. No, we still can't hear your voice. It was perfect in the beginning, and then we start losing you. I thought that was my internet, but then I think everybody couldn't hear as well. We still can't hear you. Are you having issue with your internet? We can't hear you. There's a bit like a noise for a bit, but then we can't hear you anymore. Okay. Let's try again.
Professor Wedong left the group
Yeah,
and rejoin. Okay. He is already admitted to the Zoom. Can you speak, Professor Wedong?
Professor Wedong, we still can't hear you. Can you say something? No, we're not hearing anything. Okay. Mas Fadli kontak langsung dengan beliau.
Ntar saya coba.
Oke.
Mas Isal, Bu.
Oke, Mas Isal. Let's see, I'm trying to find an alternative. I mean, we can, if your internet connection is not in a good area, we can help by sharing your screen. So you don't have to turn on your video, you just speak. Should we try that? All right, perhaps while we're sorting this out, because we are actually on time, and on the organizer, please keep contacting and retrying with Dr. Yu coming back. But plan B will be we start the Q&A session for Professor Steinke and Professor Sabika. Dr. Yu, can you hear us? Or can you speak to us? So, yeah. The thing is, we can't hear anything from you. Okay, so organizer, please keep contacting Dr. Yu, maybe on another Zoom, making retrying. Oh, you are at a ship. Oh, no. Okay, that explains. Okay. In this case, let's start with the Q&A for Professor Stein. Oh, yeah, Professor Steinke and Professor Sabika. But Dr. Yu, we still hope that we could, listen to your speech. So we're trying to have things happening in the background. Okay. Oh, my first question from, two questions from me. Professor Steinke.
Yes.
The first question was that, in the beginning of your talk, there's an inference of the southward migrations of the IPCZ using data from the southern part of the hemisphere. But do you see this being supported by data in the northern side of the equator, perhaps using sediment records in the South China Sea? Or would that be another issue?
To my knowledge, there is no, can you hear me? Yeah.
Yes.
To my knowledge, there is no comparable record in the South China Sea.
Okay.
In my opinion, I have not seen those. And another problem is in South China Sea, for example, also we have there also very high sedimentation rates. Yeah, in terms of accumulation sedimentation rates. Yeah, we would be also able to carry out very high resolution studies. But the point is, for example, the delta 18O of seawater, for example, that we can nicely use, right, in the Indian Ocean of Sumatra, for example, right, cannot that nicely apply to South China Sea. Because, yeah, you know that, right, during the last glaciation, right, Sunda Shelf was exposed, yeah. And even also during Heinrich events, we had fluctuations in sea level. And that definitely would result in shifts of the river mouses there. Yeah, that is exactly what we have shown in one publication, right? That this delta of seawater in the semi-enclosed southern South China Sea is strongly controlled by sea level changes. And therefore, I do think that might be difficult. But what would be working would be deuterium isotopes. Because actually that is a great idea, right? To do that, right? So also I know it's not so easy to measure this delta D, right? And carbon isotopes of leaf waxes, but that would be a very nice idea to test our results.
Great. Yeah, thank you. I mean, that's something that we need to remind, that it's not an easy thing to find a location that we can, try to look for these signals. For example, you can't just easily go north, but then you get South China Sea with all its complications in terms of paleo as well, right?
Yeah, exactly. And on the Sunda Shelf, there is no sediments, right?
Yeah. So that leads to the next question. I mean, it's also not easy to find, even more challenging within, for example, Indonesian internal waters. We talk about, it will be interesting to go to Eastern Indonesian Sea, but also in the Eastern Indonesian Sea, there's a lot of earthquakes, which disturb the sediments. But I'm just wondering, from your hunch, where would be a nice spot to hit in terms of trying to get those long records, but also less disturbed, in your opinion, in the Indonesian Sea?
That is easy to answer, because I would go to Sumba, because you have seen our my last slide on this Holocene, on this upwelling reconstruction we have done there, right? this Holocene variation. That is a core of Sumba Island. And there we got an almost 10-meter long core that is only covering 6,000 years. Imagine if we go deeper, if we go deeper 20, if we would have 20 or 30 meters, right? It would be really high resolution there. You know.
I see. Okay.
So that is, for example, that would be a talk idea to go there, for example. Yeah. But anyway, there the sedimentations are in particular very high there. I don't know what, we do not really know about this, the reason for that, why we have this sediment focusing there. Yeah. And to your second question, that is a very interesting question, because when we're working on this core from Sumba Island, even because we have there so high sedimentation rate, we were not able to date the core top of this gravity core by radiocarbon, for example. Yeah, because it was much younger. So we used lead 210 and americium isotopes. Cesium was not working there. That was interesting, right? But americium from this nuclear weapon tests, yeah, we see the pretty good signal. And interesting, what we have found there are this typical typical cores, but 2 times. It seems that we had there maybe some small scale mass movements on that place. Yeah.
Okay.
Most likely related, as you said, to earthquakes there. Yeah.
Yeah. Thank you. Yeah, that's something to think about when collecting in such earthquake active region.
Yeah.
So the second question is is from Fareza. His question is, how important is the proxy development and calibrations by modern samples for paleo-environmental reconstruction in Indonesia?
Can I see, please, the whole question, please? Proxy.
Yes. It's how important is proxy development and calibrations by modern samples?
Because when thinking about our research, right, one of the backbones, yeah, is dating. Yeah, we needed a good age model for our records. But the second thing is exactly that, this proxy calibration. Yeah. Yes. Luckily, so that is really important, right? When we, for example, want to do reconstruction of upwelling, right? We know that this is seasonal there of Java, Sumatra. Yeah. So that we have this seasonal upwelling, right? During the winter.
Sorry, there's leaking of a voice.
Yeah, for example, during the winter. Yeah, so we need, for example, a planktonic formniferous species that only is living during that season. Yeah, during the upwelling season. Then we can have a seasonal record. Yeah. It does not help if we, and for that reason, we have luckily off Java there was a sediment trap. So sediment traps are really, really important to get an idea about the seasonal occurrence of planktic foraminifera, for example, and that has been, we were lucky to do that, right? We have already a kind of a ground truthing there, yeah, but that needs to be definitely strengthened over a longer period. We had this only for 1.5 years, yeah. For example, longer sediment traps would be pretty nice, right? Also maybe to get an idea about the impact of El Nino and IOD, right, on the upwelling or on discharge, whatever, right, where you want to address your questions. Yeah, this proxy calibration is very, very important.
Yes, particularly in Indonesia, as you mentioned at the beginning, you show the ITF and you're like, oh, this is very complex. So
yeah,
that complexity, that's why calibration is very important in Indonesia.
Exactly.
So
yeah, and then, yeah, of course, that it depends definitely on, yeah.
Yes, thank you. So I mean, this could motivate paleoclimatologists in Indonesia, not just working on established proxies and applying on sediments, but also, here we are in Indonesia, we could have very solid studies at reconstructions that could be applied in Indonesia, but also could have impacts for tropical regions in parts of the world. So that could be nice to.
Yeah, I know you have done a lot of coral studies, for example, as well, right?
All right.
In that area.
Thank you. So the third question is from Pak Agus Setiawan. Prof. Steinke, based on your studies, is there any return period of extreme condition can be identified related to the climate conditions?
Well, I do not really understand this question.
So, well, Pak Agus is here. Maybe Pak Agus wants to.
Yeah, can you add a little bit?
Yeah. Okay. My question is, you using isotope to analyze the condition in the past years before, yeah, and from your study, from your result, is there any extreme condition that with a return period that can be identified based on your isotope analysis?
Extreme events, yep. What?
Yeah, for example, that there are some event that has a return period for example, 100 years or some longer than that, and that can be also identified in your isotope analysis result.
Yeah, as you have seen when I was talking about this about the changes in upwelling intensity over the last 2000 years, for example. Yeah, so as you know, when we're talking about monsoon, we're talking about seasonal, right? And when we are talking about IOD and ENSO interannually, yeah. So we usually do not have the resolution, right? Coral people, they have a much better resolution, right? Therefore, we are talking about El Nino-like, for example, or IOD-like, positive IOD-like conditions. Yeah, we cannot do better. Yeah, but we see centennial to millennium scale variations. Yeah, in our records, whatever we are looking on, right? This sea surface temperatures, thermocline temperatures. That is what we can see. Hope this answers your question.
Okay, thank you very much.
Yeah.
Thank you, Pak Agus. Okay, we can go to the next slide. I think there are more questions.
Yeah, there was.
Please, operator, for the next slide. Okay, this is also for Professor Steinke from Selfie Makarim. I would like to ask about the JAS figure you show that based on ENSO indications during 2002-2009 from Southeast Monsoon, JAS 2003 with El Nino, and so on. Since in Indonesia, how about the IOD pattern? The question, since Indonesia couple IOD ENSO bringing climate stability, perhaps ENSO mainly the ENSO mainly stronger at the SS than mixed layer, but at the thermocline layer, the signals of IOD is more prominent. What insight about this?
That is a very interesting, very important and interesting question. So my answer is, yeah, we cannot really disentangle that, honestly. Yeah, because the effect is the same, right? If you have El Nino conditions or a positive IOD, yeah, you have increased upwelling, for example. Yeah, and the problem is that we do not have this high resolution IOD proxy records to compare our records. Yeah, that would be also a topic for the future. Yeah, go more to the not to the east, go more to the west, and identify high resolution records for changes in IOD, maybe over the last 2000 years. Yeah.
Yes.
The second part of the question, right? Yeah. Yeah, Selfie knows better about that than me, right? Is that, but the point is, right, when thinking about that, right, having a positive IOD, right, we have a shallower thermocline, yeah. Also in the eastern Indonesian water, also a stronger upwelling, same as for ENSO, right? Yeah. I would say the complete upper water column, inclusive thermocline, is affected. So actually, I cannot say if ENSO is stronger at the surface, but in the area of Java, in this upwelling area, right? I would say that thermocline and mixed layer is affected in the same way, right?
Okay.
Selfie, what is your opinion about that?
Yeah, I'm thinking about that too.
Yeah.
Stefan, so make sure next time you can come to Indonesia, then we can have cooperation with BRIN, then we can do some observation together.
Yeah.
Let Dr. Intan can be as a PI for the cooperation. That must be great. Yeah.
No, that is exactly the point I was thinking about. I will do now in the next day some research. What kind of cooperation programs are available from China or from Indonesian side, right? To do this research.
Yeah. Great. Thank you well, Stefan.
Thank you well. Yeah, thank you.
Thank you. So I think we have the next questions are more towards Professor Sabika. Can we see the questions? Can you show the slide, please?
Pak Zen.
Pak Zen. Thank you. Because he's still compiling at the same time. Okay, the first question, Professor Sabika, from Pak Agus. Based on the existing analysis methods, is there any opportunity or possibility to correlate microplastic conditions to the ocean climate condition and variations, especially study long-term variations? So I think you mentioned about this correlation with different ocean parameters, but how about thinking in the longer terms? Is there any different methods that you have in mind for that? Please, Professor Sabika.
Okay, thank you, Pak Agus, for very good questions. Actually, okay, so I think to answer this question, yes, it's a very important to have a physical parameters of the ocean or of the water during the sampling, so that we can see the correlation of the microplastic abundance and the current trends of the climates or the physical parameters of the seawaters. For example, during the leaching or absorbing of other co-pollutants to the microplastic actually is a exothermic reaction. So when we're dealing with exothermic reaction, it's actually involving the change of the heat or energy. So that's why indirectly, when you're having more microplastic in the ocean, it will reflect to the addition of sea temperature for the climate change. So I think yes, it's a very correlated. So that's why a very extensive monitoring is needed. So we need to have a very good monitoring data. So because in UMT we don't have that capacity and capability, we cannot afford to go for a voyage every year. Because it's because of funding and so on. Maybe at BRIN, I think you guys should do the extensive surface monitoring and try to relate the in situ physical parameter with the microplastic. And I for sure that you can find a very good correlation, because what we did for the coastal area in Malaysia, we found a very good correlation of physical parameter with a few physical parameters with the microplastic abundance. So and again, and if we're looking in the chemistry side, of course, during the absorbance or leaching of microplastic of co-pollutants to the microplastics, it's involved change of the energy, change of the kinetic, and of course it will correlate, or it will stimulates the heat or climate change. That's my answer.
Thank you. Thank you, Professor Sabika. We're gonna have more Q&A for the next question. Okay, so this is about the availability of those instruments. That's why it is found that microplastic concentrations in here usually without polymer identification, which may cause overestimation. Consequently, the microplastic concentration in this water are very widely vary. Do you have any suggestion of how we can still do chemical identification for a small microplastic if we only have common FTIR? Is there any way to tweak it?
Okay, so if you have only a benchtop FTIR, of course, it limits your analysis to only a large part of microplastic. But if you still do need to use to check your small microplastic, one thing that that's a very conventional way that you can try to pull the same types of shapes of microplastic, the very small ones, into one glass centrifuge tube, and you just centrifuge it, and you can get the clump of the microplastic actually, and that you can pick and put on top of the ATR diamond. But the risk of doing this is if actually you have a different type of polymer actually, but looking the same, shape the same, the size is the same, the color is the same, but you try to put together and it's become a mixture of polymer. So you need to have a very good background in chemistry to try to identify, because the library will give you a percentage of matching. So you have to have a very basic knowledge how to identify the spectrum based on the matching of the library. If you try to pull the polymer, yes, you can do that. That's a very conventional way, I think.
All right, last questions before we go to Professor Yu. Can you show the questions, please? Pak Zen. Thank you. Okay, this is also from Cory. In your research finding on microplastic and human colectomy specimens, does the state of colectomy correlate significantly to the concentration?
Okay, actually, because when we're dealing with a human colectomy, it's actually a living human. So we try to see different of, you cannot just simply open up anyone guts and try to, can I have your guts a little bit? No, it's actually they are in kind of like some sort of procedure, like maybe involving a cancer procedure, collection of the cancer cell for the human guts, in the human gut. So we just request that with the ethics issues, we just request that we want to have guts that been involved in a possible cancerous activity, and also guts that are a little bit healthier compared to another spot. So we try to compare that. Yes, the real findings that we still this kind of research is still ongoing, but we found that a good correlation in between guts that has damage compared to other parts that not really that damaged, with the microplastic concentration at that gut, and different type of microplastic as well. So we are very looking forward to publish our new findings on this as well.
That's very
the research, this one we're working with Malaysian Science University, HUSM hospital, and also Washington's University in the US.
Thank you. That's a very intriguing research, but also very important. Okay, now we have come to, let's try again with Professor Yu. Thank you again, Professor Steinke and Professor Sabika, for presentation and questions. So let's try with Professor Wei Dong Yu. So how are we gonna do this, guys? Okay, so we have his presentations and he's connected to a phone in terms of talking. Professor Yu, are you here? That's it. Okay, so is Professor Yu connected to any of the, he's not joining the Zoom, right? Mas Faisal? But he's not talking to you.
He is joining the Zoom
Oh,
to device. But maybe if we can use Pak Faisal as the sound for Pak Wedong. Thank you, Pak Faisal.
You can start.
Okay.
Okay, you can start.
Floor is yours. Oh, we can't turn up the volume. It's still very small. I can hear him talking, but the volume needs to be higher. Hello? Yeah, Mas Isal, is this the maximum volume?
Yes.
Oh no. I can't, I think he has to yell.
Yeah, I can hear you, but the sound is a little. What is it?
No, can't hear.
Yeah, I can hear you, but hello, hello, hello. Oh,
It's very small, yeah.
it's very small, right?
Mm. Okay, this is not good. So this is already on speaker mode, maximum, but it's just that we you know, the connection is just not very strong. Can you, I mean, in your phone, can you hear him well though, Mas Isal?
Yes.
You can.
Hello? Okay, okay. I can hear you. Mbak Intan?
Yeah.
You can hear me?
I can hear you very well, but I cannot hear Professor Yu. It's just very soft.
Oh, very.
Your voice is very clear.
But for the, how about the?
I cannot hear Professor Yu. Oh no.
Hello, hello. Can you hear me? Hello.
Oh yeah. Oh yeah.
Hello.
Yeah. Oh yeah.
Hello.
Hello, 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 hello.
That's
Yeah,
good.
Faisal. Can you hear me?
Yes. Let's do this.
Oh, it's got. I don't know why. It's maybe the connection is too unstable on a ship. So yeah, I hope I can take the last minutes to talk. It's I will talk about the monsoon connecting the eastern and western part of Indonesia, and yeah. Oh wait a minute.
Operator, please.
Operator, next slide, please.
Yes, next one. Yes, this is a Asian monsoon. Looks like, so we say the monsoon is blowing from the southern hemisphere all the way to the northern hemisphere. So for China and Indonesia, we are connecting by the monsoon circulation. So every summer, China receives a lot of rainfall. In fact, this rainfall, the water source is coming from the tropical Indian Ocean. Next one, please. Yes, this is the monsoon rainfall. The red color means in a rainfall occurs in a borrowed summer season, and the blue color over the Indonesia and Australia means the rainfall comes in a borrowed winter season. So this is a monsoon rainfall and also the monsoon circulation and connecting northern hemisphere and southern hemisphere. So today I will mainly talk about the intra-seasonal variability of this Asian monsoon. And for this, when we look at the intra-seasonal time scale, it's connecting the eastern and western part of Indonesia. Next one, please. Yeah, I would like to share my very outstanding, excellent memory working together with the Indonesian team. It's in fact my first cruise that was in 15 years ago. It's 2007. I still remember we have a very good cruise in the Eastern Indian Ocean. So I sank a lot with my colleague, old friends, Agus there. Next one, please. Yeah, we had some cruise in the Eastern Indian Ocean. So my previous experience working with Indonesia is on the western part of Indonesia. So this meeting brings us a new opportunity to look at the eastern part of Indonesia. Next one, please. Yes. So when we look at the last 3 decades, from point of view of observing system development, the Indian Ocean and the Pacific Ocean has a good development. But when we look at the connection part, the South China Sea, Indonesian Sea, and the Western Pacific region, so we do find there is a large space of a gap. So that means the eastern part of Indonesia and the Western Pacific is a gap for our understanding, for our study there. So it's I really appreciate this conference address this issue. Next one, please. Yeah. So for this intraseasonal oscillations, I introduced 2 kinds of intraseasonal process. One is a Madden-Julian oscillation. You are quite familiar, I guess. So Madden-Julian oscillation is coming from the eastern, from the tropical Indian Ocean all the way to the Pacific Ocean. So that means this eastward propagation will cross a maritime continent. And this is what I mean, the intraseasonal oscillation connecting the Western Indian Ocean, Western Indonesia and the Eastern Indian Ocean, because it's just come across the maritime continent. Another way of intraseasonal oscillation is called borrowed summer intraseasonal oscillation. So for this oscillation, it's propagate from the tropical ocean to the northern hemisphere to the northward propagation. So that means this borrowed summer intraseasonal oscillation connects Indonesia with China. Next one, please. Yeah, this is a seasonality of these 2 kinds of intraseasonal oscillation. So you say it's the Madden-Julian oscillation mainly occurs in austral summer or borrowed winter, and borrowed summer intraseasonal oscillation mainly occurs in June, July, August, September, October. So it's all year round. We have a strong intraseasonal signals there. Next one, please. Yeah. You all understand quite well. The Madden-Julian oscillation was discovered a half decades ago by the Madden and also his colleague Julian. Next one, please. And I would like to share with you the recent study we found. One Chinese scientist also working on this Madden-Julian oscillation at a much early time, that is in 1963. So her name is his name is Xie Yibing. So that is why now we suggest maybe MGL can we call it XMGL. It's a 谢一丙 and Madden-Julian oscillation. Next one, please. Yeah, this is a movie. I show you what the MGL looks like. Its propagation is very clear. So the yellow color means the rainfall. The red color means a dry condition. So this wet condition, dry condition, just propagating from the Indian Ocean all the way to the Pacific Ocean and cross over the whole maritime continent. So this is what I mean by the connecting the Western Indonesia and the Eastern Indonesia. So I would like to emphasize when this MGO cross over the maritime continent, it brings a lot of air-sea interactions over the Banda Sea, over the Timor Sea, and over the Papua. It's Eastern Indonesia seas. So
we lost you again. Professor Yu, can you hear us? Mas Isal, can you also contact him directly?
Okay.
Oh, I thought that was perfect. Thank you. So it's amazing. He's in the ship and he still could give this presentation. Professor Yu, can you hear me? Can you hear us? So it seems that your connection is down again. In fact, I do not see him in the room anymore. Oh, he's in the room. Professor Yu, yeah, we just lost you again. No. Mas Isa, are you on the voice? Are you on the phone with him?
Baitan?
Yeah.
He can hear your voice, but
He cannot.
yeah, or maybe he just suggests to make a video.
Okay. How about this? Because we will be having a break. Initially, we still have 15 minutes. So we also don't want to rush Professor Yu, of course. We can have a break now, and then we can incorporate Professor Yu after the break. How about that? So before we go into different, so we're gonna have a break for now 45 minutes at about 1 p.m. here in Ambon or 11 p.m. in Jakarta. Sorry, I'm losing time now. Sorry. Oh, okay. Now it's 12:15 in Jakarta. So we're gonna have a 45 minutes break. We're gonna meet at 1 p.m. Jakarta time. But initially we are gonna have into sessions, but we're going to listen to Professor Yu's recording first. How about that, guys? Because I really appreciate Professor Yu. You're making the best effort to join us. So yes, please record your video and share it to us, and we will play it after we have the break. Okay, if that sounds good, because I know you will need time to record your video as well. Okay, so Mas Zen, you can take down the share screen. Okay, thank you everyone, and thank you for your attendance, for your contributions to all the.
Bintan, I'm sorry to interrupt.
Yes.
How about photo session with the invited speaker?
Oh yes, yes. Yeah. Thank you for the reminding. So in the previous sessions, we just realized that when we did the photo session, we didn't have Professor Yu, Professor Sabika. So we're gonna do this again. So Mas Fadli, please help with this. So everybody keep put your video on. Okay. So we don't have Professor Yu because his connections. Okay, Mas Fadli, you can give us the cue.
Wait, we have Professor Tenke and Professor Sabika. Good. Yeah. Maybe I will count from 3, 2, 1. Put a big smile.
Smile.
Okay, wait a moment. Okay, once again. 3, 2, 1. Put a smile. Okay, good.
Thank you, Mas Fadli. Thank you. We can still keep it like this. Thank you again, everyone, for your attending, and we'll have about 45 minutes break before we go into plenary session. Now, after we listen to Professor Yu's recording, you can choose your own breakout groups and into different topics. Thank you again. Have a nice lunch break. See you in 45 minutes. Bye-bye.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045 Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policymaking, and societies.
Mengawali awal tahun 2021, Lembaga Ilmu Pengetahuan Indonesia melakukan 2 penelitian dengan kapal riset Barunajaya bersama Institute of Oceanology Chinese Academy of Sciences dan First Institution of Oceanography China. Berlayar mengarungi Laut Indonesia, penelitian ini dibagi menjadi 4 perjalanan. Barunajaya melaut dari Jakarta menuju Ambon, lalu dari Ambon menuju Bitung. Kapal riset LIPI ini kemudian melaut kembali menuju Banyuwangi dan kembali menuju Jakarta untuk melego jangkar. Penelitian pertama yang dilakukan dengan kapal riset ini adalah Circulation and Ecosystem Study in the Indonesian Seas under the influence of the Western Pacific Friability. Kerjasama ini dilakukan dengan Institute of Oceanology Chinese Academy of Sciences sejak tahun 2014. Tahun ini dilakukan perawatan mooring yang telah dipasang pada 2018, unduh data, dan pemasangan mooring baru di Selat Livamatola, Laut Halmahera, Maluku, Talaut, dan Sawu. Sedangkan penelitian kedua adalah Transport of Indonesian Seas, Upwelling and Mixing Physics atau TRIUMPH. Kerjasama ini dilakukan oleh Pusat Penelitian Laut Dalam berkolaborasi dengan First Institution of Oceanography China dan University of Maryland USA sejak 2018. Pada tahun ini dilakukan pengukuran sifat fisika air laut secara terus-menerus menggunakan berbagai peralatan yang ditempatkan di dasar laut maupun di kolom air. Data ini berguna untuk mengetahui kondisi air laut secara deret waktu dan dampaknya terhadap kondisi atmosfer. Selain itu, dilakukan pengambilan sampel air laut, plankton, dan mikrobiota lainnya pada kedalaman hingga lebih dari 3.000 meter. Para peneliti LIPI tidak akan lelah untuk terus berlayar dan berlabuh untuk ilmu pengetahuan. Penjelajahan kolaborasi ini diharapkan dapat bermanfaat bagi perkembangan ilmu kelautan di Indonesia dengan mengenal lebih dalam kekayaan lautnya.
Halo semua, kenalkan kami kelompok riset dinamika lingkungan laut dalam atau KRIS Dalam dari Pusat Riset Laut Dalam, Badan Riset dan Inovasi Nasional. Dalam video ini, kalian akan menyaksikan informasi singkat terkait KRIS Dalam. Selamat menyaksikan dan kita akan berjumpa lagi di akhir video ini. Dilihat dari berbagai aspek kajian, Indonesia memiliki sumber daya kelautan yang sangat melimpah, baik di kolom air atau water column, di dasar lautan atau seabed, dan di bawah dasar lautan atau subseabed. Adapun yang harus dilakukan saat ini adalah memahami kondisi lingkungan di mana sumber daya kelautan ini berada. Kami adalah kelompok riset yang dirintis sejak tahun 2019 untuk melakukan kajian kondisi lingkungan, mengungkap dinamika yang terjadi, dan mengidentifikasi ancaman yang mengintai, baik di dasar lautan secara umum dan khususnya di kolom air dengan kedalaman lebih dari 200 meter. Potensi area kajian laut dalam sangat luas. Indonesia adalah negara dengan luas lautan 6,4 juta kilometer persegi, di mana 32% adalah lautan dengan kedalaman kurang dari 200 meter, 25% memiliki kedalaman antara 200 sampai 2000 meter, dan 43% memiliki kedalaman lebih dari 2000 meter. Riset-riset yang sudah dan sedang dilakukan adalah riset karakteristik dan dinamika fisis laut dalam, riset nutrien dan karbon laut, dan riset pencemaran mikroplastik. Sedangkan topik riset ke depannya adalah riset biogeokimia laut dalam, riset pencemaran di laut dalam, dan riset hubungan lingkungan laut dengan kondisi makhluk hidup di laut dalam. Melakukan eksplorasi laut dalam tidak diragukan lagi sebagai salah satu kegiatan yang menantang dan frontier di Indonesia. Hal ini juga mendukung salah satu visi kemaritiman 2045 di mana menjadikan Indonesia sebagai poros maritim dunia. Melalui kelompok riset ini, diharapkan dapat memberi kontribusi dengan mengungkap teka-teki terkait karakteristik dan dinamika laut dalam, serta bagaimana kondisi tersebut mendukung kehidupan makhluk hidup yang belum pernah terungkap sebelumnya. Selain itu, peran laut dalam mengatur iklim global juga menjadi fokus di dalam keris dalam. Menuju lebih dalam adalah semangat kami, dan kami telah mencoba sejak tahun 2018 untuk mengembangkan diri, menggeser batas-batas, dan mencari kebaharuan melalui kegiatan ekspedisi ilmiah di perairan Indonesia. Banyak permasalahan dan topik penelitian laut dalam yang perlu dikerjakan saat ini. Ratusan fitur dasar laut yang perlu diungkap, jutaan persegi luas lautan yang perlu dijelajahi, dan tak terhitung jumlah tantangan yang perlu diselesaikan. Kelompok riset kami saat ini terdiri dari 4 orang peneliti dan 7 orang teknisi. Dan jumlah ini masih jauh dari kondisi ideal untuk mewujudkan ide dan mimpi kami untuk riset laut dalam di Indonesia. Kami adalah jiwa-jiwa muda yang memiliki ide dan tujuan besar dalam riset laut dalam di Indonesia. Namun kami belum memiliki Anda, yang merupakan talenta-talenta berpengalaman dalam bidang ini. Mari bergabung di keris dalam dan kita wujudkan ide dan mimpi kita untuk riset laut dalam di Indonesia.
Okay. Yeah. Good afternoon, everyone, ladies and gentlemen.
Good afternoon.
Okay. Hello. Okay. Thank you, the committee. So good afternoon, everyone, ladies and gentlemen, all the participants. So we would like to start the parallel session, but I would like to inform you first that this main room will be used for the room number 4 for marine geosciences. So for those who are not assigned to this session, you can join the breakout room and choose the room that you assign. Please make sure that you have already reformatted your name as mentioned in the chat box, and we will start the parallel session. Okay, I would like to share the screen. Okay, as I mentioned that this main room will be for the marine geosciences. So all the participants in this session have been here, or so we will start the session.
Hello, excuse me.
Yes, please.
What is the topic of session room 4, marine geosciences? Is this more about deep sea exploration, or is it also going to be about, yeah, paleoclimate, paleoceanography? So
Okay,
what is-
actually for these sessions, we will cover the geology, geophysics, and geodesy actually, but
okay.
We have one topic actually, but I'm not sure, but maybe in the oceanography, maybe you can
Okay, yeah.
Go to the breakout room.
Okay, thank you.
Okay, thank you,
sir.
Okay, maybe the committee will please help the participant to assign to their rooms, because we will start this session in this room. Okay, we will start the parallel session room 4 for marine geosciences. For those who are not assigned in this session, please join your session in the breakout room. Yeah, we are so glad to greet all of you here. So my name is Muhammad Zain Tuakia. I will be the session chair for this session, and Ms. Rafida will help me as admin. So this is just simple rule and guidelines in before we start the session. So make sure that you already rename your name through this format, room number, and then your name, and then make sure you already use the virtual background that's shared by the committee, and then your time for presentation is about 10 minutes, and we will have 3 minutes in the end of your presentation for question and answer. So please stay mute your microphone until you speak. And then if you have question, please write down in the chat box, or the moderator could you to ask directly. Okay. We will have 10 presenters in this session. We will start from Miss Amalia, and then it will be followed by Mr. Eko, and then Miss Alodia, Mr. Hendra, Mr. Hananto, Mr. Nur Cahyo, and then Mr. Purnama, Mr. Arifin, Miss Vera, and the end, Mr. Ahmad. Okay, this link will be shared by the admin in the chat box. So please make sure you fill out the attendance form for the certificate purpose. So thank you. Have a great time with us. Okay, I will stop share, and I would like to invite it first. Sorry. Miss Amalia for the first.
Yes, Mr. Zain.
Yeah. Okay. Are you ready? You can start share your screen.
Okay. Thank you.
Okay, your time is about 10 minutes for presentation. So I will be your timekeeper as well.
Okay, thank you, Mr. Zain, and good afternoon, ladies and gentlemen, and all of the researcher. Firstly, please let me introduce myself. I'm Amalia, a student from Bandung Institute of Technology, and today I would like to present about my research study, the estimation of total vertical uncertainty from multi-beam echo sounder during megamal expedition, sea floor mapping for earthquake mechanism at Maluku Sea. So this is the background study. In 2022, the National Innovation and Research Agency initiated an A1 megamal expedition, and the goal of the expedition was to map earthquake and tsunamigenic potential areas around Ambon Island. One of the methods used in the expedition was the multi-beam echo sounder. As we know, the multi-beam echo sounder has an ability to map the seafloor efficiently and produce dense depth information. And to obtain an accurate bathymetry result, several stages need to be carried out. And this is the visualization how multi-beam use the sonar to mapping the seabed. Initially, a vessel reference frame, VRF, with a defined reference point called common reference point needs to be established. Afterwards, sensor offset in 3-dimensional were measured with respect to CRP. The next stage was to calibrate and validate the vessel DGNSS and heating system statically. Afterward, a patch test, dynamic MBAS calibration, is performed. Finally, the accuracy of the bathymetry result was analyzed in accordance with the International Hydrographic Organization. And this is the study area where the survey is held. The study area is located in Ambon and divided into 3 regions, as we can see in the map. The yellow polygon is the region of the survey, and the black area, in here, there are black area in the map, is part of the area sample for total vertical uncertainty calculation, and the green line shows a cross line of multi-beam echo sounder survey lines, and the red area where the patch test is performed. And here is the red area. Okay, this is the method of multi-beam SWOT survey. A 3050 LXC-beam MBAS system was used during the survey. The system was installed over side mountain on Airfield Barunajaya 8. The 3050 LXC beam system has a beam resolution of 1.5 degree times 2 degree. The system operated at 50 kilohertz and could be used for seafloor mapping with a maximum depth of 3,000 meters. The system has up to 193 soundings per watt with pointing angles automatically adjusted according to achievable coverage or operator-defined limits. And this picture is the visualization of the configuration of 3050 LXC beam, where we need to input the position and the heading of the vessel and the motion of the vessel, and also we need to input the SVP or sound velocity profile data to multi-beam online PC Hydrostar. Okay, next is the method of the vessel reference frame and sensor offset. In this expedition, CRP was determined at the IMU where X and Y values were 0 and Z was 4.63 meters above sea level. The vessel reference frame is defined as X positive in the bow direction. As we can see in this picture, this is the axis direction, and Y positive towards the port and Z positive towards the bottom. There are 3 rotations in the VRF. The angle of rotation or rotation about the y-axis is called positive roll if the left port is up. Rotation on the x-axis is called positive pitch if the bow is up, and rotation on the z-axis is called positive yaw clockwise. And this is the method for alongside calibration. So this is calibration was intended to verify the accuracy of the GNSS receiver and heading sensor used during the survey. The GNSS and heading calibration aims to obtain the alignment and offset parameters survey equipment sensor to the vessel coordinate system, navigation, and MBS transducer. As we can see in the map, there are two triangle, red triangle, and this is the benchmark, two control point benchmark were made at the port beside the RV Barunajaya, and this is the RV Barunajaya, the red polygon. And the two control points were tied in real-time kinematics RTK method to the national geodetic control points. And in here, in the map, we can see the national geodetic control point. We used to tie the two control points. And then the method of patch test MBAS calibration. The 3050 Alexa beam patch test calibration was carried out in West Out Ambon Gulf, and roll calibration was carried out on a flat seafloor. And in this map, we can see the area for calibration and also the area for pitch or yaw calibration, while the seafloor topography with a gentle slope was used for pitch and yaw calibration. Before carrying out MBAS calibration activities, sound velocity profile SVP data was needed to obtain a profile of the speed of sound in the water column. SVP data was needed as input or correction parameters for survey equipment that uses acoustic signal, especially MBAS. And this is the method to determine bathymetry result analysis in accordance with IHO SP 44 edition. And the study will refer to order 2. The water depth of the study area reaches more than 200 meters, means there will be no reduction of tides or sea level. This referred to as 44 edition, which states tides or sea level reduction is not necessary given for depths greater than 200 meters if it does not provide significant change against TVU. Okay, and this is the formula I used to calculate the TVU. Okay, this is the result and the discussion. This is the geodetic parameter with the aim that all coordinates produced in the survey are in the same reference. Therefore, a geodetic parameter is needed as a reference. And this is the geodetic datum. The ellipsoid used for this study is WGS 84, and the grid projection is UTM 52S. And this is the result of the vessel reference frame and sensor offset. There are many sensors in the vessel: IMU, waterline, the X transducer, RX transducer, and also the GNSS. And all of the result of the sensor offset is calculated in X, Y, and Z direction. And this is the result of alongside calibration. So the first table is showing about the result of the GNSS verification with standard deviation of easting is 0.03 and standard deviation for northing is 0.02. And another table is the result of heading calibration on RV Barunajaya 8 with the standard deviation of heading calibration is 0.30 degree. And this is the patch test calibration result. So before we do the calibration, prior to calibration activity, sound velocity profile data is required to obtain the sound velocity profile in the water column. And the sound speed have a range from 140-90 until 1540 meter per second. And this is the result of the patch test. As we can see here, I'm highlighting with the red box in here the seabed before patch test and the seabed after patch test. We can see this is the smoother seabed we can see in here after the patch test, then before the patch test. This is, we can see in here, right? Smoother. And then this is the calibration result. For the roll is minus 0.90 degree, pitch is minus 5.40 degree, and yaw plus 1.9 degree. And this is the MBAS data accuracy test result. This is the larger or bigger view for every adjacent lines that used for TVU sample. Okay, and this is the result of the TView check. So the multi-beam data accuracy test was carried out by taking 40 sample points from two adjacent lines. Samples are taken from overlapping points or adjacents, then were compared to the water depth, one line to another line, difference and assigned by computing the average and standard deviation values. And as we can see in the table, for every survey lines that used for TView sample, all of the region is accepted based on order 2 of IHO SP44. Okay, and then this is the conclusion. So after we do the several stages for get good accuracy of bathymetry data, the accuracy of MBS data is within the tolerance limit, which refers to the 2020 IHO SP44 edition, order 2, so the MBAS data are acceptable. And also, the study was supported by National Innovation Research Agency, BRIN, and I would like to thank BRIN for the data use permit and the conference funding. We also thank the Directorate of Research Vessel Management for the supporting during the Awan Megamall 2022 expedition. Okay, that's all for me. Thank you.
Yes, thank you, Ms. Amole, for your presentation. So you used the 10 minutes exactly for your presentation. So we will have 3 minutes for question and answer. Yeah, in the chat box. Okay, we don't have already, but if someone want to ask directly, you can just use the raise hand feature, and I will allow you to ask directly to the Miss Amalia. Okay, actually, I have one question, as I also part of this expedition. So, what do you think about the differences between the side-mounted MBS with the how to say the one that mounted directly to the bottom of the vessel?
Full mounted itu below the ship, full mounted.
Yeah, full mounted below the ship. So what do you think about the differences of, you have something to say?
Okay, I would like to answer the question. So as I know, the hull mounted MBAS will have a good imagery or good mapping, because there will no turbulence from the vessel. Because if we put the MBAS in the side mounted, there will be a distraction from the turbulence of the vessel. That's I know, that's what I know from survey yesterday.
Okay, so do you think the data is like fit already? I mean, we can use similar to the whole mounted or not?
Sorry.
I mean, so data that you get from the side mounted, I mean, we can still use compared to the whole mounted or something.
Yes, yes. We can use the data, because I have to the TView check, and based on the order 2 IHO SP44 for my TView check of the sample data is acceptable in order 2.
Okay, thank you very much. Okay, we still have 1 minute for the short question, if someone want to ask. Okay, Prof. Hananto, please, quick question.
Okay, thank you, Mr. Chairman. I just curious about the speed sound of the multi-beam. Yeah, you show us in the table. I think what's the number? Yeah, it's usually for marine seawater 1,500, but I saw the less than 1,500. Maybe you can explain. Thank you. The speed of the sound velocity less than 1,500. Mungkin you can explain why. Thank you.
Okay, this is about the sound velocity. So the sound velocity is above 100, actually.
No.
Yes.
Usually it's 1,500 actually. Sorry, but I saw less, 1,400 something. That's less than 1,500 for the water. Why? Maybe you can explain. Thank you.
As I know, maybe this is because the water depth measured for the SVP is range for 0 until 500 meters. So that's why the sound speed have a range for 100, 490 until 150, 40. I think that's the best answer I can give.
I just curious. Thank you. Thank you, Mr. Chairman.
Okay.
Thank you.
Okay, thank you, Prof. Ananto, and thank you, Ms. Amalia, for your presentation. So we will move to the next presenter. We will have Mr. Eko Subowo. He will present their research with title Sedimentary Facies of Cimanuk River Delta on North Java coastal area and their geotechnical engineering properties. So, Mr. Eko, you can start with share your screen.
Okay, thank you, Mr. Moderator.
Yeah, your time is 10 minutes for presentation.
Yeah. See? Okay?
Yeah, okay now.
Yeah.
Please, you can start.
Yeah, thank you, Moderator. Good afternoon, ladies and gentlemen. The afternoon we would to present our research in sedimentary facies of the Cimanuk River Delta on the Java coastal area and its geotechnical properties. We are from Research Center for Geological Disaster, National Research and Innovation Agency, Bandung, Indonesia. We are the outline and one introduction, objective of study, material and method, research and discussion, and next finish conclusion. The delta area is dynamic environment that can evolve rapid in geological young in tectonic, so that from Sunda and Sahul in Indonesia. The distribution of the deltaic area at Sunda Shelf with spread in Sumatra, Kalimantan, and Java. The study its location North Java coastal in Cimanuk, Indramayu. Next, the triple delta form includes the tree and wave and tide and river. The delta North Java and one and Growth and Bengawan Solo and Cimanuk number 2 Growth deltaic. It's here model and deltaic and development in area. Next, the coastal accretion, the Cimanuk Delta was very active. It has progressed seaward for sea and land, maybe 30 kilometers. And but and oncosono interpretation and from accretion and south and to north development and 1, 2, 3, 4, 5, 6. The objective of study reconstruction, the study facies and geotechnical properties of the Cimanuk and distribution CP2 site. Each sediment facies association has distinct geotechnical properties and influenced by sedimentation process. Material and method. The The location study is area Indramayu in geotechnical borehole for 1, 2, 3, 4, and CPTU con penetration 10 location and CPT its location. And in geological Indramayu 1 Kaliwungu formation of the clay conglomerate and lens, and 2 Citalang formation conglomerate breccia and sandstone, and after that the young volcanic product sedimentary rock and conglomerate tufaceous sandstone and tuff, and successfully deposit was floodplain deposit, pit slate and pit deposit, and alluvium river. We conducted investigation drilling for geological depth 2445 and CPT test 10 and 4-5 meter. CPTU might be an CPTU method pushing metal on the ground continue using metal rod of the 1 meter interval. The reading the cone tip resistance kise and the friction and pore water pressure install record by computer and computer or laptop. The measure parameter indicated the subsurface stratigraphy and the engineering behavior like the stiffness and density. Illustration and research and borehole and CPTU and might be stratigraphy. Geotechnical borehole can from borehole and CPT subsurface stratigraphy obtained, we can see from the depth 1, 5 of the lithology, clay, silt, and organic soil. The SPT value 2 and 5 indicated the soft consistency of the soil. The measuring parameter from CPT also show some pattern, low kise and relative low friction. From the depth 5, 11 meter sand with the SPT 9 to 11. Here kise and friction indicated sand loss to rather compact density. From the depth 11, 20, silt with SPT 7 and 9. Higher kise and friction. The kise friction pattern typical of the sand from 0 and 30 meter. We can see the kise friction pattern typical from X, clay, silt, and sand. When we have spatial distribution of the borehole and CPT data, we can correlation the subsurface data to understand the vertical and horizontal. The sediment facies developing and corresponding engineering properties. Result and discussion. Lithostratigraphy and distribution facies sediment and divisional and not short to not geological subsurface and alluvial fan and pitch rate and pro delta and pitch rate development at 1, 2, 3, 4, 5, 6 and strike divisional dominant pro delta and pitch rate and pitch rate deposit, tidal deposit and its borehole and CPTU point. Sedimentation process and geotechnic properties and one location depth, process association, delta plane, fluvial channel plan and fluvial channel and delta plane and pitch rate sand, pro delta and material, silt and sand, silt, sand and sediment feature, sub-base, top, place, fan, and will sorted medium and dominant clay, pro-delta, dominant clay, and value and SPT, variety, value kilopascal, and consistent compaction and hard, low density, very low and classification on material and facies distribution. Next, model, we can see the three-dimensional model of the Shimano. We can see that the dominant facies.
I'm sorry, your time is up. Maybe you can go through the conclusion very fast.
Yeah.
Okay. Thank you.
Yeah. Deep decisional and strike decisional and model Shimano area. Next conclusion. One, integration of borehole and CP2 data up depth of 40 meter in Shimano delta plane and Indramayu West Association delta plane, fluvial and floodplain and delta front, feature and pro delta. Two, the vertical lateral distribution and thickness clear show the sediment facies model and its corresponding geological properties. This information is important for further development in this area and for the further study of earth science. Thank you. Thank you.
Yeah. Okay. Thank you, Mr. Eko, for your presentation. Actually, you use your 2 minutes for your question and answer. Okay. There are any questions in the chat box? Okay. Mr. Nurcayo, please give the question for very quick one.
Okay, thank you, Mr. Zain, as moderator for the time and discussion. I appreciate Pak Eko Bowo presentation and I think the completely data, but when I would like a precautious about your the section stratigraphy. Oh, section lithography. Yeah, about the when I'm not wrong, I saw your sections more deeper. The SPT is more lower. Am I correct? So yes, the 11th slide, I think. Yeah. Okay. Before one. Yeah.
Yeah. Thank you.
The table properties.
Oh yeah.
Okay. on the screen, we can see clearly about the SPT is more lower than the upper part. Meanwhile, the material, I think the clay or sheet is from the pro data material, generally clay is too soft, very soft. So can you explain us about the characteristic date? That was, and the second questions, how do you model about this sediment facies on the Mandiri on the general section? Thank you.
Okay, Pak Mr. Eko, you can answer very fast, maybe.
Yeah, thank you. Question number one and
the SPT value.
SPT value and 0 and 20 and very soft and hard, and here maybe hard and material and massive from massive.
Yeah.
Number one, yeah.
But the delta from organic wood cell and then silt sand in the upper part is more harder, more higher, I mean.
Yeah. Maybe sorry and hard. No, yeah. Yeah, pro delta and very soft.
Yeah. Normally, correct. And when normally when we have the hard material, the SPT value is more higher.
Yeah.
Am I right?
Yeah. Thank you. Number two, and model and stratigraphy and maybe an association and facies sediment, and from it point CPTU and borehole and raw data and rekonstruksi model and from data CPTU from data borehole.
Okay.
Yeah.
Okay, thank you, Mr. Eko and Mr. Nurcaya for discussion. We will move to the next presenter. Okay, Mr. Eko, maybe you can stop your share screen.
Okay. Thank you, moderator.
Thank you,
Pak Zain.
Yes, we will move to the next presenter. Ms. Alode will present their research with title A Preliminary Magnetic Anomaly Study of the Newer Kirk Simon Banda Sea, Indonesia. Ms. Alode, the screen is yours. You can start to share your screen.
Okay, thank you very much, Mas Zain. Very nice meeting you, although it's online. And very nice meeting everyone in here. I would share my screen. Okay, hopefully you can see my screen, Mas Zain.
Yes, ready now.
Yeah, okay.
Okay.
So please let me introduce myself. My name is Gabriela Aludia, or you can call me Gabi. I'm part of the Hydrography Research Group at Institut Teknologi Bandung. And I would like to present a preliminary magnetic anomaly study of the newer Kirk Seamount at Banda Sea, Indonesia. And I would like to let you know that this expedition is actually initiated by Pusidrosal, or the Indonesian Navy's Hydro-Oceanographic Center. So we did the survey from June to July 2022. So we would like to thank Pusidrosal, of course, for giving us the chance to take part in this really, really cool expedition. And this is only a part of expedition. I think there will be some people who are presenting their own researches as well today. So this is the study area. As I've told you before, we did our survey at Banda Sea in here. And as you can see in this map, the Banda Arc represents the transition from the oceanic plate subduction to continental plate collision events, which results in a more complex deformation zone if we compare to what happened in the Sunda Java Trench. So you can compare, there are two zones in here. We've got the Flores zone, which is described as active because we've got a lot of volcanic and tectonic activities in the area, particularly at medium depth. But in the wetter zone, we've got tectonic and volcanic activity at the shallow and deep area, but not many at the medium depth. So the wetter zone is described, at least for now, as a non-active zone. And our study area is actually located at the wetter zone. And this is how we acquisition the data. So of course, before we design our magnetic survey lines, we need to do the multi-beam data acquisition first. And I think the multi-beam data acquisition will be presented by Pahendra after me, so stay tuned. So this is how the seamount looks like. Yeah, so it's a very unique seamount. It's I think it's is south. Sorry, is southeast? Yeah, sorry, west southwest and east northeast striking. Yeah, we've got a southern fault in here, so there's a normal fault seemingly at the southern part of the seamount, and along with the Newark seamount, we've also discovered the eastern ridge east of the seamount, and then we've got two undiscovered seamount. We call it Seamount A and Seamount B for now, and the Emperor of China seamount. And you can see that the Emperor of China seamount has a dextral strike slip fault in there. And it's very unique because the Newark seamount has two peaks, and the Seamount A also seemingly have two peaks. So our survey lines were designed based on the morphology of the seamount. So the shallower it is, of course, the lines would be closer. The further, the deeper the area, you can have, an interval that is further from each other. And the survey were run at 7 to 8 knots on average. We used Geometrics G882, also owned by the KRI Regal 933, which is the Indonesian Navy's Pusat Drasal. Yeah. Unfortunately, due to the unfavorable weather condition and limited survey time, we didn't really manage to obtain all the data that we wished. So the data that were surveyed are marked in red, and the planned one are marked in dashed lines. And this is the results. This is the gridded magnetic anomaly data. It's already very interesting because it's quite, the contrast is quite high. Yeah, the lowest one is minus 180 nanotesla, and the highest one is positive 120 nanotesla. And based on the data that we have obtained, we divided the results into three areas: the Newark seamount area, the southern fault area, and the southwestern seamounts area. I know that one line for each seamount is not enough, but at least we've got something from the field, and it's very interesting. And of course, before we do any interpretation, because this is quite close to the equator, we carried out a reduction to equator operator with this inclination and declination to shift the phase of the magnetic anomaly towards the equator. So this is how the Newark seamount looks like. Before we interpret the data, we need to see that there are areas with no data. So that's those are the areas that are over extrapolated. So we really need to be careful when we are interpreting those areas. We've got areas with no data as well. So we're not really sure if the data at the south here is accurate or not, but at least we know which area is covered with good data. Like for example, this one is good. This one is actually quite good. Yeah. And we can have these lines as a guide at least. Yeah. So after we reduce the data into equator, it's very interesting because we can find at least two areas with low magnetic signature. And of course, in the equator, if we've got low magnetic signature, it means that there are magnetic flow. But unfortunately, because of the bad weather, unfavorable weather condition, we could not do any rock sampling. So we cannot we cannot really define if this magnetic flow is still fresh or is already ancient. So, but at least we know that, okay, this might be the magnetic flow area, the main one. This might be a secondary one because there's a secondary peak as well in there. And it's interesting to see that here at the ridge, we didn't see any magnetic anomaly. It could be because of maybe there's no magnetic rocks in there, but it could also be because of the striking, because the object is striking north-south, and north-south striking object near the equator is very hard to see with magnetic data. And one interesting thing also is we still have some leveling error because I haven't included the cross line in here because apparently there's quite a lot of leveling issues, which we will take a look further when we actually do, our compiled research. And this is how it looks like in the southern fault. Again, there's quite a lot of data gap, but at least we can see something. There's an extreme anomaly contrast from the fault, from the hanging wall of the fault and the foot wall. And it's actually a bit further north from the actual fault in here, but it's interesting to see that the discontinuity is, it has the same or similar striking. So it's striking east northeast and west southwest, just like the fault. And in the southwestern seamounts, because the only thing that we can see in the bathymetry clearly is this dextral strike slipping fault. We want to see if that dextral strike slipping fault is seen by the magnetic data as well, and apparently we can see that. So based on what we see on the morphology and in the magnetic, we say for now, at least, they might be strike slip faults as well in the seamount A and seamount B. So the conclusions are, the first one, in the New Yorker seamount area, there might be a fresh or ancient magmatic flows. But unfortunately, of course, because we didn't do any more geophysical data acquisition and geological sampling, we cannot really validate this interpretation. We cannot say if it is fresh, if it's already dead, even though I know that some people said that New Yorker is already vanished since 7 million years ago or something like that. But because we didn't have any sample, we cannot really say anything for now. But we know where it is. And then no magnetic anomaly at the eastern ridge. It could be because of no presence of rocks, but again, we cannot validate it because we didn't have any rock for now. But it could also be because of the nature of magnetic anomaly survey in the equator. And then we can see an extreme magnetic anomaly contrast at the southern fault and sharp discontinuities at the southwestern seamounts. I think that's all from me. Again, I would like to thank everyone who has been involved in this research. Thank you very much. And if you've got any question, I would be happy to answer it. Back to you, Mastin.
Thank you, Mbak Gaby, for your very interesting presentation. We have about 3 minutes for question and answer. Actually, we have one from Prof. Hananto, the question, and maybe you answered already. Is there any
I answered it already.
Yes. So, Prof. Hananto, so
already answered. Okay.
Yes, there is no.
Yeah. So if you want to go back there and get some samples, it will be very interesting, Prof.
Yes. So
yeah.
How about the next plan?
Yeah, so because this is initiated by Pusi Drosal, I think the head of the event is here also. Pak Anom is here. So I would just like to say, because we were using Pusi Drosal's vessel, which is quite small, a small survey vessel. It's only like 60 meters long. So it is not safe for the survey vessel to sample any rocks with dredging. So because it's really, really light and it's made of aluminum. So if you want to sample them, because, I'm more interested in rocks instead of like sedimentary rocks, because that's the only thing I know as a marine geoscientist. If you want to, get rocks, of course, you need dredging. So maybe, next we can validate the data with dredging. Because I want to see some rocks as well. I think I'm not sure about how the sedimentary facies could say anything more about the volcanic, but yeah, it would be interesting to have some rocks from there. I would have to say that. Yeah.
Same with all of us, Deja.
Yes.
So, Mr. Nur Cahyo, you raise your hand. You can ask.
Okay. The question. Thank you, Mr. Zen. This nice presentation, Mr. Gabriel. But what do you think when we compare about the geophysical regional of the Pittsburgh 2015? How do you think this opening of the basin according to the magnetic lineation is more older or more elder or younger than compare with North Banda Basin?
Yeah, very interesting question, Pak. That's actually what we were hoping as well. When we were doing the magnetic survey, we were hoping to have at least like magnetic lineaments, so we can trace the ages of the lithosphere in there. But apparently, as you can see from the data, we didn't really see any magnetic stripes, so we cannot really, guess the age of the lithosphere in this area. So maybe if dredging is too complicated, because I know that we need a spatial kind of vessel, a bigger vessel like Barunajaya or something to actually dredge it. Maybe a regional magnetic survey would be interesting in there as well. But yeah, we needed to choose when we were doing the survey. Do we want to do a regional magnetic survey or a specific one? And after discussing, we were saying that maybe we just do a specific one, at the volcano. But if you want to see, how old the lithosphere in this area is, maybe we can, make a regional lines, especially crossing this area. So maybe I can just annotate something like that. Would be interesting because we've got some data, some old data in the I think, yeah, it's Hinchberger. Yeah, it's Hinchberger. Yeah. So if we could survey something like that, it would be interesting to see, the depth, the depth spreading reach in that area would be interesting, of course. But yeah, we didn't really have time and we need to choose. But and yeah, from the data, we didn't see any magnetic stripes in here for now. I think that's my answer.
Yes, thank you. Actually, the time is up, but one question from Mr. Punao, but I think you already answered. Is it active volcano or ancient?
Yeah.
So you're not sure actually.
Yeah, it's answered by Professor Hananto already. Thank you, Pak. Yeah.
So thank you, Ms. Alde, for very interesting presentation. So hope we will have the one research to detail everything together. Okay, thank you. Okay, we will move to the next presenter. The next one will be. Okay, sorry. The next one will be Mr. Hendra. It mentioned already from the previous one. He will talk about new work again, Emperor of China, Simons, by using a multi-beam seafloor imagery analysis. Okay, Mr. Hendra, you already here. You can start the presentation. Your time is about 10 minutes and for presentation and 3 minutes for question and answer. Please.
Okay, Bismillah. Thank you for the time, Pak Zain. How's everything going? Long time no see. Last time we are on board together. And also today,
I'm sorry, maybe you can put in the slideshow first.
Okay, sorry.
Your presentation. Okay, thank you.
Okay, so can you hear my voice clearly?
Yes, clearly. You can continue.
Yep. So today I would like to greet some colleagues here. I see Prof. Hananto here. So, Bu Gaby, how are you? Pak Nur Cahyo and also I see Pak Anom as well here. Pak Anom from Puswijrosal. So thank you for the time. Thank you for Research Center for Deep Sea for organizing this incredible conference here. So today, I would like, on behalf of my team, I would like to present our study regarding new worker, Emperor China Simons. Banda Sea, a multi-beam seafloor imagery analysis. So as Bu Gaby mentioned previously, actually, this data coming from the same survey, Jalak Citra 2 expedition organized by Positivo this year. But also, of course, this is the different data. So the background would be because we know that the study area would be the New Worker and the Emperor China Simons, which is located. So we would like to study more about the Simons because we found that there are still limited research or papers regarding about Simons. And because it also could bring several advantages and also disasters, so I think we believe that we need a comprehensive study, for example, by using the morphological mapping and by using, for example, what Bu Gaby did, examination of the magnetic analysis of the Simons. And in terms of the morphological mapping, of course, we know that the multi-beam echo sounder would be the very famous technology for this activity because it could create a dense digital elevation of the bathymetry, and also it could create the backscatter mosaic. So, and then for the DM itself, it could be derived into several morphometric features which could be used for characterize the shape, size, and morphology of the Simons. So for the revision, our objective would be first, we try to present the DM of the New Worker and Emperor China Simons, which we believe that this is the foremost DM obtained so far by Positivo TNI AL Hydrographic Office in Indonesia. And also we try to analyze 3 morphometric features: slope, profile, curvature, and aspect, to determine the shape, dimension, characteristic of the Simons. And finally, we try to obtain the statistical relationship between each morphometric feature, because we believe that this relationship would be useful for the classification of this morphology. As Bu Gaby mentioned previously, so the location is the New Worker and Emperor China Simons at approximately 150 kilometers southeast of Wakatobi National Park. So geologically speaking, this Simons also part of the Bada Arc zone. And for oceanographic point of view, this location is also dominated by water flows from Pacific Ocean tides and also influenced by the monsoon. And this is the vessel we use to obtain the data, RV Regal 93. So the survey was carried out from 60 June to 20 July this year during Jalak Citra 2 expedition Bandasi. And thank you for Positivo being held this organized this expedition, because by using this vessel, which also equipped with the deep water multi-beam system EM 302, we could obtain the raw data of multi-beam, which after that we process the data using Caris HIPS 11.4 software, belongs to Positivo as well, which consists of several stages, for example, correction of the SVP and also cleaning the outlier, the data outlier. And finally, we obtained the DEM with the resolution of 50 meters. After we obtained the DEM, we tried to derive 3 morphometric features: slope, profile, curvature, and aspect, using benthic trend modelers tools. So this is an ArcGIS spatial analysis extension, which could be installed inside the ArcGIS. And for the first feature, the slope feature calculated in degree, we used 3 by 3 moving window of grid cells, which created new image or layer from the TEM. So each of the value of this raster pixel would be calculated using this formula, and and the imagery would showing and could identify the flatten of the terrain, which the lower slope indicate flatter terrain, while the higher value of slope could be could detect the steeper terrain. So in this our study, we try to threshold the slope using 10 degree of value to delineate the Simon boundary. And the second feature was aspect feature, which showing surface direction calculated from 0 to 359 degree, measured from the north direction and increase in the clockwise direction. And the third feature was curvature. This is a second derivative of the bathymetric. So this layer was derived from the slope layer by using the same 3x3 moving window. And we choose profile curvature in this case because this feature could depict acceleration or deceleration of the flow. For instance, detect the landslide or maybe along slope ridge and crest. And finally, we carry out the feature selection to get the suitable predictor for morphology classification later on. So we use regression analysis to get these predictors. And because we found not only one Simon, so we try to use New York Simon as the sample. So first we created 200 random points inside the Simon, and then we extracted 3 value of the features based on these random points. And finally, we try to do regression analysis based on those points. And here is the result of the processing. So we can see here, we found in total 5 Simons, as we mentioned previously as well, because there are 3 unnamed Simons. So we try to name these Simons by Simons A, B, and C. So sorry, I will. Okay, so with the biggest one is the New York Simon, which stretches 80 kilometers in the northeast and southwest direction, and the peak itself reached only 350 meters below the sea surface. So we can imagine how big this Simon is compared with other Simons. And we also noticed that both Newark and Emperor Chinese Simon have elongate shape summit, while other Simons show conical shapes. And so the Newark Simons are also showing a complex structural. We noticed that the flank of Simon showing an erosional process. And also we found ridge features at east side of the Simons in the north and south direction. And also, we also found the structure of lineament at the south of the Simons. And interestingly, using the aspect feature, we could detect a scrap inside the red box, as we can see together, which characterized with an anomalous aspect value, which we cannot be seen from the DEM. And also, at the west side summit of the Simon A, we also found sediment movement there, and indicated with anomaly trend of aspect. And then for the Emperor China, we found that there is an incision of the summit with the direction of northwest and southeast. And also we found a small bulge in the southwest of the mountain, interpreted as an erosional process of that Simons. And lastly, for the Simon Sea, we found a longitudinal feature at the north side of the Simons, and we interpreted submarine landslide process. And also, we obtained that this Simons indicate the flat elongated summit with the peak has a dimension 2 kilometers of length with in the west and east direction. And also we found that there was a deposit material exists there at the east side of the Simon. And result of the analysis showing that all of the features are weakly correlated each other, indicated with the very small value, R2 value, S-square value, only 0 until 0.3 for the profile, curvature, and aspect features. The R-square value only showing 0.01. And for the slope and profile curvature features, it's only showing very small value, 0.0013. And finally, the slope and aspect features are also showing the same small value, almost near 0 with 0.0015. So meaning that all of the features could be used or could be very good predictors for the classification. To conclude, so based on this Jalat Citra 2 expedition this year, we could present high resolution DEM, which could be used for other studies. And from the DEM, we also already created 3 morphometric features: slope, profile, curvature, and aspect. And then we could analyze those features to obtain the shape, dimension, and characteristic of the Simon. And also we noticed that both of the New Yorker and China Simons have elongated summits, while the other Simons showing conical summits. And I believe that the other data, for example, BATNAS data or JEPU data, cannot depict these features in details. So I believe that also that this data could be used in the future study, for example, for toponymy purpose, and also the feature selection process also indicate that all of the features are weakly correlated and could be used for the classification. And for the future, we will try to investigate the use of semi-automatic classification method, for instance, using machine learning techniques to do the classification, the morphological classification, and also we will try to process the backscatter data to obtain the backscatter mosaic to get the distribution of seabed hardness of the Simons. So we will acknowledge first, of course, we thank to Indonesian Navy's Hydroceanographic Center Citrusal for organizing the Jalat Citra 2 expedition this year, and also we thank to Chief of Staff of the Indonesian Navy Citrusal TNI AL, and also we would like to thank all of the crew and commander of RV Regal 933, and we thank all participating scientists who already joined this expedition this year. For those who want to obtain the data, please contact the Citrusal using that email directly. Thank you, Pak Zain.
Yeah, thank you, Mr. Hendra, for your very interesting presentation. Actually, we have only 1 minute actually, but I hope you can answer the question in the chat box. And from Prof. Hananto, he asked about the shape of the mountain. Actually, the big one is not circular, and the small one is circular. How to explain that one? Yeah, like the Simon A and B still have their volcanic cone, while the others not.
I believe that this interpretation would be different between each other. Yeah, so what Prof. Ananto said, I think is interesting as well. So we could interpret later on and in more detail to get the shape and size of the Simons. I think because I believe that this is like visual interpretation. Yeah, and I believe that the result will be also different each other.
Okay, and very first one for the two questions from the Hannah. The first one, what is the novelty of this research? And the second, how this method could support identifying the geological processes in relation with the geological disaster.
Okay, thank you, Bu Hana. First, for the novelty, I think we already find that several references of papers regarding to this study, and we found that for the land area metric analysis of the Simon itself, there are already used in studies, but I believe that in this submarine region of the Simon, I think the study using morphometric features analysis still especially in Indonesia. So it related with the question number 2, it's proven in the study as well. So we already detect some of features in the Newark Simons. For instance, using the aspect feature, we could detect there is landslide footprint there, which can be seen from the DEM. I think that's one of the examples that this method could support for the geological disaster study, Wazin.
Okay, thank you very much, Mr. Hendra. Unfortunately, due to the time, so we couldn't accommodate
no worries, thank you.
from the Mr. Gumilang Ramadan. Maybe next time. So thank you very much, Mr. Hendra, for your interesting presentation. We will move to the next one. So the next presenter will be, Prof. Hananto Kurnia. So he will present their research with title Characteristic of Landslide Sediment of Eastern Indonesia with study case of Kai Archipelago and the surroundings. So Prof. Hananto, could you please share your screen? Maybe Mr. Hendra, you can just stop.
Okay, thank you, Mr. Chairman.
Your time is about 10 minutes for the presentation, so we will accommodate 3 minutes if there is
5%, 13 minutes, and then no question.
Yeah, 10 minutes. Sorry.
Sorry,
okay. Sorry. I will share, but please
stop share. Okay.
Yeah, thank you.
This one. This one. Okay, everybody can see my PowerPoint?
Yeah, just put in the slideshow, Prof.
Okay, okay. I sorry. Just a few. Okay. Show, okay. From okay. Okay, thank you, Mr. Chairman. Good afternoon, ladies and gentlemen. This afternoon, I would like to present my paper. The title is the Characteristic of Submarine Landslide Sediment in Eastern Indonesia with study case in Kai Archipelago waters and surrounding area. The outline first, I would like to give introduction, and then regional geology, methods, result, discussion, and conclusion. Introduction. The interaction of 3 megaplates in the Australia, Eurasian, and Pacific plates, that's what happened actually in the Eastern Indonesia. It's also has a potential to seafloor infrastructure, because some company try to build seafloor infrastructure, something like for the internet, for the transfer of energy. So we must take care of that, how the geological hazard will influence the infrastructure. Also, the submarine landslide can be generated actually by volcanic eruption. There are already happened in the Indonesia. That this volcanic eruption also produce tsunami. In Makian, Maluku, and Sangihe, Sulawesi, Teon and Nila in Banda Sea, and Iliwerung Lembata, Eastern Sunda Island. Submarine slumping is also triggered by the 7.5 magnitude earthquake that generated tsunami that destroyed Banda Island in 1852. This is the tectonic of East Indonesia. That's 3 megaplates interact here. We see in this part is the Australian plate that move up northeast, about 75 millimeter per year, and then it's confronted by the Pacific plate that move. Sorry, I think back. Pacific plate that move to the west southwest about 110 millimeter per year. That's and the west side is ASEAN plate. So because of the interaction of these 3 megaplates, that's why Indonesia is very complicated, the geological condition. I show you the plan of sea floor cables in the Asia Pacific. There's some also include the Indonesia. I just show you that that's very important, because in this era of millennial, we need to connect with its other part in the world by internet. Some of internet is connected actually by seafloor cables. So to avoid the submarine cable being destroyed, we must take care about the geological condition of the active areas. The active undersea rift zone must be avoided, and then fault zone, and as well areas that are prone to subsea landslide. That's according to Huston to 2022. Volcanic tsunami that will produce hazard, especially to the coastal area. We can see here six types underwater explosion. If we can see the underwater explosion, that's destroy the water column, and then the water column will move to the flank of the volcanic and reach the coastal area. And then the blast also the power of the blast will destroy will influence this water column, and then will also produce tsunami. And then the pyroclastic flows from the eruption come to the sea, and then also will produce tsunami. Also this one cardiac collapse because the collapse will influence the sea surface, and then will also produce tsunami. And then also the subaerial failure that come down in the sea, something like in the Krakatoa into 2018, is also will produce tsunami. It is very dangerous, particularly in the very restricted area, something like the Sunda Strait. Kemudian and then submarine failure also can produce tsunami. In the historical time, it's already recorded. We saw here in the North Sulawesi volcanic belt. It's very risk to the volcanic tsunami, as well as this Halmahera. Also in the Sunda Island, this one in Sunda Strait. It's the run-ups from 008 until 40 something, I think 48. It's very high. Also, this submarine slumping that generated tsunami in 1852 Banda in the this we can explain a little bit, especially in this in the square. Some islands are formed there, and then in the Banda Neira itself, the run ice is quite high here. So the bar here compare with the Ambon is very not compare with the Banda Neira, this much less than Banda Neira actually. A submarine landslide is defined as a sediment transport process from continental shelf into the deep ocean, which occurs on continental margins, various geological condition, and water depths. Actually, global sediment fluxes to the oceans, some take place through the submarine landslide process. And then landslide occurs when gravity exceeds the resistant stress of the seafloor material itself. movement can occur on flat or hollow surface and on lower slope due to larger sediment accumulation. Also, the submarine landslide can occur deep as low as 1 degree. This represents the high pore pressure. Sorry. Process involved in the submarine landslide are release mechanism, disintegration, and water mixing of the masses involved. Actually, strong earthquakes tend to produce smaller landslides. Strong earthquake, submarine landslide in subduction zone does not generate tsunami due to their small scale. Submarine landslide tend to occur in predominantly fine-grained sediment because clay-rich sediment tend to travel longer due to its compact condition compared with the less cohesive sand-rich tend to flow at short distances. I show you this seismic record from acquired by research vessel Gimarin 3 belongs to Ministry of Energy and Mineral Resources. So we can see here many landslide type, and at the bottom is actually a sediment field. This occur in Seram Trough, north of Seram, and some deposit, and then the blocky side. Blocky side is very we must aware about this one because it has the possibility of giant landslide. The regional geology of the Kai archipelago. The Kai actually is very interesting because it is located in the apex of bending between Sunda Arc and Seram Trough. Yeah. So from the northward subduction change to southward direction due to the collision between Australian plate and the Pacific Ocean. Also, show you the Kai Island terrain that this one, the biggest island and the Kai Island. The morphology is actually northeast to southwest direction. At the east side, it can be observed as facing cliffs several meters high. These cliffs are interpreted as recently active normal fault downstream to Aru Trough in the east. So actually, it's very active area. So the landslide always occur until now. Analysis of the normal fault, it affects not only Pliocene but quaternary rocks. Kemudian, and then seismicity greater than 7 in Indonesia. This show all the seismicity greater than 7, and the Kai itself. We have also the 7 magnitude. Yeah, so the source from Hutchins and Money. And in detail, the earthquake greater than 10 occurred at 70 kilometers in the depth. Yeah. And then based on focal mechanism is generated by a normal fault. Tectonic activities in Aru Trough are associated with rollback of the banded slab. Jadi, so this actually the driving process, the tectonic actually occurred there. The slab rollback possibly generated the 1882 Banda tsunami. The method. Actually, we use the method through literature studies of published papers by companies, and then the Marine Geological Institute of Indonesia and other sources. Identification of sediment involved in submarine landslide was achieved through seafloor sediment sampling and grain size analysis by Marine Geological Institute. And historical submarine landslide in Indonesia were also obtained through a work of literature study. Result from the study, we identified a large mass transport complex, or we call it the MTC. It distributed in Tanimbar Trough, south of Kai Island, and terminated at the transition zone between Tanimbar Trough and Aru Trough. MTC was derived from the Seram Fault and Thrust Belt, or FTB, at Kai Island. represented by Kai Arch, that collapse downslope towards the northeast to the pre-existing deep water of Tanimbar and Aru Trough. Large slump in eastern wall of Tanimbar deep seem capable to generate tsunami, and eastern coast of Kai Island is observed recently from collapsed parts that deposit to the east in Aru Trough, as I showed before. This is the multi-beam image, and then this multi-beam, and then this the interpretation. We see here the MTC widely distributed in the south of Kai Island, and we also have some seismic report here, A, B, C. The MTC is partly above the Tanimbar Trough, so that's why Tanimbar Trough become narrow, and then Aru Trough, so mostly in the Tanimbar Trough. Now we see the seismic report. This is the MTC, this northern front. We saw that the inside of the MTC it's chaotic. It's possibly derived from the activity that it's not. Usually we saw this seismic is supposed for the hydrocarbons, for example. They show the layer, but this is not so the layer. And then this one, the B section is down through this normal fault, and then this one is the C is eastern boundary of the MTC. So based on grain size analysis of the aqueous sediment at Aru Trough and South Kai Besar Island, MTC was characterized by a mix-up of gravel, sand, and silt and clay, with percentage between as shown here, and then sea depth ranging of the submarine set occurred between 200 and 700 meters, which spread offshore at the south, east, and north of Kai Archipelago. Internal quake that triggers submarine landslides relate to seismicity in Banda Sea subduction zone. This is the sample we take by the geochemistry. This one in the Aru Trough, and then these two in the south of the Kai Islands. The sample I showed before that it showed the mix of the gravel, sand, clay, and silt. It's indication of a submarine material.
Okay, excuse me, Prof. Ananto.
Okay.
Your time is up. Maybe you can just go through the conclusion.
Okay, conclusion. Thank you. The characteristics of submarine landsat are recognized from multi-beam image and identified seafloor sediment samples, and the image shows slumps and landslide sediment either in Weber Deep and Aru Trough, while multi-channel seismic profile identified the mass transport complex in the south of Kai Island. The landslide sediment were characterized by a mix-up of gravel, sand, silt, and clay fraction. The seafloor where the landslide sediment were deposited is a slope with sea depth ranging from 200 up to 700 meters and spread offshore east and south of Kai Archipelago, and any plan of seafloor infrastructure construction passing through this potential hazard areas require research to reduce its impact. Thank you for the time and for the
Okay, thank you very much, Prof. Ananto, for your interesting presentation. So actually we need something like this for the hazard mitigation. So due to the time, so we couldn't accommodate the question and answer. So maybe they will ask directly to you through the email or through the WA. Thank you very much, Prof. Ananto.
Okay, thank you.
We will move to the next presenter. It will be Mr. Nur Cahyo. He will present the research with title Quaternary seabed sediment characteristic as an indication of VMS mineral deposit at the South Banda Basin and its surroundings, Indonesia. Mr. Nur Cahyo, you can just start with share your screen. So you have the same time with the others, so 10 minutes for presentation. First, please consider one. Thank you very much.
Okay, just a minute. Okay, so I screen. Oh, okay. Sorry,
Just put
can
in the
you show
slide.
my presentation?
I can see, but please put in the slideshow. Okay, thank you very much. You can start the presentation.
Just a minute. Okay, thank you. Thank you, Mr. Jen, as the moderator for the time. Good afternoon, fellow speaker at the Zoom meeting, as well as other participants who listen via the YouTube channel. On this happy occasion, I on behalf of my college in the mineral deposit research from the Research Center for Geological Resource will present my paper entitled Quaternary Seabed Sediment Characteristic as an Indication of Volcanogenic Massive Sulfide Mineral Deposit at South Banda Basin and at surrounding Indonesia. This paper we write with my colleagues, Mr. Hananto Kurnio, and also Mr. Zen Tuakia, Armin Tampu Bolon, Pardiarto, Bambang Widi, Wahyu Widodo, and Kusnawan on our research group mineral deposit. Okay, my presentation in systematic as follows: introduction, regional geology, research, discussion, and conclusion. So, before I talk about the substance in our paper, let me introduce the introduction of my paper. The study location, which is the topic of discussion of my presentation, is generally at Bandasi, is morphometrically divided into two areas, namely in the North Bandasi on the northwest part from the location area, and then which is the southwest of Maluku and the south of Bandasi, located on the southern part of Ambon City, and then the south Banda Basin located on the southern part of the Ambon. The two basins are separated by the southwest northeast oriented Banda Ridge. The location study is administratively located in Banda District, Central Maluku Regency, Province of Maluku. And as geographically and morphometry, Banda Ridge it's bordered at our location at the west to northwest, Ambon and Seram Island at the northern part, and then inner Banda Art at the eastern part of the location study, and the Vulcanus Island on the south. Vulcanus Island, I mean, this is the Damartion, Nila, and so on. The purpose of our presentations is try to analyze and related morphometric aspect relation to the type of sediment or its characteristic and its distribution, as well as its relation to volcanic hosted massive sulfide or volcanogenic massive sulfide. And when we simple term, the main set in discussing material looks like the following diagram. The first stage to analyze and review various related secondary data, especially from the Marine Geology Institute data, such as morphometry and data and seabed bathymetry, and then we try to redraw the bathymetry data to try to find out the morphology term. And then the second stage, we try to identify of the superficial sediment textural and distribution according to grain size analysis. And finally, on the method, we look the kind of type heavy mineral and distribution. And finally, conclusion, how the volcanic hosted massive sulfide deposit relation to the characteristic on textural sediment distribution. When we saw the right corner on this slide, the present day configuration of Banda Sea, we can see submarine arch and oceanic plateau at the pale blue color, and then a volcanic island arch of fluid and material created along plate margin on the green, and then the Australia Trinity continental crust in the red one. This consisting of water deep and Timor Island. Okay, what about the regional geology? The location study, namely is South Banda Basin or SBP, is the backyard basin that resulted from the interaction of various major plates, such as the Pacific and Indo-Australian plates, as well as several microplates, including the influence of the Papua Transport Fault. I think the former presenter is more detailed discussion about that. And then the important, and I think the relation to Miss Apoldia presenter about the new geological and geophysical data, namely a magnetic anomaly. When we see the red box on that is the location study location, we have the relatively west and east pattern of the magnetic alienation, and when we compare on the referring to Hinsberger 2001, the locations is a part of the South Banda Basin, which has the 6.5 to 3.5 million annual. That's meaning more younger than the North Banda Sea Basin, which has on the south western part from the location study. And then after we have the secondary data study, we have the bathymetric and seabed morphology based on general bathymetric chart of ocean, GEPCO 2022, and then we redraw. We could know the shallowest part of which is the oceanic Banda arch. That's meaning of the eastern from the water deep. Consists of the Tanimbar, Babar, and the water of the eastern part of Seram, which has around 150 to 200 meters deep water. And the deepest part is the water deep, which reach until 7,600 more or less meter of water depth. The distance from the coast of the island to the offshore, this only less than 6 kilometers. This meaning the slope of the island is so steepness. Refer to the profile section when we can see in the corner, in the top left corner, it can be seen that its morphotectonic environment is distinguished by its distinctive morphological appearance. Like example, the inner Banda arch is found in the form of a solitary submarine volcano, and then whose path can be followed from the south to north. I mean, starting from the Wetar, Romang, Damar, and continuing to the north in the form of series quaternary volcanic island, such as Teon, Nila, Serua, Manuk, to the northmost Banda Api, including several underwater volcano. And then the water depth is the depth for at basin more than 7,000 kilometer deep. Sorry, 7,000 meter deep, located in the highly curved part of Banda Aceh and is a deepest point of the Aceh. The water depth, I mean, sorry. Okay. The Banda, sorry, the water depth is characterized by valley geometry informed by tectonic regime, and then in the middle part, the valley wider and deeper. Water depth morphometry is generally composed of turbidite and pelagic sediment and volcanic sediment, with not a wide distribution on the western slope. The second result, we try to about the seafloor sediment characteristic according to NGI data in 1998. We took several samples from by graphite and piston core. We grouped it of the 5 types sediment characteristic according on the mineral deposit. The first is deposit volcanic sediment consisting of muddy sand to sindicate or VU on the center part of the location, and the sand and volcanic gravel. And the second, we have the excuse me. Hello? Test. Okay, the second, the OS deposit is a sedimentary deposit containing foraminifera. The third, on the location study, consists of the turbidite deposit, which consists of fine sediment, silt to sand, and coarse sediment, and pterygian deposit, and also heavy pelagic deposit, consists of from clay, mud, and located on the Seram, which is one of the sedimentary deposit that composed of the morphometry of the outer Banda Aceh, other than OS deposit. And the resume of the result, how the relationship deposit type, morphometry, and seafloor sediment characteristic. We have the type of deposit, volcanic massive sulfide deposit, that tectonic setting controlled by art setting, and the associative with volcanism. There is active inner banda volcanic art such as Damar, Teon, Nila, Serua, Manduk, and Banda Api submarine volcano. And the main types of mineralisasi fluida, there's a magmatic and modified marine water resulted from the chimney on the sea floor. The ranges of temperature about 100 to 500 degrees Celsius degree, and the depth this more and less 5,000 and 6,000. Actually, on the water depth, we have the more than 6,000 meter. And morphometry, there are the 7 morphometry types: internal back art, and then Banda Aceh Simon and Gunung Api Simon, and then Banda Ris, water deep, North Banda Basin, and outer Banda Aceh. Morphometry inner Banda Aceh consisting of the volcanic sediment. And then Banda Aceh Simon consists of volcanic sediment, including sand, volcanic gravel, and pyroxene, and pumice, and turf. And the Banda Ris, this is of the OS deposit, the water depth dominantly of the turbidite deposit, and North Banda Basin composed by the terrigenous deposit. And then the outer Banda Art, they are hemipelagic and deposit. That conclusion, the shallowest part of which in outer Banda Art around the coast of Tanimbar, and the deepest part is in the water depth, which is 7,600 meters of water depth. The seabed lithofacies in Banda Art area were deposited in several morphotectonic environment. The type deposit that are pelagic, hemipelagic, including turbidite and volcanic origin deposit. This important volcanic deposit consists of gravel, sand, muddy, and silt. The composition, the grain composition is volcanic lithic. That mineral by the feldspar, augite, hyperstane, and enstatite, which are commonly found in volcanic art and tuff. And the group of volcanic grain, I think, composed of pyroxene andesite, pumice, and tuffite, or silicious gravel. The pyroxene andesite are present as phenoclase and the felsitic cryptocrystalline ground mass of diephyterite glass, which derive, I think, from Banda volcanic complex and Seram Gorong Island. And the other mineral cores and rock fragment with a minor amount of feldspar, mica, garnet, and pyroxene are component of the sandstone class in the North Banda Art. And volcanic grass characterized by bubble shards. This typically volcanic product from Banda Api and Manuk Serua volcano, whereas the pumice from the Serua and Manuk volcanic complex that still volcanic active still. And the
Mr. Nur Cahyo. Hello, can you hear me? Hello. Yeah. Okay. I think there is problem with Mr. Nur Cahyo. Oh, okay. Okay. Maybe. And then actually, he finished also the times for presentation and question. So we are sorry. We will move to the next presenter. So the next one, it will be Mr. Purnawa Senjaja. He will give the presentation for their research with title Kawio Barat Volcano, Geochemistry of a Submarine Volcano in the Sangye Arc. Please, Mr. Purnawa, you can start with share your screen. Yeah, your time is also 10 minutes for presentation, so please consider about it.
Okay, thank you, Pak Jain, as a chairman in this session. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The honorable ladies and gentlemen of the seminar participant. Because of Professor Hanato talk about disaster, I would like to express of my condolence for the fatality because of the earthquake in Cianjur recently. Today has reached 271 fatality. There's many fatality today. Let us pray for the victim and the families left behind. Maybe Allah has a good plan for all of us from yesterday earthquake, so that we can take lesson and wisdom from this great disaster. Okay, ladies and gentlemen, on behalf of my team, Pak Jeffrey, Pak Iwan, Pak Agus, Pak Didit and Professor Haryadi Permana. I, Purnama Sanjaya from Research Center for Geological Resources, BRIN. It's honor to presenting our research project in this prestigious event, this May 2022. The title of my presentation is Kawayo Barat Volcano, Geochemistry of Submarine Volcano in the Sangihe Arc. This is the outline of my presentation: introduction, data and method, result and discussion, and then conclusion. Kawayo Barat was the target of the 2003 Indonesian Australian Survey of Submarine Hydrothermal Activities Expedition in the Sangihe Archipelago, Sulawesi. This expedition was discover some submarine volcano along the Sangihe Arc, located at the northern of Manado, capital city of Northern Sulawesi province. This submarine volcano is still active and in the Sangihe Arc, with border the Maluku Trench along the east side. This arc is about 550 kilometers long and 70 kilometers wide, consists of 20 islands, and 12 of them are quaternary volcanoes. The Kawayo Barat summit, which is 5,200 meters below the sea, composed by fresh pyroxene basalt lava, which are covered by deep sea sediment rock. Samples were taken from the seabed using dredge during expedition at the location of SD 13, 14, 15, and 16. The results were obtained were rock fragment, volcanic ash, and fresh volcanic rocks such as basalt and andesite. This is a view of the seabed of Kawayo Barat volcano, such as a pillow lava and lava block. We can see hydrothermal activity as a smoker of fluid escape through the surface along the fissure. Okay, and then the result: a total of 18 samples were geochemically analyzed at Cheshire, Australia, using SRF and ICP-NS for major, trace, and rare earth element. Volcanic rock from Kawayo Barat submarine volcano can be identified by major element such as silica oxide 46 until 51%, titanium oxide 0.6 until 0.8, iron oxide 6 until 10%, aluminum oxide for 14 until 18%, sodium oxide 2 until 3%, manganese oxide around 0.18%, phosphorus pentoxide 0.2 until 0.3, and have a relative high potassium oxide 1 until 2%, with ratio of potassium versus sodium more than 0.6. And then magnesium oxide 3 until 6%, with high Mg number more than 50. For trace element, they have a high strontium until 900 ppm, yttrium, niobium, lanthanum, and you can see in the slide. Okay, we plot the result to the Harker diagram for showing magma differentiation during the crystallization. We could see that some elements have negative correlation with the increasing of silica, such as iron, magnesium, calcium, and titanium oxide. And some elements have positive, I mean that negative, and then we have a positive correlation with the increasing of silica, such as aluminum, potassium, and sodium oxide, and also trace element strontium and rhodium have positive correlation by silica. Plotting to the geochemical classification diagram are showing that the volcanic rock from Kawayo Barat are basalt and basaltic andesite. And based on IFM diagram, they are lies on the alkaline magma affinity. Another diagram showing same result. They are alkalic rock high K alkaline and some sample in the region of Sassanid. Where did the volcanic rocks form? Under what the tectonic setting? Based on this discrimination diagram, which help interpret the tectonic setting, according to diagram from Mulan 1987, Korea Barat volcanic rock are calc-alkaline basalt, and oceanic floor calc-alkaline or island arc based on the Peirce and Ken diagram. According to the Peirce 1977 diagram, we can see this volcanic rock occur in the organic and oceanic island based on the ratio of strontium and yttrium with yttrium element from Devan and Drummond 1990 diagram. This volcanic rock are adakite, distinguished by typical arc in general. In the multi-element normalized rare earth element diagram, they display strong positive strontium anomalies with niobium depletion, and depleted heavy rare earth elements suggest that they are typically alkaline and subduction related. According to elevated strontium and high ratio of strontium versus yttrium, this rock have adakite-like geochemical characteristic, which are probably formed by slab melting or interact with the mantle to form the low silica adakite or high IMG number andesite group. Okay, how did this adakite form? From some literature, it is found that adakite can take place in the several tectonic settings such as subduction of the oceanic crust at modern convergence zone, involving young oceanic crust also, shallow angle subduction, collational subduction, post-collational, or tear gap. Moyen 2009 made a sketch of the different way to obtain a high ratio of strontium versus yttrium to form that all diverse rock referred to adakite. Korea Barat adakite are included in the low silica adakite and or an high MG andesite group based on this sketch. So can look the tectonic setting of this area from several literature strength the hypothesis that adakite rock from Korea Barat are the result of the slab melting from Maluku or Sulawesi plates, because there's a collational subduction in there. On the sosonic magma affinities may give the impression that the slab melting of the Maluku plate which are produced adakite, because the location that submarine volcano location in the backup basin away from the trends. Is it my voice is clear?
Yes, it's quite
Hello?
clear. Yes,
Okay.
it's quite clear.
Because Pak Nur Cahyo, my sound is disappear. However, this need to be supported by geophysical research to see the subsurface configuration of subduction oceanic plate. So as a conclusion, volcanic rock from Korea Barat submarine volcano are basalt and basaltic present site. The volcanic rock have geochemical adakite characteristic, high K alkaline and sosonite, which are probably formed by slab melting or interact with the mantle to form the low silica adakite or high MG andesite number. Collational subduction in this area still leaves question, which plate melts? Is it the Maluku or Sulawesi plates? The sosonite magma affinities give the impression that the slab melting of the Maluku plate, which are produced adakite because its location in the backup basin away from the West Maluku trends. Okay, that's all. Thank you, Mr. Chairman. I give you time to you.
Thank you, Mr. Purnama, for your presentation. It's very interesting. So you just finish exactly the 10 minutes. So we have 3 minutes for question and answer. So if anyone want to ask, you can ask directly, and I will allow you. Okay, Mr. Purnama, aside from the geochemistry, so what kind of data that you have now to compile everything, and then you can conclude something?
Actually, this is the old data from Professor Haryadi Permana. So they have many data also from geophysics and then morphology, submarine also. Maybe we will make a comprehensive data for this volcano, submarine volcano. I think today is just presenting the geochemical data from the volcano. Is it clear?
Yeah, it's clear. And as we know, the Kawio is like well known already internationally. So, do you have any plan for the next time for another purposes in that area?
Until now, there's no project in there because there was the old data we digging again and show in the public because the data of geochemistry is not published yet. Okay, I think there is no expedition to that area again in the next year or two. I don't know.
Okay, thank you. Okay, we still have 1 minute. Prof. Hananto, please, you can ask directly.
Mr. Sanjay, very interesting presentation. I just like to know, is there any continual activity for the researchers, because it's also have potential for VMS. Maybe you can propose to the. Yeah, I just want to know. Thank you.
Okay, thank you, Pak Professor Hananto. This is the whole project from with Australian with Professor Haryadi as chairman. So maybe next 2 years, maybe in 2 years ahead, we can propose the activity in that area again. Thank you, Pak Hananto.
Thank you, Mr. Chairman.
Okay, thank you, Prof. Hananto and Mr. Purnama. We finish your presentation now, and then we'll move to the next one. Mr. Arifin will present the research we title The Influence of GMPA Logic 3 on acceleration spectra value in coastal bedrock of the Spesa region of Yogyakarta. Mr. Arifin, you can start your presentation with share your screen. Your time is 10 minutes for presentation, so please consider about it. Yeah, just put in the slideshow and then you can start. Yes, please. Yeah, we can still hear you. Yeah, Mr. Arifin. Maybe you are in the mute. You can unmute your microphone and then you can start the presentation. Yeah, Mr. Arifan. We can still hear your voice.
Hello.
Yeah, it's clear now. Okay, you can start. Yes, your time is 10 minutes, please.
For the opportunity for us to
Yeah, I think there is trouble with your microphone, so we couldn't hear your voice. Maybe you can unplug your microphone and just speak directly. I'm sorry, we couldn't hear your voice.
How about now?
Yeah, now it's clear. Please keep it.
Okay, I'm sorry for the technical problem. Okay, we will give the presentation about the influence of GMPE logic tree on acceleration spectra values in coastal bedrock of the spatial region of Yogyakarta. We are the team consists of myself. My name is Arifan Jasabana and Miss Dwi Sarah, Hori Sugianti, Adrin Tohari, Eko Subowo, Anggun Mayang Sari, Syakira Trisnavia, Mas Somim, Afnindar Fahru Rozi, and Mansur Irsyam. We are the research from Research Center for Geological Disaster in Bandung, come from agency, National Research and Innovation Agency Indonesia. And the other come from Research Center for Data and Information Science, same come from BRIN Indonesia, and the last come from Faculty of Civil and Environmental Engineering ITB Bandung, Indonesia. Okay. Here our outline in this presentation. The first one is about introduction, and then continue with method, then result and discussion, conclusion, and reference. In this slide about introduction, as we can understand that Daerah Istimewa Yogyakarta or Special Region of Yogyakarta is an area that has a diverse topography. Starting from the north side in the form of mountain, also the presence of the Mount Merapi, and then continuing the slope toward the coast in the south. It is blessed with beautiful scenery and fertile land, making the DIY is a rapidly developing area both on the lowland side and the coast. But on another hand, the threat of geological hazard surround this area through many seismic sources. We will show in the next slide. It is necessary to conduct a study that discuss about the safety of the existing infrastructure in this area in terms of the design of earthquake dynamic. To develop the new and the retrofit infrastructure, it is necessary to provide information about the acceleration caused by the earthquake. So we have the objection in this study. The PSHA or probabilistic seismic hazard analysis will be carried out about 1,000 and 2,500 years return period using the help of the OpenQuake software. So what method we use explained by this slide. We as we said before, we use the OpenQuake software. In the first step, we are collecting the earthquake source. We have the data source from the Pusken in 2017. And then we make the logic tree source and GMPE, continued by making the configuration file, and then continue again with the running in server. Why we running in server? Because it is very heavy if we operate in our laptop. It needs the very high specification of the computer. And then after all the process finish, we can download the data and then continue again with the making map using the GIS software like the ArcGIS or the QGIS. In the left side, we saw the GMPE pushgen for the logic tree 1 and the GMPE new for the logic tree 2. The difference in the benioff mechanism. In the new GMPE, we use the BC Hydro 2018 and the Zhao et al. 2006 for IntraSlab. And in the right side, in the bottom, we show you the earthquake data we use to make the analysis in the special region of Yogyakarta. We make the radius 500 kilometers for the boundary. So all of the earthquake source inside the circle radius 500 kilometers, we use it, including the background source and then fault source and then the megathrust source. Here the result and the discussion. In the beginning, we only consider the 8 point which is light in the coastal area. As can be seen in here table, we want to make the relation about logic tree 1 and the logic tree 2 in each of the point. And then from the reviewer we have before this seminar, we make the all data in the Yogyakarta region to make the comparison about the logic tree 1 and the logic tree 2 in its mechanism. For example, in table A, we make the comparison about PGA 2000 and 500 years return period in x-axis. We plot the acceleration using logic tree 1, and for y-axis we plot the acceleration for logic tree 2, and the R square or the determination coefficient, we get the very high value, almost 1. In here in table A, we get the 0.9932, and in graphic 2 we get the R square 0.9957. And the other like similar trend, we get the R square with almost reach the 1. And then beside we plot the graphic, we also make the special plotting in the Yogyakarta region area, which can be seen in this slide. We can see, for example, in the picture with label E and F. Here E and F means spectral acceleration 0.2 second for 2050 year return period for logic tree 1, and for F for logic tree 2. We can see that there are very little different in the coastal area, which show us that if we using the logic tree mode 2, we get the higher, but in small part in the coastal area. This is the last slide of our presentation about conclusion. The first one, the push-gain version of the GMPE logic tree and the update version, which involving GMPE Zhou et al. 2006 and Abrahamson et al. 2018 in the Benioff zone, didn't provide the significant difference. There is increase the acceleration in narrow area for the spectral acceleration 0.2 second in 1000 and 2000 years scenario. The coefficient of determination or R square at the test point saw an excellent value, which is 0.9879. That's show us that the push-gain version of the GMPE is still feasible if we want to analyze this with the other software, which is not include the new GMPE in the PSHA program database. Then the third conclusion is based on the spectral acceleration 0.2 second acceleration map. Building plans with a natural period of 0.2 seconds are required to withstand an acceleration up to 1.5 G. On another hand, for retrofit activities, an acceleration of 1 G is sufficient. Moreover, if it is applied to building with natural period of 1 second for planning design, it is necessary to withstand up to 0.6 G, while only for retrofitting, we only need the withstand with the 0.4 G for the force. I think that's all what we can share to you all. I hope we have the nice discussion. I give back to the chairman. Thank you.
Thank you, Mr. Arifan. We are sorry to misspelling your name in before.
Oh, yes. No worries.
You use about 12 minutes and we have
I'm sorry.
1 minute for discussion.
Okay.
If anyone has question, you can ask directly. Okay. Maybe I have one very short.
Okay.
How about relationship between GMPA and liquefaction? Actually, I'm not really in this field.
Okay. Thank you for the question. The GMPA is the equation to calculate the acceleration in some location. While you want to doing the liquefaction, you need this parameter, which is acceleration, but in the surface area. So the GMPA will be give the another result if you use the many different GMPA.
Okay, thank you very much, Ms. Arifan, and the time is also up, finished, and we will
thank you.
Thank you very much for your time.
Thank you very much. Thank you.
We will move to Ms. Vera. She will present the research with title Grain Size Trend Analysis for Sediment Characteristic of Cirebon Water. Ms. Vera, you can start with share your screen. Okay, for all of you, I just want to remind you that maybe you missed the attendance list. Please fill out it. We need for the certificate purposes. Okay, Ms. Vera.
Can you hear my voice?
Yeah, I can hear clearly. So
Okay.
just put in the slideshow and then start the presentation. Your time for presentation is about 10 minutes. Please consider about it.
Okay, wait. Can you see my presentation?
Yes, I can see clearly.
Okay, thank you. Okay, thank you, Pak Zain. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. My name is Vera Stati Agusta. I'm a researcher from Research Center for Climate and Atmosphere, National Research and Innovation Agency. Currently, I still pursue my master study in ITB program of Earth Science. So this presentation is a part of my research collaboration between ITB and MTCRC Korea. So the title of my presentation is Grain Size Trend Analysis for Sediment Characteristic of Cirebon Waters. And this is the introduction. So Cirebon Waters are an open bay coastal water to the Java Sea, that relatively shallow sea, less than 12 meters, and have a curved and rough coastal configuration. The coastal area in Cirebon water are dominated by silty sand, and several areas are have a mangrove forest. And the Cirebon water are strongly influenced by distribution of river flow, such as Kalijaga, Kedung Pene, Sukalila, Kepetakan, Mundu, Bandengan, Kanci, and Kasunanan. Kemudian, the existence of river effect, influx of sediment material from land to sea, and it occur on the mouth of Kalijaga, Mundu, Bandengan, Kanci, and several part of Kos Kejaksan, Lemah Wungku, Mundu, Astana Japura, Panganan, and Losari. Ocean current also affects the sedimentation distribution. The condition of current speed in Cirebon water, which is relatively weak, with high rainfall intensity, cause the sedimentary material dominated by muddy sand. The ocean current in Cirebon water is influenced by seasonal monsoon, such as northwest monsoon in January and southeast monsoon in August. In estuary area, sand sediment will be significantly found because large particle will be deposited faster, while fine particles such as clay and silt will be carried away by current and waves. The sediment deposition rate on coast of Cirebon water is 0.43 until 1.49 km per square per year. The study of sediment distribution in Cirebon area has been conducted by several previous researchers, such as in the western part of Cirebon waters, and in the coast of Gunung Jati and Mundu. The study was focused on understanding the sediment dispersal in Cirebon waters. And this is the material and methods. Surface sediment deposit in the Cirebon water was sampled using grab sampler installed on the ITB MTCRC research vessel, and about 0.5 kilogram of sediment were taken from 30 sediment sample. The sampling location are shown in the picture, and the survey activity were carried out on November 30 to December 1, 2021 to obtain the sediment sample during northwest monsoon period. After that, the grain size analysis was done at the chemistry laboratory of BRIN using the Master Sizer 2000 particle size analyzer. It is to understand the spatial distribution of sediment. The calculation of grain size parameter, such as mean, sorting, skewness, kurtosis, are conducted using geometric method and visualized by Surfer 11. After that, to understand the sediment transport mechanism, we use diagram CM, and to understand the sediment deposition process, we use diagram Stewart. And this is the result. The surface sediment has been processed and analyzed to determine the spatial distribution of sediment grain size. From analysis, it is known that the sediment deposit in Cirebon water are relatively dominated by silt with range 7.765 to 33.43 micrometer and classified as very coarse silt to fine silt. The spatial distribution showed that the fine grain sediment are deposited along the coast area of Cirebon Regency, while the coarser grain sediment are deposited further into the open ocean. And this is a table that give information of the grain percentage and the sediment types. We can see that the types range from fine silt to very coarse silt. And this is the distribution of grain size parameter. From mean, we can see that the sediment in Cirebon water is in the range of 7.765 to 30.43 micrometer and show the predominance of fine silt to coarse silt. The pattern of distribution of the mean grain value differ from northeast to the southwest. The distribution pattern shown that the distribution of fine silt in the southwest region and slowly turn into coarse silt in the northeastern region of the study area. This condition can indicate the increasing of energy level current from the southwest to the northeast of Cirebon waters. After that, the sorting parameters, it is shown that there's the range from 2.073 to 3.221 micrometer. This condition indicates the sorting of each station is relatively poorly sorted. The poorly sorted value was seen in sample 16, and the fine sorting shown in the sample 26. There are a correlation between sorting and the variation. The poor sorting indicates the variation of grain size in the sample. After that, this is the skewness parameter. From the result, the skewness value is in range minus 0.45 to 0.27 micrometer with the criteria of fine skew to coarse skew. The negative distribution deviation will tend to have coarser grain size with relatively poor sorting. This condition indicates an increase of distribution rate of sediment from the southwest to the northeast part of Cirebon. After that, for the kurtosis parameter is in the range of 0.768 to 1.436 and classified as platykurtic to leptokurtic with domination of mesokurtic. Some region with poor sorting condition correlate with the platykurtic classification as in the northeastern region of study area. After that, this is the result for diagram CM and diagram Stewart. The relation between value of percentile first and median in the CM diagram implies a deposition mechanism which has a close relation with the grain size. From CM diagram, it can be seen that the entire sample was dominated in the pelagic suspension mechanism. This is related to the mechanism of deposition of fine sediment, such as silt that relatively floating in the body, in the water body. The deposition mechanism caused sediment with fine grain to continue to be suspended and transported far away as pelagic sediment. And for the Stewart diagram, can be used to imply the deposition rate by analyzing the relation between sorting and median. Based on the result, it can be seen that the sediment sample were deposited in quiet water condition with a shallow deposition rate, and these conditions are generally correlated with the fine grain sediment with poor sorting. And this is the conclusion. A grain size trend analysis explain that sediment in Cirebon water have a very coarse silt size to find silt. The mean and skewness value indicate that there is an increase in current energy and sediment transportation rate from the southwest to the northeast of Cirebon water. Shorting is relatively poor short, and kurtosis is predominantly mesocurtic type. The sediment deposit mechanism is a pelagic suspension and deposit in condition of quiet water with shallow deposition. And this is my reference that I use. Thank you.
Yeah, thank you, Ms. Vera, for your very interesting presentation. So you used about 11 minutes, so we still have 2 minutes for question and answer.
Okay.
So still I didn't see the question in the chat box, so maybe anyone want to ask, you can ask directly. Just use the raise hand feature. Okay, actually I have one in your map about the grid. How to make it very precise, the grid in the sample point, something like this.
Yes, I use, thank you, Pak Zain. So for visualize the data that I collect before, we try to get the value of each parameter, such as mean, shorting, skewness, and kurtosis. To visualize, we use server 11. And we use we use a triangular linear interpolation algorithm to make the grid.
Oh, okay.
And yeah, and we collect the sample by using because we use a vessel from ITB and MTCRC, so we collect the data by grading the Cirebon waters.
Okay, we still have time to maybe Pak Nur Cahyo will ask, please.
Yeah, thank you, Mr. Zen. Good afternoon, Ms. Vera.
Good afternoon.
Thank you, interesting presentation. How do you took the samples, by graph or gravity coring?
We use a graph sampler.
Oh, graph sampler. Okay.
Yes.
After that, you use the sediment analyzer to find out of the sediment characteristic.
Yes.
Do you make depositional mechanism after your conclusion? I mean, the source of sediment source, and then how you can okay, good. Yeah, okay. This result pelagic suspension and median.
Yes.
Could you relation to the sedimentation? We all know the Cirebon and the other city at the northern Java. They have the the rate sedimentation is high. Do you measure the sedimentation from the sample?
We didn't do the rate sedimentation analysis, but from several previous research, we know that the sedimentation rate in Cirebon is quite higher than the erosion rate. Yeah. But we didn't analyze the sedimentation rate.
Yeah, it's okay. But maybe I could propose for the next your project.
Yes.
Could you use the image interpretation or from the dating carbon or
yes.
for your sedimentation measure?
Yes. Yeah. Thank you. Thank you, Pak.
Yeah.
Thank you, Mr. Nur Cahyo and Ms. Vera for the discussion and the presentation. So we will move to the last presenter, Mr. Ahmad will present the research with title VS30 correlation based on CPT and SPT and data related to Semarang coastal carrying capacity. So, Mr. Ahmad, you can start with share your screen. Yes, please. You can just.
Okay. Is my presentation seen clearly?
Yeah, it's in.
Okay.
So your time is about 10 minutes per presentation.
Okay. We should start. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Thanks for the comment and attendee. First of all, I'd like to introduce myself. My name is Ahmad Farus Somim, and also an honor for my teams mentioned as well for Research Center for Geological Disaster, and then I have a team for Research Center for Geospatial and the Research Center for Oceanography, National Research and Innovation Agency, BRIN. In the presentation on the main topic of marine questions in the room 4, we are going to discuss about the comparison of the S30 based on SPT and SPT and CPTU data, case study of Semarang coastal zone. So I divide this presentation into 4 parts. I conduct on each part. In the first part, we talk about locus and purposes. The next one is about the method we are using. Third, the result set can be carried out and sum up with a conclusion and remarks. Let me start this presentation with information of our study area in Semarang. Semarang is the administrative capital of Central Java province of Indonesia. This area is located in central location of the northern part of the Central Java province. The city covers an area of approximately 374 square kilometers. The coastal area of Semarang city is around 5,000 hectares, approximately 2% of all the covers in the Semarang city of area. This is the area, especially in the coast, have an opportunities and strengths to development plan. This one in your screen shows the suitability map of industrial land with a special plan for 2020. Semarang is developed by government. So the increased development launched by the city government of Semarang through long-term development has created obstacles that can reduce the benefit of development outcomes, which are pretty high with lower adaptability. So with the existence of these vulnerability zones, it is very necessary for the government and for another subsidiary to identify the feasibility factor of the soil in an area to represent the seismicity hazard of infrastructure development. In this term, seismicity is an essential factor in contributing to the vulnerability of the area. For existing topography and subsurface condition in the term of geological setting, Semarang consists of 2 morphological units, and the southern part is composed of hills, while the alluvial coast of the plain is located in northern part. It includes alluvium deposit, volcanic product, and sedimentary deposits. The center of the land, mainly in the heart of Semarang city, is basically sedimentary layers, and the majority of volcanic products are found in the south of Semarang, basically in the part of the hills. So the most recent layer are from Semarang coastal plain and were formed in marine environment for the coastal zone actually. In the term of disaster mitigation, especially in the coastal area within in the term of earthquake resistance, for building and infrastructure, usually require earthquake hazard map at ground surface. So one of the required parameters for determining the ground amplification factor is the average shear wave velocity in the upper depth for 30 meters from the ground surface, as we call with VS30. The speed of shear wave is very important parameter to use. It represents that the greater the value of shear wave velocity, the more soil stiffness value, which means the soil is getting harder and denser. The vibration caused by earthquakes tend to amplify in soft ground compared to hard soil or rocks. And the layer of the depth of 30 meters determine this amplification of the waves. So ideally, shear wave velocity measurement is conducted directly in the field with geophysical methods or maybe another passive and active method to determine VS30 value that is property of wave propagation. However, by the reason of the practical, there are also empirical formulas that available to correlate with another geotechnical parameters, such as QC from cone penetration test or from NSPT from standard penetration test. As we know, the SPT test, the CPT test, cone penetration test, is using to determine the geotechnical engineering properties of soils. And delineating soil stratigraphy consists of it use the method to pushing the instrument, the cone instrument, with the tip of facing down into the ground at the controlled rate. Meanwhile, the SPT, standard penetration test, is an in situ dynamic penetration test, as well as designed to provide information on the geotechnical engineering with the thick well sampling group with a sampling method. So this study have a purpose to do a comparison between VS30 data derived from the derivative data on SPT, both SPT and CPT. So that by the results, the relationship between the two will be obtained and the optimum alternative method can be proceeded. So the data used this study consists of two types of database of data. Shortly, the first one is VS30 data derived from an SPT by the year of 2016. And this data have been obtained from the literature available for spatial comparison. It covers 256 sites as of VS30 contour maps. And the range obtains along a range in between 100 and 450 meters per second, which divided into 7 classes. And the other database for the comparison is data acquisition for average year. This data then converted to shear wave velocity as a lab result. And this data represent the shear wave velocity calculated with the equation. So the sample data were used as an independent variables, as well as CPTU2, 3, 6, and 9 on 2011. So the method is simply use a comparison model from the derivative of the mathematical function of CPTU for them that can extract the VS30 data against the reference data. The analysis and results from the data obtained, the sample point of study area spread in the base map, you can see off the screen, as dependent variables. If we are referring for the geological map of Semarang, the research area is in the area with majority of alluvium formation deposit, and is included in soil class with D class with value of shear velocity range from 160 until 360 meters per second. This base map shows that the VS30 map based on CPTU point is well correlated with the VS30 zone as a reference lab. And all this distribution of CPTU data has overlaid with various contours of the VS30 value. And from the extracted value in 4 sample we choose from the shear wave correlation data against is depth, the value of shear wave increased with depth. From the correlation results, the maximum value of shear wave at 30 meters from the CPTU2, you can see in the red rectangle area in the first one graphic, is about 225 meters per second. And then the second sample one is CPTU3, is about 202 meters per second. The third sample for CPTU6 is 273 meters per second. And the last sample point is the CPTU6 that have the value of shear wave around 216 meters per second. So the value of VS30 derived by CPTU and NSPT is still included in the spatial range value. As shown as table, you can see in your screen, the tables, especially for sample point CPTU2, 3, and 6 included in VS30 range by NSPT derived data extract. On the other hand, the point of CPTU9 produced a bit out of range of VS30 and SPT. This phenomenon can occur due to data interpolation and the lack of proximity of the NSPT point. To clarify the different VS30 result, it's advisable that in the future studies, it is necessary to compare with NSPT data directly, or another example for geophysical method such as passive and active method. So the summary for this kind of study based on data we obtain, the comparison between the value of shear wave on the 30-meter depth derived by CPTU and SPT data can be performed. Most of the damage from the earthquake is what we call about amplification, especially in the thick alluvial area, and it's located in the north of Semarang. Is clearly dominated by softer lithology of the area. Based on established premise of the study that influence at most 30 meter and shear wave velocity is an indicator of best stiffness. As the comparing data within VS30 derived by CPT and SPT from shear wave value, the results can be drawn that the VS30 from CPT of 4 data terminally has the value within the range of VS30 from an SPT reference. So that the cone penetration test or CPT is passive, relatively inexpensive, and offers in-situ soil behavior type for instantly during the acquisition of data. Then all of data in the field gathered can inform geotechnical design and field characterization decision immediately. And it's also have fully versatile to obtaining shear wave velocity. Thus, it can be conclude that the comparison of both VS30 data indicates almost the same value range within contour map and the discrete value. The shear velocity indicates that the area around the CPTU6 with high value represent more consolidated soil than another 3 sampling points. This the area have less amplification for maybe another seismic wave or we call earthquake wave. And this kind of analysis end up with the building recommendation of design and planning for managing, especially for the coastal area in many places in Indonesia, especially for Semarang in terms of this study. So that's all for my presentation. I hope you can understand what I mean, and I give back the time to moderator. Thank you so much.
Okay, thank you, Mr. Ahmad. Actually, your time is finished, but we will give the chance for Mr. Rizky to ask very short, and you can answer.
Yeah, thank you.
Sorry, Miss Rizky.
Yeah, it's okay. Okay, thank you for your presentation, Pak Solmim. Yeah, I want to ask about your explanation. You mentioned there is a wave propagation method for determining the VS30 beside SPT and CPT, right? So could you explain what kind of the method for that case? Thank you.
Oh, okay, I got the point. Thank you for the question, Mrs. Rizky. Okay, in mapping hazard and approach to wave propagation, as well the previous slide, I mentioned that there is wave propagation method and SPT, either CPT methods. And based on your question, you asked about what kind of wave propagation method. Basically, with me majoring in geophysics, there is a micronization for determining the properties of shallow area in the development zone, especially for infrastructure and building. So the common methods, you can choose either multi-channel MASV. We can choose either MASV or HFSR, multi-channel analysis, surface wave, or horizontal or vertical spectral ratio. That was the common methods to do and proceed this approachment by determining VS30 properties in the area. So maybe, yeah, this study maybe approach with CPT, not for geophysical methods, maybe for passive and active methods for survey area. Is it can be clearly, you got the point?
Yeah, thank you very much.
Okay, Ms. Rizky for the question, and Mr. Ahmad for your answer. And we are sorry due to the time, so unfortunately we could accommodate question for Mr. Cahyo Nurfatan. So maybe please ask directly to the Mr. Ahmad. Sorry for that. Okay, we are finished all the presentation in this session. So on behalf of the committee, thank you very much for your attending and your presentation in Marine Geoscience session. So we hope we will having you again in the next symposium. So actually I have the information from the committee. So for all the presenters, so please take into account regarding to the similarity score of your manuscript. Make sure that your revised version of the MS Word has the score lower than 15%. 15%. Otherwise, the IUP will individually withdraw it. So thank you very much for your attention. So please don't leave the room now, because we would like to take a picture for all the audience presenter in this session. So I will ask the Ms. Rafida to helping me for take the picture. So please turn on your camera and give the best smile. Maybe Miss Rafida, you can guide us or something like that.
Okay. Smile.
Sebenarnya ketutup masker.
One more, one more. Okay, thanks. Back to Mr. Zain.
So thank you very much for your participation and attending. So please don't go anywhere. We would like to play the video for our invited speaker, Prof. Yu, and then we will have the closing remark after that. Please enjoy the video.
Okay, so sorry for this inconvenience. So I try to make a recorded video and share with you. Today I would like to share some information on the monsoon connecting the western and eastern Indonesia, and also the north-south hemisphere as well. My talk is mainly from the point of view of inter-seasonal time scale. First, look at what is the Asian monsoon. So when we look at a global scale, the Asian monsoon is the strongest and the largest circulation system over the Asian region. It's connecting the southern hemisphere and northern hemisphere. So many countries are influenced by this Asian monsoon, including the Indonesia and China. And this is the Asian monsoon rainfall. So the red color means the rainfall occurs in a borrowed summer season, and the blue color means the rainfall occurs in a borrowed winter season. So this is the case for Indonesia. Your December, January, February is a rainy season. And before I talk to the real work, I thanks a lot to my Indonesia colleagues. I working quite a long time with them. So I share, this is my first cruise with Agus, this 15 years ago. And after that, it's a lot of a cruise working with many Indonesia friends there. So I really thanks many Indonesia friends. And this is one of the example of the cruise in Eastern Indian Ocean. And my past 10 year or 15 years experience is in a Eastern Indian Ocean part. And this conference gave us an opportunity to discuss the eastern part of Indonesia water. So that is an amazing and very exciting opportunity. So why I say it's the eastern part of Indonesia is a very exciting place. Because when you look at this global ocean, tropical ocean observing map, during the past 2 or 3 decades, a big progress, but still some gap. When we look at South China Sea, eastern part of Indonesia water, and low latitude Western Pacific, this is a very huge gap region. We need to work together. And I really hope this conference will promote the collaboration over this gap region. And for this gap region, there is one process that is very important. That is a monsoonal interseasonal oscillation. Monsoon has 2 kinds of interseasonal oscillation in the different seasons. In a borrowed winter season, the interseasonal oscillation is normally called Madden-Julian oscillation. This is discovered by the Madden and Julian. And in a borrowed summer season, the interseasonal oscillation has a different propagation orientation. This northward propagating interseasonal oscillation is called BCSO. So MGO is propagating eastward, and BCSO is propagating northward. They are 2 totally different directions. And they are the totally different season. As I mentioned, MGO in a borrowed winter, and BCSO in a borrowed summer season. Yeah, this I already mentioned the Madden and Julian discovered MGO some decades ago, and I would like to draw your attention to a earlier paper by Chinese scientist Xie Yibing. He discovered the interseasonal oscillation in 1963, so that is why we call now XMGO. This is a movie, a lovely show MGO propagation eastward from the tropical Indian Ocean. The red color and blue color represents a dry condition and wet condition. So you can say the dry wet alternative phase is occupy Indonesia. Yeah, you will for sometimes there's several weeks of rainfall, flooding, and following on several weeks there is no rain at all. That is very dry condition. So it's MGO in its extreme case can bring a lot of trouble. For example, in Jakarta city, I know December you will risk a flooding time in that period. So that is associated with this Madden-Julian oscillation. And with the propagation of this Madden-Julian oscillation is connecting the western part of Indonesia and the eastern part of Indonesia. And much beyond that, it's connecting the Indian Ocean and also the Pacific Ocean. And this is a 3D structure. You can see the very large scale convection system moving together. And why this Madden-Julian oscillation is so important, because it's when we look at this map, and nowadays we have a weather forecast, and we also have a seasonal outlook that is a climate forecast. But between them, there is a gap we are trying to address. That is a S2S prediction. So the prediction scale is still very low. And also, MGO is not a local process, not influence Indonesia alone, but has a global impact. So that this is a teleconnection map. You can see the South Asia, Southeast Asia, East Asia, and even North America are influenced by this Madden-Julian oscillation. So that is why we see a lot of campaigns are driven by this scientific concern. The one of them is 15 years ago is MISMO, is led by the Japanese team. I would like to say this for this MISMO experiment, the Indonesia contribute a lot. You can see the Indonesia research vessel, I think Barunajaya 3, it's joined this expedition. And following that is a Serena project. This is led by the French team. And afterwards is a Sandy Dynamo project. This is led by the Japanese and US team. So it's Indonesia all play a very important role for this international campaign. The most recent one is a Year of a Maritime Continent. Indonesia has played the leading role, and BMKG is the leading agency for this campaign. And you can see we have a lot of outcomes from this big campaign. But I would like to draw your attention for this project. Much focus is still on the western part of Indonesia. And in the eastern part of Indonesia, the observation, the process study is still not too much. So it's still, we need to promote the Year of Maritime to develop to the eastern part of Indonesia and all the way to the western Pacific region. So eastern part of Indonesia is important, is reflected in this map. It shows the interseasonal variability, SIT amplitude. You can see the large amplitude SIT variability is in the Banda Sea, in the Timor Sea, in the eastern part of Indonesia water. So why this SIT is important? So later on, I will show you. And we know the maritime continent have a very complex topography, and this topography sometimes can block the Madden-Julian oscillation from eastward propagating. And that is why we divided the Madden-Julian oscillations. Normally, we can divide it into two groups. One group is blocked by the topography over the maritime continent, and the other group, they can survive. They can go through all the way to the equatorial Pacific region. They can cross a date line. So this is two different group, one block and one crossing. And this is another way to say the block group and the crossing group. I draw a red line there. This you can say the block group, they could not go through into the Pacific region. And the crossing group, they can all the way go into the date line. With this map, I try to emphasize the importance of SIT. So the color shaded is SST anomaly. And when you compare the blocked group and in a western part and the crossing group in the east part, you can see the SST signal is different. Once the MGO crossing the maritime continent, that means in the eastern part of Indonesia, the temperature is much higher. But if the MGO is blocked over the maritime continent, so you mean you can see the SAT is relatively lower than the crossing group. So that means the SAT condition really modulates the Madden-Julian oscillation behavior when it's cross over the maritime continent. If the SAT temperature is much higher, and at this time the Madden-Julian oscillation is very likely to cross over the maritime continent into the Western Pacific region. And our model, how well our model now is the conclusion, the model is still very challenging to reproduce such kind of Madden-Julian oscillation. So that means our forecast or prediction skill is still low. And another example is the importance of Madden-Julian oscillation. It can trigger the Indonesia Australia monsoon. So this is a paper working together with the Indonesia team. You can see the first propagating Madden-Julian oscillation really triggered the Indonesia monsoon. So for Indonesia, your monsoon onset is always triggered by the first eastward propagating Madden-Julian oscillation. So that is why we need to pay more attention. And when we look at this monsoon onset process, so the shaded is SAT color. So you can see before the monsoon onset, the SAT, the maximum SAT is over the maritime continent. So that means the maritime continent SAT reaches peak. This is a precondition for the monsoon onset. So it's a very strong signal indicator the monsoon is almost onset there. And I think I skipped there. So it's when we look at the observation map again, so I want to emphasize the South China Sea, the eastern part, Indonesia water, and the low latitude, the Western Pacific is a huge gap. And this huge gap there means we have difficulty to understand why some MGO is blocked by the maritime continent and why some MGO is can cross the maritime continent. And I didn't talk about the borrow-summer intra-seasonal oscillation. It's a similar story. So some can propagate well not to the northward, and some didn't, becoming quite weak. So for this gap region, we need to working together, try to design carefully the field campaigns, collecting data, and try to understand the process and try to improve our coupled model there. And Indo-Pacific warm pool is a huge water with a warmest temperature there. So that means for over this Indo-Pacific warm pool, the strong air-sea interaction is a center for our weather and climate. And the east central part of this Indo-Pacific warm pool region, we really need to address a lot of scientific questions. And finally, this is my slides, try to summarize and bring some take-home message. So 3 messages. First one, for the Eastern Indonesia, this is a corridor for the MGO propagating from the Indian to the Pacific Ocean in the borrow winter. And also this is a corridor for the borrow summer intra-seasonal oscillation propagating from the southern hemisphere to the northern hemisphere in the borrow summer. And this is kind of a propagating, it's a very interesting process. As I mentioned, the blocking and the crossing mode are totally different. Why some cases is blocked, but some process can cross. It heavily depends on the SAT condition over the eastern part of Indonesia waters. The second information is MGO and BCSO provide the physical basis for the subseasonal prediction. And this is very important for the disaster reduction. I gave the example of big flooding normally occurs in December, January in Jakarta. So this is a very much associated with a convection phase of the MJO. But this process is fully reproduced in our models. So this is a very challenging issue we need to address. The last point I want to share with you is ocean upwelling is very significant over eastern Indonesia seas. I didn't give the details, but this upwelling process is very important for the ecosystem and the fishery there. I heard an excellent presentation this morning talking about the biodiversity, the new species in the oceans. And this habitat, this biodiversity is supported by the physical process, including the upwelling process. We need to really understand the physical process, how to support biodiversity, how to support the habitat there. So I think eastern part of Indonesia Sea, South China Sea, and low latitude Western Pacific is a region of a huge gap, and we need to work together and try to address a lot of scientific issues to make our ocean support a sustainable development of our society. So I think I will stop here. And finally, to thank all my friends from Indonesia supporting us during the past 10 to 15 years, develop a lot of field campaigns. I really thanks all of you. Thank you.
Yeah, thank you very much for.
Okay, Pak Zain, back to you.
Oh yeah, thank you, Mr. Fadli, for playing the video. So thank you very much for all of you to attend this ISEIME 2022. So we are hoping we will meet you again in the next ISEIME in the next time. So have a good time and see you next time.
Thank you all.
So now you can leave the room. We are finished already. Thank you very much and see you next time.